A very big hello to my wonderful champions. So, hi Rishi. Rishi is there. Hi Troll Samrajya. Okay, that's a very different name. And yes, ma'am, please come live. Yes, ma'am is live now. And how was your physics paper? I hope you guys are asking to each other, because I have no physics paper to give. Yay! So my dear children, we will just wait for some time. How was your physics paper by then? We'll wait for some time so that everyone else can also join the live session. And if at all you have your friends who have taken biology and who feel to be struggling in biology, please ask them to join this particular live. Because, hi Sinchu, hi Sanskar, everyone is there. Hi. So yes, please share this video with all of your friends as well. Because, why? Because this video will make sure that every aspect of your biology is being covered. Okay, so we are going to focus on the chapters starting from sexual reproduction, flowering plants, going up to evolution, my dear children. So everything is going to be covered. I am from PU2 crash course. Nice. Pradeep, hello dear. Rock gamer, hi. A very big hi. So my dear children, I see full vigor. So can we start then? Are we ready? Just give me a thumbs up if at all we are ready. Hi Mumtaz, hi Rock Gamer, hi Pradeep. So all of you, I want all of you to be here throughout the video. Do not go anywhere. So no one can stop you from scoring full on full in biology, because it's you and me who is going to study. Starting with the first chapter that we have, it is sexual reproduction in flowering plants. We'll deal with the 1 marker first, then we'll go to the 2 marker, 3 marker and 5 marker. Agreed? Done? Yes. The first question that we have is, pollination does not guarantee the transfer of right type of pollen. Is it true or false? Thank you Mansi, thank you Mumtaz, thank you Sinchu, thank you Tanish for giving that. Yes. So pollination does not guarantee the transfer of right type of pollen. Yes. Does it guarantee? You tell me the answer. Statement 2. Compatible pollen-pistil interaction guarantees double fertilization. Compatible pollen-pistil interaction guarantees double fertilization. That means if pollen-pistil interaction has taken place, obviously one gamete is going to fuse with the female gamete and another gamete is going to fuse with the the polar nuclei. Does it guarantee that? What do you say? Answer is A, B, C or D. Hi cutie ma'am. Hi cutie dear. Okay. Manasi says the answer for this question is C. Statement 1 is correct, but statement 2 is not correct. Both are right. Okay. Rishi says the answer is C. My dear children, let me tell you, even if this video goes long, the live will definitely go long. I am going to be here with you throughout, but I guarantee you your concepts will be clear. So be there. Okay. Pollination does not guarantee the transfer of right type of pollen. Yes, correct. Any type of pollen can come. It does not guarantee. Next thing, compatible pollen-pistil interaction guarantees double fertilization. No, it does not. How can it guarantee double fertilization? Even if pollen-pistil interaction took place, okay, suppose on the stigma some pollen has landed and the pollen-pistil interaction took place, pollen tube has come. My dear children, there is no guarantee that this pollen tube can reach the ovule. Do you know that? Multiple pollen tubes can be formed, but only few will reach. So basically this particular statement would be wrong. I cannot guarantee it. So your only answer for this question is what? C. Yes. So Mansi and Rishi and Shalam and Tanish, all of you are getting your answers correct. Shabina is asking how much marks come from this session in bio. I will say, so basically I have taken 7 chapters, my dear children. So Shabina, the 7 chapters, whatever marks these chapters carry, you will get that marks. Okay, just that you need to be 101% efficient with answering the questions. Sanskar ma'am, all chapters are covered today. No, just from sexual reproduction to evolution. Part 2 will be there. That will be a recorded video, because I know you do not have much time. At least the high-weightage chapters, I will be there for you. Okay. And Vinay King, all student, like the video. Yes, like the video. Let's go ahead with the second question. Statement 1. Apomixis is the production of seeds from unfertilized ovules. Is it? Apomixis is the production of seeds from the unfertilized ovules. Statement 2. Embryos produced from apomictic seeds are not generally identical to the parent plant. What do you think? Apomixis is basically when, suppose this is an ovule. Okay, here you have the female gamete. Usually the female gamete, you know what? How much is the amount of, chromosome it has? It has only haploid content of the DNA. Rest haploid content comes from the male gamete. But over here in apomictic seeds, this particular female gamete is already diploid. It is not haploid, rather it is diploid. Now do you think it needs another N? No. Now since it is already diploid, it will transform into what? A zygote. The zygote later forms the embryo, and it's all done. You do not need any other parent plant. So apomixis is the production of seeds from unfertilized ovules. Yes. So whenever zygote is formed inside the ovule, you will now call this as a seed. This statement is correct. I am going to read your answers, okay? Just a second. Now, embryos produced from apomictic seeds are not generally identical to the parent plant. Array, you are taking one parent only, and that parent is producing same type of organism. So obviously it is going to be identical. How can you say it's not identical? This is wrong. So statement 1 is true. Statement 2 is what? False. And now let me see who all got your answers correct. Okay, so okay, Isri is there saying A, Manasi A, Jayashree A, Jnanavi A, Shalam also A, Vinay King okay A, everything is super easy. Ma'am, the annual paper of bio is easy. Yes, let's assume that. Okay, Tanish also A. So I hope it is clear. This is from model question paper. Do not be worried. So I'm not saying that you should just focus on model question paper. You should focus on other questions also. So we will solve all of it. Do not be worried. Now question number 3, even in the absence of pollinating agents, seed setting is assured in. Try this question. SFG, ma'am, hi ma'am, I am an old student of 2024-25 from you all teachers. Now I am in RVC college with rank 185. Myself Ajay. Oh my God, that's so great. Congratulations to you. You are so, I'm so proud to see you over here, and thank you for coming and commenting done. I hope you are doing good. Okay. Try this question, dear. Even in the absence of pollinating agents, seed setting is assured in. Children, if at all you can you tell me the answers 3A, 3B, 3C, 3D? That will be even more easier for me to understand which question you are solving. Yes? Can you try? That means here you do not need pollinating agents. Still, seed is going to be formed. Seed pollination is going to be happening. Fertilization is going to be happening. Everything. Cucurbita is what my lamb says, and Jayashree says A, Jnanavi says C, Mansi says G, Vinay says C, Mansi says 3C. Yes, that's how you should answer. And yes, Commelina. The answer for this question is Commelina. You guys are correct. So Commelina, Cucurbita, Commelina, oxalis and viola. Commelina, oxalis, viola. Cow, you can remember like that. Not cow, but yes. Commelina, oxalis, viola, my dear children, they are having clistogamous flowers. That means closed flowers. Here you have male and the female parts. From here to here things will go. You do not need any pollinating agents. Okay, so this answer is correct. You guys are correct. Those of you who gave me the answers, you are correct. So this is the closed flowers that we have. These flowers are usually towards the root or just above the root, and the open flowers are at the top. Okay, so clistogamous and chasmogamous flowers. Going ahead, solve question number 4. This is not from your model question paper. This is from question bank. Select the correct sequence of anther wall layers. Can you solve this up? Shalam says C, and Janvi also said C, Tanish also said C, Rishu also said C, Shabina also said C. Good. Awesome. And my dear children, as Vinay said, do not forget to hit the like button and share it with your friends also who are studying biology, and tell them, come attend JJ ma'am's class. It is going to be helping you. Okay? Chalo. Select the correct sequence of anther wall layer. 4B. Okay, 4B, my lamb says 4B. 4B is what? Tapetum, middle layer, epidermis, endothecium. What? Anther wall layers, can it be 4B? First thing, my dear children, you go to a doctor, ENT doctor for ear, nose, throat, right? Similarly, over here, you have EMT, not NT, EMT. E for epidermis, E for endothecium, M for middle layer, and T for tapetum. So, EMT. So, epidermis, endothecium, middle layer, tapetum. Your answer for this question would be B. Okay, your answer for this question is B. Who gave me the correct answer? It was Mansi, My Lamp, and Jnanavi, Alina, Janvi. Oh my God, so many people. My Lamp again. Jayashree, Prajwal. Okay, great. Ma'am, I said B. I'm so sorry. Okay, ma'am, I said B. I'm so sorry. Okay. Ma'am, giveaway of iPhone for rank below. Okay, so your answer for this question is B, and you guys are correct. Those of you gave me the answer. And always remember, 3 of these layers are responsible for dehiscence of anther. When the anther breaks down, my dear children, for the breakdown of anther, you need these 3 layers. T, tapetum is for nourishment of the future pollen grains. Nourishment of the future pollen grains out there. They are having dense cytoplasm, and they have 2 nucleus. They are binucleate in condition. Dense cytoplasm and binucleate. Always remember that. Okay, Next question, question number 5. Select the correct sequence of events in microsporogenesis. Try this question out. Try this question out. Select the correct sequence of events in microsporogenesis. Microsporogenesis. What exactly happens over here? In board exam, they will sorry. Ma'am, maths and chemistry is what? Puttir Gowda, I'm so sorry dear. Maths and chemistry, maths is soon gonna come. Chemistry is over. Hello, Shadow. Okay, try this question. In microsporogenesis, what is going to happen? Microspores are going to be formed. These microspores are going to form what? Pollen grains later on. So first of all, what do we have? We have microspore mother cell. We have microspore mother cell. Okay, this microspore mother cell came from where? From the sporogenous tissue. Sporogenous tissue is made up of multiple cells, and you know what? All of them are MMCs. All of them are MMC. You are saying the answer for this question is A. Let me see. Now, MMC, my dear children, it undergoes meiosis. It undergoes meiosis. One gives 2 cells. They are haploid, because it underwent meiosis. Now, meiosis 2 happens, my dear children, and now you have got 1, 2, 3, 4 microspore tetrads. And these microspore tetrads will separate and basically give me pollen grains. So this is how things should go. So your answer for this question would be A. Sporogenous tissue, microspore mother cell, microspore tetrad, and then microspores. Microspores, tetrad, microspores, and then your pollen grain. So whatever you said is correct answer. Sanskar, all MCQ from annual came from the question bank itself. Is it? Hello, hi, I'm 02. Hi, dear. Okay. So this is how it goes. Microspore mother cell is there, meiosis 1, meiosis 2, microspore tetrad is being formed, then microspores, and then pollen grain. Try to solve question number 6. Let me read out the names of the people who are giving me correct answers. Mansi gave me the correct answer, Shalam, Alina, Rishi, and Vinay gave me the correct answer, Janvi gave me the correct answer, Purvika gave me the correct answer. Namaste, ma'am, I'm 02. Okay. Namaste, dear. Chalo. Try to solve this question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. Can you tell me the answer for this question? Ganesh, please subscribe to our WhatsApp channel and you will get the PDF there once this particular video has been done. Okay? Answer is C. Answer is C. Is it answer C? So you have to find what is true for a typical female gametophyte. It is 8 nucleate and 7 celled at maturity. Is it true? Yes, ma'am, it is true. There are only 7 cells. So if at all we see, my dear children, do I have a picture? Let me see that first. Yes, I have. So over here, if at all I see this particular embryo sac, first of all, you need to understand 1 megaspore will be there. 1 megaspore will be there having 1 nucleus. The nucleus keeps on dividing. How many times? Thrice. So once it divides, you have 2 nucleus. Then 4 nucleus. Then you have 8 nucleus. Until now, it was free nuclear division that was taking place. It is now forming the embryo sac. From the megaspore, it is now forming the embryo sac. Over here, it underwent 3 divisions, 3 mitotic divisions. You got 8 nucleus. Now this 8 nucleus, I have to arrange that in the embryo sac. 3 of the nucleus is covered by the cell wall and you get the antipodal cells, 3 up there. And basically, 2 of them come over here, they are known as synergid cells. 1 more cell come over here, this is your female gamete, that is the egg cell. Now 2 more nucleus is left, they both come over here and they are known as polar nuclei. So how many cells are there in total? 1, 2, 3, 4, 5, 6 and this one is 7. But how many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus, 7 cell condition and it's a free nuclear division. There is no cell wall formation happening. Okay, there won't be cell wall formation happening over here. Mayank, will this be sufficient, ma'am? So you have to stay consistent with me throughout and I guarantee you at least I will be covering these many chapters out there. Okay, then if at all you want, you can go ahead and read your NCERT just for your revision. That's all. Fine. So your answer for this question, let me go back first of all. Just a second. Okay. From the statement, it is 8 nucleate and 7 cell at maturity, correct. It is free nuclear during development, correct. It is situated inside the integument but outside the nucleus. This we do not know, ma'am. So basically, my dear children, ovule is there. Ovule has integuments. Okay, inside the ovule only you have the cells of the nucleus. Your embryo sac lies within. Your embryo sac lies within the nucleus and obviously within the integument also. So this statement is wrong because we are sitting outside the nucleus. Now it has egg apparatus situated at the chalazal end. So my dear children, always remember there is a place where you have a pore where there are no integuments. You call this as micropilar end. And the opposite side which is there, you call it as chalazal end. So over here, the egg apparatus is situated at the chalazal end. Do you think it is situated at this end? What is egg apparatus? This particular thing that you saw right over here, this thing that you saw is egg apparatus. It is never situated in the chalazal end. Rather, it is situated in the micropylar end. Always remember, everything important happens in the micropylar end. Everything not so important happens in the chalazal end. Okay, megaspore division starts in the micropylar end, and yes, everything in the micropylar end. Okay, so over here again, if at all I come back, it has an egg apparatus. This would be a wrong statement. It is not at the chalazal end. So A and B are the only correct answer. IM02 was the first person who gave me the correct answer, I believe. Is it? Let me see. So yes, now going ahead with the next question. Try this question. Question number 7. Identify the correct sequence of arrangement of cells in the mature embryo sac, micropylar end. Okay, now let me read out the people's name who gave me the correct answer. Ma'am, duration is going to be long. You know what? I told at my home that I will be coming at 9 o'clock. It is going to be long. Why? Because I am completing all the chapters, right? So yes. 02 answer C, 6C. Okay, Sanskar. Okay, dear. Vinay gave me the answer. Jayashree gave me the answer. Rishi, Baba. My Lamb gave me the correct answer. Kirtana gave me the correct answer. Mansi gave me the correct answer. Okay. Now 7A. Try this. Mansi says 7th answer is A. Okay. Identify the correct sequence of arrangement of cells in the mature embryo sacs, micropylar end. Micropylar end. Okay. In the micropylar end, first thing that you have is synergids, then you have X cell, then you have central cell, then you have antipodals. So if at all you have confusion, look over here. This is the micropylar end. First synergids, then X cell, then central cell, then antipodal cells. So please be sure if at all you're reading your questions correct or not. Well, the answer for this question is A. So Mansi, Jayashree and Shalam and Deekshi, okay, Deekshita. Deekshit also got the answer. Okay, Shalam, Kirtana, Mayank. Oh my God, it's too difficult to read your names. You know what? But you guys are beautiful names. Anyways, so you guys are doing awesome. So let's start off with the next question that is there. Question number 8. Statement 1 says, in viola, both autogamy and geitonogamy is prevented. In viola, both autogamy and geitonogamy is prevented. What do you think so? Answer is A. Deekshit. Answer is A. Yes, the previous answer, it was A. Good. Try out this question. My dear children, I'm telling you again, if at all you can, please tell me the answers like this. 8A, B, C, D. That will be more easy for me. Okay? In viola, both autogamy. Autogamy means what? Self-pollination, my dear children. And viola has closed flowers. So self-pollination will definitely happen. Who says that it can be prevented? Now, geitonogamy is what? Geitonogamy is pollination between the flowers of the same plant. Now, here, in the case of viola, they have open flowers and closed flowers. In the closed flowers, obviously, autogamy is going to happen. In the open flowers, again, autogamy can happen. Geitonogamy can also happen. So, how can we say that this is prevented? So, this is wrong. Statement 1 is wrong. What about statement 2? Statement 2 says geitonogamy is similar to autogamy since pollen grains come from the same plant. Geitonogamy, which is this, is similar to autogamy because pollen grains come from the same plant. Yes, in that aspect, it is definitely same. In that aspect, it is definitely same. There is no genetic variation. So, statement 2 is correct. Statement 2 will be correct. Statement 1 will be wrong. So, your answer for this question is 8C. Let me see. Statement 1 is incorrect, but statement 2 is correct. Yes, my dear children, your answer would be C for this particular question. So, this is geitonogamy. The thing that is happening within is autogamy. If it is happening between two plants, it is xenogamy. It is true cross-pollination. Okay? I will read out the name of the children who gave me the answer. Let's solve this question. Try out this question, my dear. So, Mansi and my lamp and lip is there. Okay. Kirtana gave me the correct answer and Kirtana, dear, please check the answer. My lamp gave me the correct answers. Mansi gave me the correct answer. Shalam, Prajwal, all of you gave me the correct answer. For those of you who are getting your answers wrong, please not be worried. Try it out one more time and you will get your answers correct. Okay? You guys are rock stars. Do you know that? Now, try this question. 9th question. The following picture is an example of. What is this picture of? First of all. And what is happening up over here? This picture is of Papaver somniferum. Papaver somniferum. And what exactly is happening up over here? Any idea? Any idea? Mansi says answer is B. Okay. And Prachi says B. Lippo says B. Okay. Jayashree also says the answer is B. Tough competition. Deekshit also says the answer. Deekshit says the answer is C. Shalam says B. Okay, let me see. So over here you see that multiple carpels are there. These things that you see are what? Multiple carpels. So there is multicarpellary condition for sure. But the thing is, all the carpels, my dear children, will be having only one ovary. So they are all fused. And you call this fused condition as what? Syncarpous condition. What do you call this as? Syncarpous condition. So they are multicarpellary and they are syncarpous, and this is gynoecian, that is the female reproductive system. So you guys are correct when you said the answer for 9 is what? B. You guys are absolute rock stars. Can you give me the example of multicarpellary and apocarpous gynoecian organisms? Apocarpous means basically they are all having separate ovary. Apocarpous is seen in another organism which you call as Michelia. Another plant which you call as Michelia. Please remember it. This can also be a question. Okay, so this is the condition that is being shown over here, and this particular flower, you might have seen it. It is known as Champagam in Malayalam, Champak in Hindi. It smells so good. So basically over here you have multiple carpels and all of them are separate, my dear children. All of them will be separate. Yes, Lip, it is Michelia. Okay, so this is Michelia and this is your Papaver somniferum, where you get the drug opium. Okay, now we are going to get ahead from Gujarat board Gujarati. Arey wah. Okay. Okay. So, my dear children, listen to me carefully. Now, I am going ahead with one of the biggest tasks, that is to solve 2, 3, 5 marker. Now, here I want your 101% attention, because obviously I know interaction is going to be less and study is going to be more. So, please be careful, attentive with me, and do not leave and go somewhere, okay? Because it will be really saddening for me. Now, write the significance of sporopollen. This is a model question paper question. What is the significance of sporopollen? It is the toughest organic material, substance that we know. And basically it forms the exine of the pollen grain. The pollen grain have multiple layers, right? The outermost layer that we have, it is known as exine, and exine is discontinuous. There are certain spores in the exine. You call the spores as germ pores. You call them as germ pores. Okay, and rest is made up of sporopollen, and since it is the resistant material, it is the reason why pollen grains can be stored as fossils. Okay, pollen grains are stored as fossils. Lip is asking what drug? Okay, Papaver somniferum gives opium drug, that is your heroin, morphine, all of it. Okay. So this is your answer for the significance of sporopollen. Please write your answer in points. Otherwise, you might be losing up your marks. Do not do that. This is a 2-marker question. Do not mess it up. Now, I will be going ahead with questions, but if at any point you face any doubts, please ask me. Now, there is another question in MQP3. Micropyle remains as a small pore in the seed coat of the seed. So, if at all you see, my dear children, wherever integument is not there, I told you that is a micropyle. And why do you think this micropyle stays there when this ovule will turn into a seed? Because, my dear children, the seed will need moisture. It will need air. And from where will it get it? Obviously, from the small pore. That is why it remains like a small pore in the seed coat of the seed. So, when it becomes a seed, the integuments become seed coats. Still you will be having that pore. Why? For the exchange of gases as well as for moisture to be maintained. Okay, so how do you write the answer? The micropyle of the ovule persists even after fertilization and becomes a tiny pore on the seed coat. It helps in entry of water and oxygen during seed germination. Okay. So, coming back. Apple and cashew are not called true fruits. Why? Usually what happens, the ovary which is there, it turns into a fruit, and the ovule which is there, it turns into a seed. Okay, but sometimes it's not just the ovary but the other floral parts of the plant. For example, if you see the thalamus over here, the pedicel over here, that can also participate in fruit formation. In that case, you call these fruits as false fruit. And you know what? Apple and cashews are example of that. Apple, cashew, strawberry, all of it are examples of false fruits. But why? Apple, my dear children, it is formed not just of the ovary but also of the thalamus. So true fruits develop only from the ovary after fertilization. But in apple and cashew, the edible part develops from the floral parts other than the ovary, like the thalamus in apple and pedicel in the case of cashew. So if at all you see this picture, okay, here you have the seed, here you have the ovary. Until this particular point, it is the ovary. But outside that, this thing that you do crunchy and eat it, what is that? It is the thalamus. Always remember. Okay. So, hey, beautiful ma'am. Thanks, dear. Okay, you're also beautiful. going ahead with the next question. Differentiate between pericarp and perisperm. Before that, are you understanding what I'm teaching you? Can you give me a thumbs up? Can you give me a thumbs up right over here in the chat session if at all you are understanding what I'm teaching, or it's just me who is teaching and you are not understanding? Is it like that? Yes, my lamb, because thalamus contributes, so it is a false root. Exactly. Awesome, awesome. My children are so great. Okay. Can we go ahead? Give me a thumbs up, all of you. No? Okay, let's. Thank you, Mansi. Thank you so much. Now, differentiate between pericarp and perisperm. So, my dear children, I'll tell you one very easiest way to explain pericarp. So, over here, you know mango, you have eaten mango, right? The peel that you peel off, it is what? The outer layer of your ovary, which is you call as exocarp. Exocarp. Another thing that you eat up, the fleshy part, you call this as mesocarp. And you might have seen gutli, the tough coat which is there, which you throw away. Okay, that is what? The endocarp. Inside the endocarp, you will have the mango seed. The seed will have its own covering, seed coat and everything is there. But this is separate covering that we have, that is endocarp. So basically, these three layers that we have, it forms together one very important thing that is known as pericarp, all the wall of the ovary. This is nothing but the wall of the ovary. So please do not worry about this. This is what you mean by pericarp. Now, what is perisperm? Perisperm is something totally different. For example, my dear children, when we studied about the ovule, okay, when we studied about this particular structure, I told you that inside the ovule you have what? The cells of the nucellus. Here you have so many cells, and these cells, my dear children, are known as cells of the nucellus. Usually what happens is that they are there for nourishment. After the embryo sac is being formed inside, these nucellus cells, they go away, they got degenerated or they get used up. But sometimes they persist. And they are there persisting for the nourishment of the embryo. So when in the embryo sac an embryo will be formed, these guys will be providing nourishment. So this persistent layer of nucellus is known as what? This persistent layers of nucellus is known as what? What? It is known as perisperm. And it is found only in two plants according to your NCERT, and that plant is what? One is beet and other one is black pepper. Beet and black pepper. So B square. So B square. Okay? Got? Here, sorry, it's not exocarp, it is epicarp. It is epicarp, okay? Not exocarp, epicarp. So epicarp, mesocarp, endocarp, and that forms the pericarp. Fine? So, ma'am, 3 mark, Haley, 3 mark would be coming up, dear. Rishi, yes, it is epicarp. Yeah, corrected it. Don't worry. And yes, will you send the PDF? I'll definitely send the PDF. Okay, chalo, going ahead. Children, please do not forget these two examples, okay? Black pepper and beet. Do not forget that. Now, going ahead, distinguish between albuminous and non-albuminous seed. What do you mean by that? Albuminous and non-albuminous seed. What do you mean by that? So basically, my dear children, I told you there are two types of fusions that are going to take place. Okay, two types of fusion takes place over here. So where will I explain you that? I'll explain you that right over here. Okay, this is known as double fertilization. Listen to me carefully. There are two male gametes that are going to come with one pollen grain. One male gamete comes and fuses with the egg. Okay, and what do you call this as? You will call this as zygote. When both of them come together, you will call this as zygote, and you call this fusion as syngamy. Syngamy. Okay, now another male gamete which is there, it will come fuse with the two polar nuclei right up over here. When they come and fuse over here, 3 nucleuses are fusing, which is why I will call this as triple fusion. I'll call this as triple fusion. And what do you get at the end of it? You get the primary endosperm nucleus. And this cell which is there, the central cell, will be now turning into a primary endosperm cell. It will be primary endosperm cell. Okay, agreed? Now, over here, what exactly happens is that to nourish this zygote, this primary endosperm cell will be forming endosperm layer and will be nourishing this zygote so that it will transform into embryo. Whenever the endosperm is still there, you call these seeds as albuminous seeds. You will be calling the seeds as albuminous seeds if the endosperm is still there. But if this embryo says that I do not want endosperm, I would want to have my own type of reserve material, that is in the form of cotyledons. So what will the organism do? It will take all the endosperm and will make cotyledons out of it. In that case, endosperm is not going to persist. In that case, what will you call them? You call them non-albuminous seeds. The one where there is no endosperm left, that is known as non-albuminous seed. Okay, so now if I go over here and if I discuss, you will get to understand things better. Albuminous seeds, they retain some endosperm even after seed maturation. So when the seed is getting matured, the embryo is being formed inside the seed, my dear children. At that point also, they will have some endosperm still left. Okay, but in the case of this, endosperm is completely used up during the embryo development. Why? Because the embryo said that I want to make my own cotyledons. And you know what? These type of plants are dicots. These type of plants are dicots. They make their own cotyledons as storage materials. Okay, they will make their own cotyledons as storage materials. Got it? Fine. Over here, the ones that do not make two cotyledons, they just have one cotyledon. Okay, that is monocots, my dear children. They come under this because they still have some endosperm left. They do not utilize the complete endosperm. Now provides nutrition to the embryo from the stored endosperm in the case of albuminous seeds. But over here, nutrition is stored inside the cotyledons, not in the endosperm. Got it? Example is wheat, maize, barley. Very easy to remember. I'll tell you. Anything that you cannot break into two easily is coming under albuminous seeds. Wheat, maize, barley, castor, okay, all of it comes under this. Coconut, all of them come under this. Non-albuminous, your bean, pea, groundnuts, you can easily break them down to two. Why? Because they are dicots. Okay, done. Ma'am, name, NG Rao. My name is Jasmine Joy. And what's your name? NG Rao. NG Rao and lip 3N. Yes, it was triple fusion. At the triple fusion, you get 3N. Krishna, ma'am, all important diagrams. Krishna, my dear, I have already made a video on fixed questions. Can you check it out? At the end of it, you have the list of all the important diagrams, which you definitely need to practice. Okay? Now, draw a label diagram of mature two-celled pollen grain. So, any diagram which is up over here, I'll explain you here itself. So, please don't worry. So, how do you make the pollen grain, my dear children? First of all, make a big cell. In this big cell, you have a very small cell. This big cell has an irregular nucleus. Here, the small cell has an elongated nucleus. What do you call this very big cell? As vegetative cell. The small cell that you have is what? Generative cell. So, two cells are being done. Okay. Elongated nucleus, please remember it. Irregular nucleus. This is the plasma membrane. Now, this cell also has another layer that is known as intine. Intine, I told you intine is made up of Priyanka Chopra. Priyanka Chopra, why? Because pectin as well as cellulose. Intine is made up of pectin and cellulose. Now going ahead, what do we have? We have exine. What is our exine made up of? Exine is made up of, tell me in the comment section, come on. Okay, Philip. Oh, nice name. Okay. So, over here, where were we? Yes, can you tell me what is the exine made up of? This is a very important question. We discussed this as well. Exine is made up of, come on. All my dear little champions, exine is made up of. Sandeep, hello dear. A very big, Sandesh, sorry, I'm so sorry. Okay, no one is interested to tell me what is exine made up of. That's so sad. I think, am I slowly coming and, okay, sporopollen. Thank you so much, Mansi. Okay, it is what? Sporopollen that the exine is made up of. It is a very important component. Never ever forget it. Please remember it in your dreams also. Okay, and wherever you have space, you call this as germ pore. Germ pore. Okay, now also remember one very important thing, and that is this vegetative cell is also known as tube cell. It is also known as tube cell. Why? Because, my dear children, the generative cell later on will undergo mitosis, divide into two, and later forms what? Two male gametes. Two male gametes. These male gametes have to move outside through what? A pollen tube. Who forms the pollen tube? The vegetative cell forms the pollen tube, which is why it is also known as the tube cell. Okay, remember that. And it moves out through the germ pore. And 60% of the angiosperms, the flowering plants, they are going to shed the pollen grains in only 2 cell stage. That means just the vegetative and generative cell. And rest 40%, what they are going to do? 40% of them are going to shed the pollen grains in 3 cell stage like this. Okay, majority is the one that is going to shed in 2 cell stage, other ones 3 cell stage. Sporopollen, sporopollen, sporopollen. Thank you so much. Okay, Shalam, Trisha, Itrisha, and Prachi, Swastik, My Lamp, thank you. Krishna and Rishu, okay. Rishi, yes. So basically, this is it. Please remember it. It's very important. And the diameter is 25 to 50 nm. The diameter of a pollen grain is this much. Done. Next, draw a label diagram of LS on embryo of grass. Grass is what? It's a monocot. It's a monocot. That means it just has one cotyledon. And this particular picture, you know what? Ma'am, this is model question paper. Baba, I have included questions from the model question paper, from the question bank, everywhere. So you need not worry. Everything is going to completed over here. Okay? Then? Okay. So this question, who all get tortured by this particular question? Can you tell me? After this chapter, Rishi is asking, can we go to molecular basis of inheritance chapter? Yes, we can. No problem. We definitely can. Okay? Yes, monocot. So over here, I'll tell you how to make this picture, okay? Make a shark's mouth. This is the shark's mouth. And then shark's hand is there. Only one hand. Then shark's tail is there. 2 tail is there. And then shark has a small fin. So another fin out there. Now make a triangle like this. Join this up. Make the triangle. Join this up. Another triangle, join this up. Okay, now what do you need to do? Extend this triangle and let it go like this and go to one tail. It went to one tail, come back and join up over here. And my dear children, make an extra triangle over here as well. What is this? This is your shoot apex. That is your plumule. Plumule is covered by what? Your coleoptile, PP, always remember. Now over here you have what? Radicle, not root, radicle. Radicle is covered by what? Root cap. And basically below it, what do you have? Coleorhiza. Where is your cotyledon? This is your cotyledon. What is the name of this cotyledon? Scutellum. And what is this? This is a rudimentary cotyledon which could not form. So this is epiblast. And your picture is done. Yes, even I also hate drawing this thing. But yes, I'm a teacher. What do you do? Ma'am, do you cover zoology chapters? I cover everything which is there. Vishnu? Okay, so Swalpa Kannadahalli Heli, ma'am. Nanu Kannadaga che. Wait. Nanu Kannada Kalita Idini. I'm still learning Kannada, which is why I understand a bit of it, but I am not able to respond much. But I am still studying, okay? Jasmine is an awesome name as well. Thank you. Okay. Draw a neatly labeled diagram of a typical anatropous ovule. What do you mean by anatropous ovule? Anatropous means ulta ovule. Okay, somewhere ulta. So usually what happens, you have the ovule. Ovule is inside the ovary, right? And ovule feels very cold, which is why it wears 2 sweater. 1 sweater, 2 sweater. And over here it does not button itself, which is why this is the micropylar end. This is micropyle. These are the integuments. Now listen to me carefully. Ovary supports the ovule or provides nutrition to the ovule with the help of a structure that is known as placenta. And placenta is connected to the ovule with another structure that is known as funicle. And the point of attachment which is there, it is known as hilum. And the opposite side of micropylar end, it is known as chalazal end. Chalazal end. But ma'am, here you are being asked about, okay, here you are being asked about what? The anatropous ovule. For anatropous ovule, what do you need to do? You need to shift this placenta, funicle and everything over here. So let's shift that. Let's shift that. So placenta, funicle, everything, my dear children, would be shifting to one particular side, and this is known as your anatropous ovule. That's all. As simple as that. Chalazal end is still at the opposite side of the micropylar end. I hope you got it. Okay? Ma'am, what is the duration of this particular class? This duration, as I said, I told at my home that I'm going to come. Okay, I am going to come by 9 o'clock, so you can assume like that. 9:30, okay? Children, Can we focus over here? Let's talk about anime later on. I know you guys are big fans of anime, but let's not do that. Okay, it's very demotivating for me to see someone talking about something else when I'm teaching, right? Okay, so please don't do that. I hope I make sense, right? Now, the next question, important question. Why are some seeds referred to as apomictic seeds? Mention its advantages to a farmer who uses them. Why is something mentioned as apomictic seeds? Why? I already told you, we studied about apomixis. In apomixis, my dear children, they are found without fertilization. Formation of seed without fertilization, because over here, the female gamete itself is diploid. It does not need any male gamete to become diploid in nature. So it forms what? A zygote by itself, it forms the embryo by itself, and this whole thing is basically now known as what? An apomictic seed. It is the apomictic seed. So the embryo develops without meiosis and syngamy, producing seeds genetically identical to the parent plant. Now the question is, mention its advantages to the farmer who uses it. So what is its advantage to the farmer? What is its advantage to the farmer? Any idea? Any idea? Baba, you only think. If at all this particular plant which is producing apomictic seeds, it is super good. It is pest resistant, drought resistant, everything is nice. I do not need to look for any other plants. I just need one apomictic seed from this plant and it's all good. So your hybrid vigor is maintained. Farmers can reuse hybrid seeds every season without loss of yield or quality, because every information has been maintained. It is cost efficient as well. How? Farmers do not need to buy hybrid seeds every year. Do they need to buy hybrid seeds? No. This particular plant is producing apomictic seeds, so there is no use of buying things out as well. Going ahead, uniformity in crops. All plants are grown from the apomictic seeds will be looking same. It will be having the same property, uniform growth, and everything will be nice. Okay? All ears on you. Thank you so much. Okay, going ahead, this is a very important question. So the previous year, you know what, children were losing marks. Over here, only a few of them lost marks, but still I remember it profoundly. That is why I'm telling you. So there was a question that farmers basically used crop improvement program to give better hybrid variety of crops. What is this crop improvement program where hybrid variety of plants were being produced? And everyone was like, oh no, I do not know this question. Is this from biotechnology principles and processes and things like that? Vishnu Priya, ma'am, do you cover zoology chapters also? Yes, dear, I do. I do, okay? So listen to me carefully. The answer for this question is artificial hybridization. Artificial hybridization. What exactly happens over here? Suppose I have a plant. Okay, I have a plant that is having really good features. Okay, I have a plant that is having really good features. I need to fuse with another plant which is also having some other good features. First of all, what I need to do is, suppose if at all this plant is bisexual, that means it has both male as well as female part. What I need to do is, I will be removing the male parts. Why? Because otherwise they will put down their pollen grain on the stigma. So the first thing that I am going to do is what? Emasculation, removal of the male part. Then after that, I am going to cover it with the help of a bag. So this is known as bagging. Why do you do that? We want the stigma to be receptive. We give time, space, everything out there. And later on, when we feel it is receptive, we are going to take the pollen grains from the parent that we wanted. And we are going to shut it, open it, put the pollen grains out there and then rebag it. And then rebag it. This is known as artificial hybridization, and this is very much useful for production of what? Hybrid plants. Yes, Prachi, you are absolutely correct. My lamp need to buy seeds every time. Exactly. You guys are super good. I'm so proud of you. Okay. Got it? So please write the answer like this. Please explain each and every stem selection, the parents, emasculation, bagging, everything. You'll get the PDF of this, so please do not worry. Okay. What is the importance of such experiment? No unwanted pollen comes. We can create our own different types of plants as well. Okay. Chalo. Next question. Picture related to pollination is given below. Answer the question that follows. So the picture is missing, so I'll make the picture by myself. So over here you have the picture. So my picture might not be that great, but still you will be able to understand. What is this picture of? This picture is of what? Ma'am, when is our next live class? Dear, I will not be coming live tomorrow since I thought that you need help today. Tomorrow you will be doing a self-study. So this is the live session that I will be having now. Okay, when is the next life class is a big question. Doesn't the stigma already identify which pollen should create tubes and all? It's not like that. So basically, if at all we are providing species specific pollen grains, once they land, that's when the interaction happens. If at all it's all good, the interaction is fine, then only they form the pollen tube. Okay, so obviously species specific plants are being taken up and being done. Okay, A flower, A, B. Okay, thank you. A flower and a bee. So over here, my dear children, insect pollination is happening. First question is, will this pollination confirm fertilization? Will this pollination confirm fertilization? Do you think, even if pollination has taken place, fertilization, my dear children, can we think it happens? No. Pollen tube can form, pollen tube may not form. If pollen tube is formed, some may go and get into the ovule, some may not. So, pollination does not confirm fertilization. First of all, write the answer. So pollination only brings the pollen grain to the stigma. It is just a transfer process. Fertilization occurs only if the pollen is compatible, germinates on the stigma, and also forms a pollen tube that reaches the ovule. Okay, if the pollen grain is incompatible, it will not germinate. Fertilization is not going to happen as well. Cool? Now, insect pollination. Thank you so much. Now, what are the floral rewards provided by the plants to the insects to revisit? What does the flower give to the insect so that it will revisit? First thing, nectar. Second thing, pollen grains to eat. So, some pollen grains it will eat, some pollen grains it will carry. And, my dear children, shelter. These are the three things that acts as floral rewards. Okay? Now, mention the characteristics of flower in this pollination other than the floral rewards. They are going to be big, they are going to be bright in color, large, colorful and fragrant flowers to attract the insects. Apart from that, sticky or spiny pollen grains so that it can stick on the insect's body. Okay, and also stigma is sticky to capture the pollen grains board. If at all, an organism is like a fly and a beetle which does not like good smell. So, obviously, the flower should be having a very bad smell. Okay. So yes, it is my lamp, Mansi, Jayashri, thank you, Krishna, thank you, and my lamp, thank you again. Okay, come on. Now the next question, this is from model question paper 1. This is an important question, try to solve this question. Okay, colorful and fragrant, yes. So my dear children, this important question we are going to solve together, okay. Over here you have the features, you have two things, male gametophyte, that is your pollen grain, and female gametophyte, that is your embryo sac, given on both the sides. You have to fill in the details. In the case of male gametophytes, how many number of cells are present? Now you can tell me the answer. You can tell me the answer. This is A, this is B. 1A and B. 1A and B. Number of cells present in the male gametophyte is how many? How many cells are there in the male gametophyte in the pollen grain? Come on, come on, come on. I'm waiting for you. Come on, come on. Two cells. Two cells. Pampapati, I'm not sure if I'm telling it correct, but ma'am, I have a doubt. Will these questions help in tomorrow's test? Tomorrow is Sunday. Monday's test, definitely it will help you. And yes, number of cells present in male gamete, you are saying 2. So those of you who are saying 2, Addi. Why Addi? My dear children, pollen grains, my dear children, it can have 3 cells or 2 cells. Vegetative, generative, then generative can also divide to produce 2 male gametes. So in that condition, either 2 or 3. Either 2 or 3, remember? Okay. And what about this? Number of cells present in the female gametophyte. It will be always 7 cell. 7 cell are there, but 8 nucleus is there. Okay. So those of you who said 2 to me, please make sure you are getting this correct. Okay. Next thing, number of gametes present in mature male gametes. Over here, what is the answer for this? Number of gametes present, there will be 2 gametes. 2 gametes are there. In the case of female, only 1 gamete will be there. Only 1 gamete is going to be there. Now, types of cell division involved in the development of male gamete. In the gametophyte formation, you have mitosis. Mitosis is the one that forms gametophyte. Earlier meiosis happened, you got the microspore. Later on, it is mitosis, right? So, answer is mitosis. What about this? How does female gametophyte form? I also told you 1 megaspore. Only 1 megaspore develops, and this megaspore undergoes what? Yes, 7-celled, 8-nucleated condition. You're awesome. Okay. So children, you keep on answering. I'm seeing it, okay? So over here, yes. So female gametophyte, 1 megaspores, nucleus divides how many times? 3. 3 on mitotic division will be there. 3 nuclear divisions. So over here, again it is mitosis. Now last one, ploidy of the cells of. What is a ploidy over here? Okay, ploidy of the cells. Ploidy of the cells up over here in the mature male gametes, they are all haploid in nature. They are all going to be haploid in nature. Vegetative, generative, all of them haploid. What about female gametophyte, my dear children? All of them are haploid except one. What is that except one? Ayo. That except one is this guy. Because here haploid, here haploid, 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 haploid. This is a central cell, and central cells and start 2 nuclei. Haploid, haploid becomes what? Diploid in nature. So just the central cell will be diploid, rest everything is haploid. Now if you see the answer, this is the answer. Please go through it once. Okay, please go through it once. So Vishnu is someone who told me something, right? Vishnu, yes. Ma'am, if possible, could you please consider short live session? So, you know what? I have already recorded a video. Okay, it's a revision video because, you know, live might go even longer. So, I've recorded it. It contains all the very important chapters. Okay, the information of all the very important chapters are being converted up there. That is all the questions. I've done a quick revision of them. Okay, Vishnu, yes. So all my dear children, you are saying haploid, haploid, diploid, diploid. Thank you so much for answering this question. I hope you have got it. Obviously, I was not able to understand which point you're solving, so which is why I went ahead. Now, there is one more part of this particular question, and that is give the advantage of seed. What are the advantages of seed? Protection and dormancy. Okay, seed coat is providing protection to the seed, embryo basically, right? So that is one advantage. Second one, food for the embryo. Seed has stored food for the embryo in the form of cotyledons or in the form of endosperm. So that is the next one. Dispersal. Seed is there better adapted for dispersal. Now if dispersal is happening, obviously the species will be going on. Okay, so that is also there. Also remember one very, very important thing, and that is there are 2 types of seeds which stayed for a very long time. You know, one was found in King Herod's time, and other was yeah. So basically, I'll tell you. One is Lupinus articus, and the other one is Phoenix dactylifera. Phoenix dactylifera. So there are 2 plants given in your NCERT, and it said that their seeds remain viable for a longer time. Can you tell me who's lived even more longer? Was it Lupinus articus or was it Phoenix dactylifera? Yes, Sushmita. See, dispersal to continue the date palm. Yes, date palm is basically Phoenix dactylifera. That is the date palm. And Lupinus articus is the other one. So my lamp is saying it is Lupinus articus that stayed for a longer time. Is it? Can you tell me the years? Can you tell me the years for which it stayed viable? This is also an important MCQ question. Phoenix dactylifera for 2000 years. That is date palm. Phoenix dactylifera is basically scientific name of date palm. Okay, and it remained viable for 2000 years. What about Lupinus articus? What about Lupinus articus? It was found in the Arctic region, my dear children, and it was viable for how long? Any idea? 10,000 years? 1,000 years? Mansi says 10,000. I'll lock 10,000. So for 10,000 years, Lupinus articus was there, and date palm which was there, it was viable for 2,000 years. Always remember that. 1 million years. Okay? Yes, you guys are correct. Nidhi, Vishnu, Philip, Sushmita, Mansi, my lamp, all of you. Okay, let's go ahead. Now, explain the different outbreeding devices developed by the flowering plants. Any guesses? Somi also got it correct, okay? Explain the different outbreeding devices. What do you mean by outbreeding devices? The ones that do not allow self-pollination to happen. It says go breed out, do not please have inbreeding things. because it leads to inbreeding depression. The same toxic character will get accumulated. So that's why we do not want inbreeding at all. Now, what are the features that we have? Always remember, this is a very important question. First is time difference. Okay, that is one way. That is pollen release and the stigma receptivity is not synchronized over here. I have written in the easiest format ever. Okay, the next one, yes, self-incompatibility, my lamp. And no, Baba, emasculation and bagging is coming under artificial hybridization. This is what? Outbreeding devices. Okay, now spatial separation. Anther and the stigma are positioned differently. Anther over here, stigma over here. Do you think pollination can happen smoothly? No. So that is also one thing that the plants have taken up. The third thing, self-incompatibility. That is one very important thing that you guys were saying. Okay, Pampapati. Yes, am I right? I took your name. Okay. The next one is self-incompatibility. That is a genetic mechanism to prevent the pollen from fertilizing the ovule. So that in the pollen-pistil interaction, there is some problem that is happening, which is why the pollen is not able to form the pollen tube itself. So that is self-incompatibility. Now is the production of unisexual flowers. If unisexual flowers are produced, do you think pollination can happen smoothly? No, Baba. And there are two types of plants. Okay, one is monoecious plants, other one is dioecious. This is important. This is where you might do mistakes also. Listen to me carefully. Dioecious plant is when? Okay, dioecious plant is when, suppose over here you have two plants. One is a female plant and one is a male plant. Female, male. Here, two plants are there, female plant and male plant. This is dioecious condition. So whatever you do, you need to have xenogamy to take place, because from here to here it cannot go. Both of them are female flowers, right? Gytnogamy, autogamy, nothing can happen. So always remember, in dioecious plant only xenogamy can happen. Only xenogamy can happen. Now on the other hand, what about the other plant, monoecious plants? Monoecious plants have two sexes. So one can be female flower, another can be male flower. If at all it is like this, then obviously autogamy is prevented, but gytnogamy can still happen. Even though unisexual flowers are produced, but gytnogamy can still happen over here. Example of dioecious plants are super important. It is papaya and watermelon. Papaya and watermelon. Okay. So please remember that the actual one is shown by dioecious plants. Branda boy, how many marks will come if I follow you? 100. If you follow me, you will understand all the concepts and it is definitely going to help you out in your exam. But you have to follow me 100%. Okay? Hello, dear. So, yes, my lamp, dioecious plants such as papaya. Different. Yes, all of you are... Okay. So, chalo. Now, I think this is assuming this is going to be the last question of this particular chapter. Yes. Let's deal with it then. Question number 13. You're helping us a lot. You're most welcome. So answer the following. Double fertilization is a unique event taking place in the flowering plants. Briefly explain. I told you already. Do we need to explain it again? Double fertilization is happening. One is syngamy. Okay, male and the female gamete fusing. That is forming zygote. It is syngamy. Other one is basically when male gamete fuses with the two polar nuclei. That is triple fusion. Okay, so over here, how do you write the answer? That is one male gamete fuses with the female gamete and this leads to formation of diploid zygote. This is known as syngamy. Another male gamete fuses with the two polar nuclei in the central cell to form what? The primary endosperm nucleus. This is known as triple fusion. Since two fusions are happening in the same embryo sac, you call this as double fertilization. And tada, we have got the answer. Now over here, you are asked to arrange the things in the sequence. In the sequence. Branda boy could have been something with chemistry, but my dear, I am teaching biology, teaching for a very long time, and I can guarantee you it is definitely going to help you out. Okay, that does not mean you do not study by yourself. Listen, whoever in this world says that, hey, you study this and only this, you cannot just rely on that. You need to do your own preparation, as in your own revision as well. If at all you are not doing that, then it is not going to help you out, right? So please do that. Okay. Hello Sheikh, hi. Okay, so arrange the following terms in the correct development sequence. Pollen grain, sporogenous tissue, microspore tetrad, pollen mother cell and the male gametes. What do you think is the correct one? First is sporogenous tissue. In the sporogenous tissue, you have the pollen mother cell or the microspore mother cell. The microspore mother cell undergoes meiosis to form what? Microspore tetrad. The microspore tetrad will now give what? The pollen grains. The pollen grains will be having the male gametes. This is how you need to fuse it. Yes. And brand boy, I called you Brinda boy. Oh my God, I'm so sorry. You are awesome. Do not just be worried. You just give your 101%. Okay? Done. So my dear children, so I have officially completed sexual reproduction in flowering plants. Okay, from my lamb. Thank you so much for typing up that. Now, you tell me, did you understand each and everything? If at all you did, can you just give me a thumbs up? Everyone who have understood sexual reproduction in flowering plants, I just need your thumbs up. Thank you, Sushmita. Thank you for giving me the answer. And Pampati, flabbergasted by your English. Oh, thank you so much. Yes. So, my dear children, can you give me a thumbs up if at all sexual reproduction in flowering plants is complete? Okay. Anything else that you need to study is, I'll just talk about it. Okay. Sheikh, thank you, Rishi. Okay. One very important thing that I want to tell you is, please know the examples of water pollination and wind pollination. For wind pollination, the features of the wind pollinated plants you should talk about, you should know about that. Just go through it. So if at all you have a notebook right now, so please write it. Wind pollination, I have to read it. Okay? Because everything else we have covered it, right? So wind pollination example is grasses and corn cob. But please know the features of the wind pollinated plants. Now there is also one more thing, that is water pollinated. Example is Vallisneria, and you have one more thing, that is Zostera. Zostera is a surface. Zostera is in the submerged condition pollination happens. In the case of Vallisneria, it happens on the surface level. Okay, it happens on the surface level. So please know that as well. Water lily, water lotus, everything that has water do not undergo water pollination. Please know about them also. Okay. As of now, what I am thinking is that I have covered everything, but please. Okay, now pollen viability is also one more thing. Let's discuss that also. Okay, if at all you have your NCERT, open the page of your NCERT for pollen viability. It's just behind the pollen grains. And you tell me, which one stays viable for months? Which are the plants that will be staying viable for months? Can you tell me? Alaina, ma'am, way too stressed, not understanding how to handle boards, practicals, NEET, KCET. Amorphophallus is the example of insect pollinated plant, no? Amorphophallus is the example of insect pollinated plant. Similarly, yucca and moth also comes under insect pollination. One of the largest flowers. Yes, that is there in the insect pollinated. So usually I don't see the questions coming up. Alina, one very important thing. As of now, you have your preparatory examination. Focus on that. Whatever examination you have in front of you, focus on that. I always tell everyone, as my parents also told me this one very important thing, that is, focus on the problem that you have right now. Do not think about future. When you think about future, it becomes all the more difficult. So make it right, whatever you have in your hand right now. And then later on, when this is right, you feel good. And later on, you will be able to work on yourself when you have time. Okay, so please have that. Vishnu says rice and wheat is something that will be staying viable for months. Is it? Can someone tell me Rosaceae, Leguminosae, Solanaceae? Yes, they are the ones that are going to what? Be viable for months. In your NCERT, is there something written as they will be viable only for these many months or things like that? Anything like that? Just give me a second, okay? So listen to me carefully, okay? Cereals such as rice and wheat, they are going to lose their viability within 30 minutes. Whereas Rosaceae, Leguminosae, Solanaceae, they are going to remain viable for months. So these are the things that you need to remember. Okay, and you can also store the seed. You can also store the seed in minus 190 degrees Celsius, 190 degrees Celsius in nitrogen, in liquid nitrogen. Know about that only. Okay, got it? Chalo. So basically, one very important thing, if at all you are someone who is preparing for KCET examination, the first thing that I want to tell you is, please go take up ABHYAS KCET test series. It is definitely going to help you out. Why? Because there are 35 KCET mock tests. There is chapter-wise KCET PYQs as well. And there are so many rankers that were there previous years, so you can trust us. The link will be there in the description section. Now, Rishi said, please ma'am, can we cover molecular basis of inheritance first? Which is why I'll directly go to that chapter first. Okay, and then I'll come back. So, molecular basis of inheritance is right over here. Okay, so I hope we are ready to go with the next chapter. And Vishnu, thank you, Mansi, Sheikh, Shalam, Prachi, Yasmin, Mailam, Philip, everyone, Isri, Pampapati, and Kunal, Jayashri, Sheikh, Musmita, everyone who gave me thumbs up, thank you so much. Okay, you're acknowledging, and that means the world to me. Starting away with molecular basis of inheritance, and I have the first question for you guys. This is from your model question paper 1. Another chemical name for the nitrogenous base thymine is, what is the other name for thymine? And everyone, have you clicked on the like button? Have you clicked on the like button? And have you shared it with your friends also? Have you shared it with your friends? Ma'am, give a break before you start another chapter. So, Lamp, what about taking a break after this chapter? 2 chapters at a time. What do you think? Is it okay? Okay. After this, we'll take break, okay? Because otherwise, it might not be completed. It might be too late. Okay. Option 1, Purnima says D, Brand Boys, and Mansi says D, Philip says D, Prachi, okay. Give me the answer. And Shalam also says D. Vishnu also 5-methyl-uracil. So yes, thymine is also known as 5-methyl-uracil. Remember, always in RNA, you have uracil. But in DNA, you have thymine. Which one is more stable? DNA. Why? Because it has thymine. Why? Because in the uracil, since it was less stable, you added methyl group at the 5th position. That is why you call it 5-methyl-uracil. So over here, your answer for this question is D. Okay? Next question. If an mRNA 3-AUG undergoes translation, how many types of amino acids are present? How many types of amino acids are present? Sheikh Aksa, my biology exam is from Monday and I'm not able to manage my preparation. I'm trying my best, but I feel very anxious. I just wanted to share this with you and request your guidance. I get your anxiousness. Listen, you feel that anxious because probably you're worried, as in there might be a lot in front of you to cover, or probably your exams might have not went that great. But let me tell you, you have this much time. You have tomorrow's time, you have today's time. If at all you still try, you are going to give your best. Okay, so do not lose your hope. Think that, no, I am going to give my best whatever happens. And that's all. That's all. Okay? So, okay. So 2 is 2 type. 2 type is, someone is saying, Jayashri is saying 2 type, Sri Sai Baba is saying 4. Okay, Soumya says 2, Prachi says C, Mansi says 2C, Shalam says 2C, Swastika also says 2C. Since you are saying C, I have to check it out. Is it C? So why C? So usually what children do is, if mRNA is written like this, they will be like, okay, 3 codon here, AUG is methionine, UUU is phenylalanine, UU again phenylalanine, UU again phenylalanine, UU again phenylalanine, and this is the stop codon. This is the stop codon. So basically in that case, this and this, anyway, all of this, they are coding for the same thing. So this and this, so which is why the answer for this question is 2 type. But the answer is not 2 type, because my dear children, translation happens in which direction? It happens from Y dash to 3 dash. It happens from 5 dash to 3 dash. And if it is happening from 5 dash to 3 dash, you have to read like this. So it will be AAU 1, UUU 2. UUU is the same thing. UUU again the same thing. GUA, this is the 3rd amino acid. So how many different types of amino acid do you get? You get 3 types of amino acid. So your answer for this question is B, not C. B, not C. Okay. Can you please send the solution PDF of all the biology model paper? So until 2 biology model papers, I have put it out there. The 3rd biology model paper, I have solved it here itself in this answer PDF. Okay, so if at all you've referred through this, you will get the model paper answers as well as the extra questions. Will that be okay? So your answer for this question is 3. So none of you got the answer as 3. So what do we understand from this? That we didn't read the question properly. I hope you got it now. Give me a thumbs up. Give me a thumbs up if at all you have got it now. Give me a thumbs up if at all you have got it now. And shall we go ahead with the next question? Question number 4. My lamb, oh my God, I would have lost 1 mark. No, you will not lose any marks. Okay, shall we go ahead? UAA does not code for amino acid. No, yeah, UR does not code. But Baba, you are reading from here to here, from 5' to 3'. So it is AAU, not UAA. You get it? It's AAU, so one type. Okay, Vishnu PR, ma'am, please do. I'm not doing live because I want to save your time. Rather, I will be giving you a recorded video. Do not worry. Okay, it is definitely going to help you. Cool. I am here. I am telling you it works. Chalo. So you read it like this and your answer is this. Now try this question. Match the following. Alec Jeffrey, F. Sanger, Jacob and Monod and Griffith. Alec Jeffrey, F. Sanger, Jacob and Monod and Griffith. What is your answer for this question? You have to do like this. Ma'am, I'm a CS student but attending bio. Can you guess why? No guesses. Okay. Alec Jeffrey was the person who gave DNA fingerprinting. How do I remember it? Basically, F is over here, fingerprinting is over here. This is the easiest way a person can remember things. Next thing, F. Sanger is there, S is there, annotated DNA sequencing also has an S there. My answer is right up there. Lac operon is there, where Jacob and Monod is there. Both of them have O, so operon is the answer for this particular thing. Griffith, my dear children, is responsible for the transforming principle, not transferring principle. It is transforming principle. So your answer for this question would be what? Your answer for this question is what? So C is what Sri Sai Baba says. Yashmin says, give me thumbs up. Thank you so much. And Sheikh give me B. Okay. Mansi says B, and Prachi also B. IM02 says B. Shalam also says B. B means A goes with 4. Okay. And B goes with 3 and C goes with 1. Okay. Let me see. Yes. This is your answer. Dharma, you are very beautiful, ma'am. Thank you, dear. You are also so pretty. Okay, Soumya also gave me the answer. It is B. Chalo, good. Next question. Try this question. Question number 4. GGU, GGC, GGA and GGG all code for glycine. Which salient feature of the genetic code does it prove? All of them are coding for glycine. Which is the feature that is going to be shown over here? What do you think is the answer for this question? Which salient features is there? B, degenerate codons is what? Arundhati. What? Arundhati is there. Hi, Arundhati. So you are saying degenerate codons? Yes. So when multiple codons are coding for the same amino acid, you are going to say it is what? Degenerate codon. So this is absolutely correct. My dear children, if at all you are saying that one codon is coding for only one amino acid and not multiple, that feature is known as unambiguous nature. Okay, that is not shown over here. If at all you are saying bacteria and human beings, all of them, in them GGU codes for the same amino acid, that shows universal feature. Initiator codon means the first codon that is going to initiate the process of translation. So over here, only answer that you have is B. Try this question. So guys, Jayashree gave me the correct answer and others gave me the wrong answer. Yasmin also gave me the correct answer. I hope it's clear. 4th question, is it clear? Arundhati, namaste dear. B, ma'am, am I right? Yeah, you are right. Brand Boy says, I will watch all your videos. Thank you so much. That means a lot. Yes, I hope that helps you as well. Okay, next question, try to solve this. Everyone, please give me your answers like this. Okay, 5A, 5B, 5C, 5D, like that. Whatever is your answer, give me like that. Put the number also, which is very, very important. So in a single stranded DNA, if the ratio of A and G is 60 and 40, then the ratio of T and C is what? If the ratio of A and G is 60-40, then the ratio of T and C is what? Okay. Sweta says C, Prachi says B. I really want to see the answer for this particular question. Read the question carefully. I'm telling you, there is something hidden here. Okay, there is something being hidden here in this particular question. A, Purima A, Shweta says C, Prachi says B. Okay. Mansi says A. Orange says B. Okay. Wonderful. Wonderful, wonderful. My dear children, you guys are all wrong. Why? Again, I'm saving you. Answer is none of the above. Here it is asked about single stranded DNA. In single stranded DNA, if at all A and G is 60 and 40, how can you expect how can you know the other ones? It can be T, it can be C, it can be G, anything out there, right? If at all you're talking about double stranded DNA, then okay, wherever A is there, there T will be there. You get it? Here it is single stranded DNA, so it is what? It would not be the answer. Got it? So here your answer would be, your answer is going to be this. Got it? Oh my God, yes. It happens, it happens. So over here, please remember, the answer is this. Do not be worried. Get things correct, that's all. Okay, done. Let's go ahead. Now, the components of deoxyguanin is, guanosine is, deoxyguanosine is what? The components of deoxyguanosine is what? Guanine plus ribose, guanine deoxyribose plus phosphate, guanine, ribose and phosphate. If it is a double-stranded DNA, then obviously you have to, okay, if it is a double-stranded, then how many number of thymine, adenine is there, that many number of thymine will be there. You get it? In that case, if at all you're talking about ratio. Okay. Solve this. The components of deoxyguanosine would be what? Six A, 6B, I am getting multiple answer. 6A, 6B, B. Well, your answer for this question would be this. Why? Listen, guanosine. Okay, guanosine is basically you're talking about nucleoside. Nucleoside is when you have the sugar and you have the nitrogen base attached to it. In this particular case, you call it guanosine, adenosine and things like that. Okay, over here, your sugar is deoxyribose. Here it is deoxyribose, which is why you have the term deoxy into it. You have the term deoxy into it. So deoxyribose, okay, and guanine is the nitrogenous base. So your answer for this question would be this. If at all it was deoxy guanosine monophosphate and things like that, it would have been B option, D option, C option, sorry. Okay, so here your answer is just B. Ma'am, can you discuss the marks allotted for each part as in? So as of now, I'm talking about the 1 marker session. Soon I'll be talking about the 2 and 3 marker. I'll tell you the marks, okay? Okay, next question. Now solve this question, everyone. A mature processed mRNA transcribed from a gene is 636 nucleotides wrong. Assuming if it codes continuously without UTR, untranslated regions, and without stop codons for a polypeptide, how many amino acid does it code? So it is saying that all of these nucleotides are going to code for amino acids. Can you tell me what is the, what do you call it? How many number of amino acids can you form from this? Any idea? How many number of amino acids can be formed from this? If at all there is no UTR, there is no stop codon. Seven A is this. Jayashree also says 211, okay. Shweta says 7C, 7A. I get maximum people saying A and B, A and C, sorry. Listen, 636 is there. 3 codons come, sorry, triplet nitrogenous bases comes together to form a codon, right? So 3 divided by this, it is equals to what? 212. Your answer for this question is plain, it will be 212. Because over here, they are mentioning there is no stop codon. If at all stop codon was there, you should have -1. -1 thing, that it will be 211. But since stop codon is not there, it will be just A. Okay? It will be just A. Yes, Zia Patil, yes, everyone, you guys are correct. Next question. Try this question, everyone. Question number 8. This is also from question bank. Until then, I'll read out the names of the people who gave me the correct answer. Sheikh Aksa gave me the correct answer, and Shweta, no. Okay, Shalam gave me the correct answer. Orange gave me the correct answer. Purnima gave me the correct answer. Zia gave me the correct answer. Prachi, Rishi, Mansi, all of you gave me the correct answer. For people who got their answers wrong, need not worry. You've got it correct now. You understood it, right? Now you are not going to repeat it. So you guys are awesome. Don't worry. Now solve this question. Please children, if at all you can, write the answer like this. 8A, B, C, D. Statement 1 says deoxyribonucleotide. It's not nucleoside now, nucleotide. Triphosphate. Deoxyribonucleotide triphosphate acts as substrate during DNA replication. So basically, suppose over here I have a DNA strand which is having A, then G, and then C. On the other strand, if I want to add what you call nucleotides, my dear children, I need to bring them up. How do they come? Do you know that? So they come in this particular format. So over here, A is there. So I would be having a nitrogenous base, that is T. And this T, my dear children, it is attached to the sugar. It is attached to the sugar, that is my deoxyribose sugar. Okay, deoxyribose sugar. And now this sugar is attached to 3 phosphates. It is attached to 3 phosphates. The exact image looks like this. So over here you have sugar, here you have nitrogenous base, and here you have 3 phosphates. What do you call this as? Deoxyribonucleotide triphosphate. Okay, it comes like this. Fine, agreed. But ma'am, you know what? Only 1 phosphate is there usually remaining. How come that? So basically this is exactly what happens. When these type of DNTPs comes together, my dear children, here also you will be having phosphate, right? The phosphate bond is gonna break. This bond is gonna break, my dear children, and over here lots of energy is going to produce. This energy is going to drive the whole process of replication. Got it? So deoxyribonucleotide triphosphate acts as substrate. Are they acting as substrate? Yes. Statement 2, deoxyribonucleotide triphosphate also acts as source of energy. Yes, this is also correct. So both statement 1 and 2 will be correct. Both statement 1 and 2 will be correct. Your answer is A. For this question, your answer is A. Okay. Your answer is not C. Did you understand it? Are you understanding? Or is it getting difficult? What's the scenario? Yes. Yes, Yograj. It is A. Everyone who had understood, give me a thumbs up. Can you give me a thumbs up? So whenever over here you have something like this, then there is next nucleotide, and this nucleotide is going to form what do you call bonds between them. But for that, this phosphate has to be free, and that's how you get the energy. Yes, Prachi. Now the answer is correct. Got it? Can you give me a thumbs up? If it's clear, everyone who understood this question is important. Okay, this question is important. Thank you so much. Thank you so much. Okay, next. In pea plants, each character is governed by a gene which is. In pea plant, each character is governed by a gene which is. Thank you, Purnima. Thank you, Rishi. Thank you, Yogi. Thank you, Sri, Prachi. Everyone. Okay, thank you. Try to solve this. In a pea plant, each character is governed by a gene which is. Pea plant is what? Eukaryote or prokaryote? It is a eukaryote, ma'am. And in eukaryotes like us, what is the case? We have one gene. This gene is going to code for only one protein. Agreed? One gene is there. This gene is going to code for one protein. You call this as monocystronic gene. Whereas in the case of prokaryotes, there is polycystronic gene, as in basically in one particular thing you have multiple genes. You might have seen in lac-operon there is Z, Y, A. There are multiple genes coding for multiple proteins within a cystron. So basically I'll call it polycystronic. So pea plant is eukaryotic. It will be having monocystron, polycystron, operon or intron. What will be your answer for this question? Pea plant is eukaryote. Then your answer would be what? Polycystron. Baba, polycystron means you are a prokaryote. Pea plant is what? A eukaryote. A gene, a protein. A gene, a protein. In this particular case, what will be your answer? This is monocystonic. And this happens in eukaryotes. What will be your answer? It will be monocystone. A is your answer. Yes, Shweta. Yes, it is A. Yes, Purnima. Now you are getting the thing. Prachi also. Good, good, good. Prajwal King DC. Awesome. Mansi, your answer for this question is A. Cool. Works. Shall we go ahead then? Let's go ahead. Question number 10. Try to solve this. Which of these are correct? So why I am making you solve MCQs? Because my dear children, these MCQs will also help you in understanding concepts that can show up somewhere in a 2-marker session, right? So that's why I am telling you. Which of these are correct? UTR, untranslated regions, are located on the DNA. Are they located on the DNA? A UTR is present both at the 5' before the start codon and also at the 3' after the stop codon. UTR provides stability to the mRNA. UTR is required for efficient translation. What will be your answer for the question? That is 10. 10 A, B, C, D. What is your answer? Always remember, UTR is there in an mRNA. Okay, so this is the 5' end and this is the 3' end. Here you have the start codon, here you have the stop codon. Before this, you have some untranslated regions that are not going to undergo translation. This is UTR. After this also you have UTR. These are also untranslated regions. Why do you have that? To make sure the translation happens smoothly, to make sure that the mRNA is stable. That is the use of UTR. But according to what I told you right now, can you tell me the answer for this question? It's there in the mRNA, it's there before the start codon, after the stop codon, to provide stability, to provide efficient translation. Now tell me the answer for this question. So I see okay. Shweta says the answer for 10 is C. Okay. Mansi also says the answer for 10 is C. Is it? 10D. 10D is what? 2, 3 and 4. Exactly, Purnima. Exactly, Prachi. The answer for this question would be 10D. Because over here, UTR are located on DNA is being mentioned. It's not on DNA, it's on mRNA. This is a wrong statement. This is a wrong statement. So in that particular case, your answer for this question becomes what? D. Got it? Done? Yes. Now, next question. In a DNA strand, if there are 100 nucleotides and 30 are adenine, the number of thymine, guanine, cytosine respectively are. Try to solve this question. Vishnu is saying, please do evolution chapter next. Okay, we'll think of it, okay? Evolution, actually, more than evolution, the chapter weightage is given to human reproduction, human health and diseases, and all of them. So, which is why I'm a bit confused, but let me see, okay? 2 marks question, Yavaga discuss Martira ma'am. I will discuss it after this, okay? I will discuss it. Come on, children, solve this question. If at all, there are 100 nucleotides. Okay, there are 100 nucleotides, and out of that, I'm telling you, adenine is how much? Adenine is 30. And they are now asking you, if adenine is 30, how much is the thymine? How much is guanosine? How much is cytosine? So how will you answer this question? How many number of adenine is there, that many number of thymine will also be there. So 30 goes with 30. Agreed? 11A. Okay, let me see. Everyone is now in full mood. 11A, let me see. So 30 here, 30 here. Out of 100, 30 plus 30, 60 is gone. Out of 100, 60 is already gone. What are we left with? 40. Now 40 if I split into 2, here if it's 20, here if it's 20, I have got my answer. So the number of thymine is 30, the number of guanine is 20, cytosine is also 20. You guys gave me the correct answer. Yes, Shweta, Prachi, Mansi, Purnima and Shalam, all of you gave me the correct answer. Awesome. Super awesome. Chalo, let's solve the next question then. Okay, now we are entering into the 2 and 5 marker session of molecular basis of inheritance. Are we ready? Zia Patil, 11A. So dear all of you, did you understand all the MCQ questions? Can we go ahead with the 2 marker and the 5 marker question? If yes, give me a thumbs up. If yes, give me a thumbs up, and after we are done with the 2 and a 5 marker, I will give you a short break. Okay? And please drink water. Water is so much important. If you're not drinking water, basically you will get dehydrated, and I do not want that. Hello, Khushi. Okay. Thank you so much. So I am going ahead. Okay. Name the methodologies used in human genome sequencing. This is MQP1, question number 24. So who was asking me? Vittal was asking me, no? So over here, Vittal, here we are with the 2-marker question. So what are the methodologies with which we can do human genome sequencing? There are 2 approaches. One is expressed sequence tag. My dear children, the DNA, when it is transcribed to RNA, in the mRNA, only exons are going to be there. DNA has 2 regions, exons and introns. Introns do not code for anything, exons code for proteins. Okay, only these exons are passed to the next DNA out there. Now these exons, my dear children, are going to be sequenced, out there. And this is known as expressed sequence tags. Only the genes expressed as RNA were identified and sequenced. This is one method. Second method is whole genome sequencing. That is known as sequence annotation. We sequence the complete DNA out there. Entire genome is being sequenced. That is another method. So there are 2 methodologies. Please follow them. Mahadevi, hi dear. Okay. Chalo. Next question that we have, draw a labeled diagram of nucleosome. How do we draw it? Nucleosome, my dear children. So first of all, listen to me carefully. The DNA is very long. How much long? We have 6.6 into 10 to the power 6.6 into 10 to the power 9 base pair long DNA out there in our body. And we need to fit it inside a very small nucleus. Very small nucleus. So for that, obviously we need to do something, right? We need to make them condensed. So what usually happens is that we have 8 histone proteins. How many? 8 of them. 1, 2, 3, 4, 5, 6, 7, 8. At the back also you have few of them. And now this histone proteins which is there, I will call them octamer. What octamer? Histone octamer. Why octamer? Because 8 of them are coming together to form this structure. Now, my dear children, there is one more histone protein which is right up over here. You will call this as H1 histone. What do you call this? H1 histone. That is a separate thing. Now, the DNA which is there, it comes around, wraps twice. When it wraps twice, you know how many number of nucleotides are being covered up? Almost 200 base pairs of 200 base pairs of the DNA, nitrogenous bases are going to be covered up over here. But why do you think the DNA is wrapping around the structure? Because, my dear children, always remember DNA is negatively charged. DNA is negatively charged. And what about your histone proteins? Your histone proteins are positively charged because you have 2 very important amino acids. They are positive amino acid. And what is it? It is going to be arginine and lysine. Please remember their name. Arginine and lysine. These 2 positive amino acids are there, which is why your negative DNA wraps around it. And this whole structure is known as a nucleosome. Now, suppose this nucleosomes are there in much amount. You will see a bead on a string-like structure, and that is what you call as chromatin. What do you call it? Chromatin. Monday bio preparation going bad. Mahadevi, come attend this live and it will be going jhakkas. Very good. Ma'am, all important questions. Yes. So all important chapters, important question I'll cover over here. Tomorrow you will be getting another video on the rest of the chapters. That will be a recorded video. Okay? Done? Vijit Kumar, yes, 200 base pairs. So this came in MQP2 and MQP3 also. Please make sure you go to the picture very nicely. The whole structure is known as what? A nucleosome. Going ahead, which sequences of bases transcribed from DNA are found both in hnRNA and mRNA? Try this question. Which sequences of bases transcribed from DNA are found both in hnRNA as well as mRNA? Now, ma'am, in exams, will they ask purines, pyrimidines type of diagram? They have not asked until now. Okay, they have until now not asked. Please go through the picture anyway, but they won't ask you is what I believe. Okay. Children, have you shared this live with your friends? I want everyone to be here who all are there studying biology. Everyone should be there, and who all are watching this video, do not forget to hit the like button and share it with your friends also. Okay? Okay. So, my dear children, you have transcription happening. First of all, you have DNA. The DNA is transcribed to form what? Your mRNA. But is it a direct process? No. First of all, you have hnRNA, heterogeneous nuclear RNA. Okay, this RNA will contain everything that the DNA was also containing. Exons, introns, everything. Now, basically, this particular RNA undergoes a process that is known as splicing, where you remove all the introns and you just have the exons, and that's how you get the processed form of RNA, which is known as mRNA. Here the question is, which basis of sequences are going to be common in hnRNA and mRNA? So if at all you see this particular diagram, this is your hnRNA, this is your mRNA. Over here, what are you removing? You are removing the introns. What is left behind? Only the exons, and that is why we say exons are something that is common. Exons are something that is going to be common. Heterogeneous nuclear RNA, that is a full form of hnRNA. Okay. Always remember there are three types of processing that is going to happen. One is splicing, other one is capping. Capping means you add 5-methylguanosine. Okay, you add 5-methylguanine over here at one particular end, at the 5' end. Over here at the other end, 3' end, you will be having so many adenine groups. That is known as tailing. So capping, tailing and splicing are the three processing that we do. Done? Shall we go to the next question? Okay. So this is what I said. When we start off the live, it is like full. Ma'am, this notes please. You will get this notes in the WhatsApp channel. So make sure you become a part of our channel. Okay. You will get the notes out there. So when the live starts, everyone is like, I'll score full on full in biology. And when the live is like halfway, everyone is like, oh my God, it's too difficult. I'll go sleep, get some sleep. Don't do that. Stay consistent, cover it up. Tomorrow you will have a full day to revise whatever you have studied. Yes, I am here standing for you for a very long duration. Why? So that you get helped with the things. Okay, so my dear children, be there, share it with your friends, and make sure everyone watches the video. Okay, now mention the levels where gene expression can be regulated in eukaryotes. Always remember, in eukaryotes you have nucleus, you have cytoplasm, everything. In the case of prokaryotes, you know what? Everything happens at the same location. Transcription, translation, everything in the same location. So you cannot regulate it. But in the case of eukaryotes, you can. Listen to me carefully. In the case of eukaryotes, within the nucleus, what is happening? Transcription is going to happen. So I can stop transcription from happening. This is the first level where I can regulate. So one is transcription level. Second one, after the transcription, you have got the hnRNA. This hnRNA have to convert to mRNA, right? This needs processing. I can stop the processing to happen. So processing level, at that particular level, it can be stopped. Next thing, the mRNA is being formed. This mRNA has to be moved outside. So transport of mRNA from the nucleus. Transport of mRNA from the nucleus. That is the next thing. So here, if I do not allow the transport of mRNA from the nucleus, then what will happen? Again, the whole process, gene expression will not be taking place. Now the last level where where the gene expression can be stopped is, suppose mRNA is out here and the tRNA is coming, but I'm not allowing translation to happen. So at that particular level also, I can stop or regulate the gene expression out there. So at 4 levels, I can regulate expression in the case of eukaryotes. Okay, in the case of eukaryotes. I hope you have got it. Done? Next question. Write a note. Okay, write a note on the role of sigma factor and the rho factor in transcription in the case of prokaryotes. So my dear children, in the case of prokaryotes, you know, RNA polymerase is going to go bind on the, what do you call this as? This is your DNA for undergoing transcription, right? In the DNA, you have a promoter region on which this RNA polymerase has to come and bind. But it is very shy. Since it is shy, it meets sigma factor. Shy starts with H, sigma also starts with H. So you can remember like that. So since it is shy, it comes with sigma factor to come bind over here. Then once it is bounded, sigma factor goes away and the whole process is going to start. So to initiate the process of transcription, you need sigma factor. Now you know what happens? Later on, when the RNA polymerase moves, it is feeling very lonely out there because it is forming RNA, but no one else is there with it. Everything else is DNA. So it is crying. It wants to terminate it. So during termination, it needs rho factor. Rho as in crying. You remember like that. So rho factor comes and binds, and that's when it will be terminating the whole process. I hope you have got it. So basically, sigma factor binds to RNA polymerase, guiding it into the promoter region for transcription initiation. It attaches after the process of initiation. Okay, now rho factor facilitates termination by binding to RNA and unwinding the RNA-DNA hybrid in rho-dependent termination. That means it will be helping in terminating the whole process. That's all. Cool? Ma'am, where is the WhatsApp group link? It will be there in the description box. Okay, it will be there in the description box. You can check it out. Okay, now next thing. Write the difference between euchromatin and heterochromatin. My dear children, I told you nucleosomes, they come together. Over here, suppose the nucleosomes are far away from each other. You see a lot of space. In this condition, where it is very loose, you call this as euchromatin. If at all it is very much condensed, you will call this as heterochromatin. Listen, euchromatin has spaces of DNA. Now, this DNA can undergo transcription. This DNA can undergo transcription like this, which is why we say that it is transcriptionally active. This is what euchromatin is transcriptionally active. Over here, do you think RNA polymerase can come and bind anywhere? No. So this is what? Transcriptionally inactive. So you have transcriptionally active DNA, transcriptionally inactive DNA. Now since it is loose, and if you put any stain out there, it will lightly stain. But here, since there is no space, if you put some stain, it will stain darkly. So these are the differences between, what do you say, heterochromatin as well as euchromatin. Okay? Ravi Chandra is asking, ma'am, can you give most important fixed question, the 2 marker, 3 marker and 5 marker? So dear, I have made a list of fixed questions video. Check it out. It was released at 1 p.m. today, so you can go check it out. Okay? Okay, and I'm explaining those questions out here, so please refer to these things as well. Okay? Now, name the virus used in Hershey and Chase experiment. What is the name of the virus? Anyone? Any guesses? What was the name of virus which was used in Hershey and Chase experiment? Hershey and Chase experiment was the last experiment to basically prove that DNA is the genetic material. Is the genetic material. So what was the name of that particular virus? Any idea? It was known as bacteriophage. It was known as bacteriophage. Got it? Clear? Clear, clear, clear? Going ahead, list the goals of Human Genome Project. What? Yeah, bacteriophage, exactly, Mansi. By seeing that section, how many marks will come? Only by seeing that section, you will not get any marks. You will have to practice each and every question. If you do, you will get 60 plus marks for sure. Okay? So I have tried to put all the repeated concepts and questions out there. So you can definitely try it out. Hi dear Kavita. Okay, now list the goals. How do you list the goals? List the goals was to identify all the genes. They assumed that there were almost 20,000 to 25,000 genes out there. So that is one thing. To determine the complete sequence of DNA. Apart from identifying the genes, they also wanted to determine the whole sequence of the DNA. Then they wanted to store this information in what? Database. After storing, they wanted to have the tools, improved tools for analysis of this data as well. Then they wanted to transfer this information to other sectors and industries as well, and also address the ethical, legal and the social issues. So basically these are the goals of Human Genome Project. Okay, Human Genome Project. Got it? Yes. Now next, question number 8. DNA is genetic material in both prokaryotes and eukaryotes. Yet the packaging process in their cells is not the same. Explain. This came in this year's model question paper. DNA is a genetic material in what? Both prokaryotes and eukaryotes. Yes, agreed? Yes, ma'am. Yet the genetic material or the genetic packaging is not the same. Why? Because over here in the case of prokaryotes, everything is here itself. The DNA is just roaming around here because they do not have any nucleus and anything. So genomic size is less. The amount of DNA is less. And also they can be anywhere. So they do not need a proper packaging. But in our case, the DNA has to be within the nucleus, which is why it has to be nicely packed. So you need to understand that. Ma'am, really WhatsApp link is not there in the description? Is it? Okay, it's there. It's written to get lecture notes, PDF of this lecture, join Deeksha KCET WhatsApp channel. It's there. Can you check it out? If you want the link, I can, but I do not know if I be able to copy or not. Anyways, I'll give you. So once this live ends, yeah, you have got. Cool, cool, cool. Okay, come. So where were we? So we have to fit in everything inside, right? So what do we need to do? We need to do packaging. So DNA is the genetic material in both prokaryotes and eukaryotes, but its packaging differs due to differences in genome size and also cell organization. Okay, now in prokaryote, DNA is naked, circular and present in the nucleoid region, as in a region where the genetic material is present. Without histones, they do not have histones. Always remember, prokaryotes, no histones, because of the small genome and the absence of nucleus. Whereas in eukaryotes, DNA is associated with histones, and everything that I discussed about nucleotides should be mentioned up over here. Okay, nucleosomes, everything that I spoke about nucleosomes should be mentioned up over here. Done? Can we go ahead with the next question? Explain Griffith's transforming principle experiment. Now this is my favorite experiment. Explain Griffith's transforming principle experiment. Come on, my dear children. This is a 5-marker question. We have to be so attentive. This came in model question paper. We can expect similar type of things coming up anywhere in this year, this time. So first of all, Griffith want to find out what the genetic material out there. So for that, he chose a bacteria. Its name was Streptococcus pneumonia. It was causing pneumonia. Okay, and there are 2 strains of this bacteria. One is the R strain. And the second thing that we had is the S strain. You know, S strain was known as S strain because it was smooth. Why was it smooth? Because it has an extra polysaccharide covering, an extra membrane out there. And this polysaccharide covering was making it virulent. Virulent means if at all an organism is infected with this S strain, it is going to die. But the R strain, it was rough strain, but my dear children, rough strain did not have the extra coating, and that exactly why it was non-virulent. It was not virulent. Now we'll go ahead with the story. A little mouse was there. Its name is Puchu. Okay, so this Puchu mouse was given what? Live R strain. When live R strain was put inside the mouse, the mice lived. Why? Because it is what? Non-virulent, not causing any trouble. Live S strain was put inside Puchu. Puchu died. Why, my dear children? Because S strain was virulent form. Then he was like, let me do some experiment. Let me treat it with heat. Live S strain was treated with heat. And when it was treated with heat, now we will call this as heat-killed S strain. And then when it was put, you know, Puchu was living. Out of sudden, we saw that Puchu was living. How is that possible? Maybe because of heat, some problem is occurring up over here. Okay, so now he got to understand live R strain and heat-killed R strain are not causing any trouble to Puchus out there. So he thought, let's mix it up. So he mixed it up and put it inside Puchu, and Puchu again died. How, my dear children? How? We do not know. But one very important thing he could find. Inside Puchu, he found live S strain. He never put live S strain. But where did this guy come from? Probably the heat-killed S strain was transferring something from here to the live R strain, which is why the live R strain is getting converted to or getting transformed to what? Live S strain. And that's where he was like, oh my God, something is happening. I need to find the transforming principle. So the thing that was getting transferred is known as transforming principle, and he was confused whether it was DNA, whether it was RNA, or it was a protein. And who gave the solution for that? Do you have an idea who gave the solution for that? Who told that probably it could be DNA that is the genetic material or the transforming principle? Any idea? Well, you need to write your answer like this properly, and then you will be getting your marks. At the end, please write, Griffith concluded that some heritable material, that is transforming principle, was transferred from S strain to the R strain. Now you have to tell me who gave the correct explanation, as in what was the genetic material. Who gave the correct explanation, as in what was the genetic material? Any idea? No idea? Yes, idea. Puchu was epic. Thank you. Any idea? Any idea? Any idea? Come on, guys. So Griffith experiment was done. After that, few people came in, and that was Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy. They were the people who said that DNA could be the genetic material. How did they know that? So basically, over here, when the live S strain from the group 4 was found, they basically made sure of one thing. The heat-killed S strain which was there. The heat-killed S strain which was there, they treated it first with RNAs. They treated it with RNAs so that all the RNA molecules could be digested. Now once the RNA molecules are digested, now if we mix it with our strain and we put inside Puchu, let's see if at all Puchu is dying. It was seen that Puchu was not dying. It was seen that this particular mouse was not dying. That means the thing was still there. RNA is not that thing which is going to get transferred. Now they treated it with what? Protease. Now they treated it with protease so that any protein which is out there, they get digested. When protease was being put, still the transformation was taking place. So that means nothing was changing up over here that could cause any changes up over here as well. So protein also was not the thing. But later on, when it was treated with DNase, the DNA molecule was getting hampered. So in that particular condition, it was found that Puchu was living. Puchu was living. And that's when they got to know that DNA could be probably the one which is getting transformed. But people still then believe Avery, MacLeod, McCarthy, and that's when who came into the picture? Hershey and Chase came into the picture. Hershey and Chase went ahead and did their experiment with bacteriophage and E. coli. Okay, they did their experiment with bacteriophage and E. coli. They took E. coli and basically they took 2 bacteriophage. Okay, they took 2 bacteriophage, which is basically nothing but a virus. One bacteriophage was tagged with S35 and one was tagged with P32. So phosphate is a member of our DNA, so it obviously mixes with the genetic material, whereas sulfur is a part of the protein, so it goes and fuses with the capsid, the outer layer. Now both of them were allowed to infect the E. coli. Okay, so obviously the genetic material was being passed. Over here also genetic material was being passed, but this was not radioactive. The capsid was radioactive. Okay, the genetic material was not radioactive. So I'll show some colors over here. I'll show some colors up over here. Now both of them, when they were centrifuged and they were separated, it was found that basically the pellet which was there, okay, the pellet which had the cell, that did not show any color. That did not show any color. I colored what? The capsid. That is the outer thing, and that was found in the supernatant, that is in the extra fluid out there. The colored thing was found in this particular thing where I was treated with P32. So it is DNA that was getting transferred from here to here, because only cells that have got the DNA will be showing the colors, and over here that was happening. Okay, that was your Hershey and Chase experiment. Please go through it. It is super important. So Griffith experiment is being asked in MQP2. You can expect these type of things also coming up there. Now, state the arrangement of different genes of lactose operon. Lactose operon in the E. coli, this is 3 marker, and describe the role of lactose in regulation of lac operon. That is another question. Now, this question is also super important, very easy to solve. Make sure you listen to me carefully. Okay, this is MQP3 question as well. So, lac operon goes like this. Okay, lac operon has a promoter region. This is an inhibitory gene. This is what? An inhibitory gene. Again, a promoter region and an operator region, and you have 3 genes, Z, Y, A. What is this? This is polycistronic. Multiple genes are being there in one particular cistron. Multiple genes are there coding for multiple proteins out here. So listen to me carefully. This I gene which is there, it codes for a repressor protein. Repressor protein. This repressor protein, you know what? It is very jealous of these guys, because Z, if it undergoes transcription, it is going to code for beta galactosidase. Z codes for that, Y codes for permease, and A codes for trans-acetylase. Trans-acetylase. So this is the story up there. Okay, now listen to me. Repressor protein is very jealous of this, which is why it does not want transcription of all this thing to happen. It goes and binds on the operator region such that if any RNA polymerase comes and try to transcribe, it is not able to do the transcription. You get it? So repressor protein is right up there. Now suppose, okay, and what is this beta galactosidase going to do? Whenever lactose is there, whenever lactose is there, it is going to break the lactose into glucose as well as galactose. That is the role of beta-galactosidase. What is the role of permease? It increases the permeability of what? Of the cell membrane of the E. coli. Transacetylase deals with every toxic substances which are being produced throughout the process. That is the usual role. Now, whenever lactose is not there, do you need beta-galactosidase to produce? No, Baba. That is why the repressor protein binds up there, because it does not want transcription to happen. If lactose is not there, why do you think there is a need for these particular enzymes? So which is why transcription is being stopped there. This condition is known as switched off condition. Switched off condition. Now suppose I am growing the E. coli inside a medium that has lactose. Inside a medium that has lactose. Lactose will slowly sneak inside. When lactose comes in, you will know what happens. Lactose comes in. When lactose comes in, lactose become best friends with the repressor protein. Now the repressor protein is not free to go and bind on the operator region. They are over here. Now if they are over here, at this particular point of time, the RNA polymerase will come and do the transcription, and here you have the enzymes. Now the lactose which is there, it is acting like what? It is acting like inducer. It induces what? Lac operon. It switches it on. It is switched on condition, and after this the lactose which have been taken up, my dear children, will be broken by the beta-galactosidase, because lactose is going to provide energy up there. Permease which was there, it is going to increase the permeability such that more lactose can get in. Now there could be a question, ma'am, how did the first lactose get in then, if at all the transcription was not happening? Listen, transcription happens, but the level of it is very less. So yes, there are some permeability, which is why you have the lactose being inside. Also remember, lactose or allolactose, both of them are going to act like inducers. Both of them are going to act like inducers. They are going to induce the thing. So how do you write the answer? Write that click. Promoter, I gene, promoter, operator, structural genes, this. What do they do? Everything. You have written, you made the diagram also. And then, my dear children, you write the role of lactose. The repressor protein binds to the operator. This blocks RNA polymerase, so structural genes are not transcribed. In the presence of lactose, what is going to happen? Mention that and you have got the answer. Okay? Going ahead with the next question. This is the question. Given below a sequence of nucleotides in a particular mRNA and amino acids coded by it. Okay. Write the properties of genetic code that can be correlated from the above given data. So what all features do you know from this particular thing? Anyone out there? Come on. Now you need to solve this. You need to solve this question for me. So over here you have all the mRNA codons out there. Here you have the amino acids. You have to tell the properties of the genetic code with the help of this information. Ma'am, can you say human health and disease specific part will come? Yes, I'll talk about human health and disease as well. Ravi. Okay? Come on. What does it show? Over here, AUG is there. AUG is coding for what? Methionine. It is coding for methionine, and it is also a initiator codon. It is also a initiator codon. It initiates the process of transcription. So I get one feature, that is what? Okay, that is what? Methionine which is there, AUG which is there, it is a codon with dual function or dual nature. This is one very important thing. Next one, UUU codes for phenylalanine. Again, UUU is coding for phenylalanine. UUC is coding for something. UUC is also coding for phenylalanine. GGA is coding for glutamine. UUA is coding for leucine. GUG is coding for valine. What do you understand from that? Each codon which is there is coding for a single amino acid. What feature is this? When one codon codes for a single amino acid, this shows unambiguous nature. Unambiguous nature of the codon. And now there is one more feature. UUU, UUC, both of them are coding for phenylalanine. That means what? Multiple codons which are there. Okay, multiple codons are coding for the same amino acid. This shows what? Degenerate features. If multiple codons are coding by for the same amino acids, you will be saying that what? It is degenerate feature. Applicable question. Shalom, AUG is acting as an initiator. Exactly. Vijit, stop codon. Yes. Okay. Shivanand, hi dear. Cool. And also, my dear children, if at all you see, all the codons are in triplet. Okay, so the codon is triplet. 61 codons code for amino acid, whereas 3 codons are not going to code for any amino acids. They are going to be stop codons. What are the names of these stop codons? If at all you are asked, it is UAA, UAG, and UGA. UAA, UGA, and UA. Yeah, UAG. So UA, UGA, and UAG. And all the codons, my dear children, they are read continuously. There is no full stop comma. So that is also one very important thing. So these are the features that you can find. Also remember, the codons are universal. This is the extra thing that I have told. Also remember that. Okay, the codons are universal. In bacteria, in us, the same codon codes for the same amino acid. Exception is there. Exception is in the mitochondrial codon. Mitochondria also has codons. And also in some protozoans also, these exceptions are there. Please remember this thing. Session PDF, my dear children, you will get it after the session is being done. You will get this PDF via the link given in the description box. Join the WhatsApp channel. Okay? Yes, Prachi, triplet codon, degenerate initiator codon, terminator codon, polarity from 5 to 3, universal codon, unambiguity. Awesome. Awesome. You guys are super good. Super good. Prachi, awesome. And everyone who gave me the answers, super good. My dear KBN, please join the WhatsApp channel. You will get the session PDF. Do not spam, dear. Okay, we're soon gonna end the chapter molecular basis of inheritance. Before that, we have, I think, how many questions? Few more questions, that's all. I think 3 of them are still going to be left. 3 or 4 are going to be left. So, yes. Describe the steps involved in DNA fingerprinting technique. What is DNA fingerprinting technique, my dear children? Suppose I did a crime. Okay, suppose I did a crime and you and me are there. I'll take someone's name. Suppose KBN and me are there. Okay, KBN is there and I am there. I did the crime, but I told KBN did the crime. Anyways, from the crime site, you got some blood sample. You got the blood sample of the person who did the crime. Okay, and now what did you do? What did you do? So basically, from the site, the DNA that I have got, I sequenced it. The first thing that I did is, I sequenced it and then I made a complementary pair for it. I made a complementary strand for it, and this is a very small strand that I made, and basically I attached it with some radioactive probe. Fine. So this is the probe that I already have in my hand. And now what do? I have a doubt. The police has a doubt whether KBN did the crime or I did the crime. So both of our samples were taken. And our DNA was being cut using restriction enzyme. Okay, both of our samples were taken, DNA were cut using restriction enzymes, and now, my dear children, our DNA were put in electrophoresis chamber. So our DNA got to separate. Our DNA, my dear children, it separated. Fine. And now you cannot see the DNA which is there in the electrophoresis chamber. You need to blot it on a nitrocellulose sheet. So whatever was there here, I blotted it on a nitrocellulose sheet. And now I have the probe. The probe, I put it there. The probe will go and bind. The probe will be going and binding to the complementary part. Okay, it binded to the complementary part. And now it is visible to me. Now what I am going to do, the DNA that I got from here, I am going to cut the DNA with the same restriction enzyme. I am going to make it through the same electrophoresis chamber, and then I will be having the same pattern. But how will I get to understand the pattern? Because the probe, my dear children, over here and here, it will be attaching at the same area. So I get to understand that basically the police get to understand that I did the crime. This is the use of DNA fingerprinting. Okay? Mr. Paradox. So dear, over here, I will be completing all the 2, 3, 5 marker, every questions out there of all the important chapters in this particular live. So this live is super important for you guys. That is why I came to take the live, so that if at all you have doubts, I could take that up. Now there are few more chapters, the medium weightage chapters, starting from biotechnology, principles and processes to biodiversity and conservation. I have made a separate recorded video for that, where I'll be discussing the 1 marker, 2 marker, 3 marker and 5 marker. That will be released tomorrow. So this video is important for you to watch. Why? Because you will have time to revise these chapters tomorrow then. Okay? And Shivanand, thank you so much, dear. Where we get the WhatsApp channel, dear, it is being pinned up over here. Please join the WhatsApp channel there. Okay. So isolation of DNA is the first step. DNA is extracted from the sample. It can be blood, it can be hair, it can be saliva. It can be blood, hair and saliva. Always remember, in the blood there can be RBC, WBC, everything. You cannot take all the cells. RBC does not have nucleus, so sorry, if nucleus is not there, DNA is also not there. So with the help of RBC we cannot do anything. Okay, so always remember blood, but blood have multiple cells, other cells also, right? So blood, hair, saliva is there, DNA is extracted from them. Then digestion by restriction enzyme. You cut by restriction enzyme. Then you separate by gel electrophoresis. Once you do that, you transfer everything to a membrane. And then you hybridize with DNA props. You hybridize with what? DNA props. And then you visualize it what? Visualize this under the X-ray film, and you get to understand the DNA pattern. You get to understand the relationship. If at all the pattern looks the same, it means the same person has did the crime. Okay? Fine. Now, ma'am got tired. Now, question number 13 of this chapter, the long questions. Answer the following. This is an important question, okay? Construct a complete transcription unit with a promoter and a terminator on the basis of the hypothetical template strand given below. So this is a hypothetical template strand, and you have to make a transcription unit with the help of this. How can you make that? Let's go, my dear children. Always remember this strand which is there, it's in this direction. 3 dash here and 5 dash. The arrow moves this side, no? So this is 3 dash and this will be 5 dash. So let me make it properly. If at all you do not mind, let me make it properly. This is 3 dash. 5 dash. Cool. Over here, what do I have? I have A, then T, then G, then C, then A, then T, G, C, A, T, A, C. I'm just making like that. Okay, got it? And now what do I have to do? Wait. Okay. Make another strand right over here. This will be from 5 dash to 3 dash direction. Now to make a transcription unit, my dear children, always remember what you have to do is, you have to make a promoter region right up over here. Okay, and over here you have to make a terminator region. So what is this? This is your promoter region and this will be your terminator region. What is this strand? This is your template strand and this is your coding strand. Which of this undergoes transcription? Do you know that? This is the one that undergoes transcription. The one in 3 dash to 5 dash direction undergoes transcription, always remember. Because transcription, RNA polymerase makes a new strand in the opposite direction, that is 5 dash to 3 dash. Now over here we have to find the opposite of this. A base pairs with T, T base pairs with A, G base pairs with C, C with G, A with T, T with A, G with C, C with G, A with T and C with G. Right? This is your coding strand. So this whole thing is what? Now your transcription unit. Now you have one more task to do. Write the RNA strand transcribed from the above transcription unit along with the polarity. So the RNA strand that you're getting from this would be what? This is a DNA. You have to make an RNA strand. I told you this strand undergoes transcription. Now when you make the RNA, you're going to get the complementary strand only. So the only thing that you need to do is, do not put multiple efforts. Replace the T by U. Wherever T is there, replace it by U, because you get the same thing only, right? So 5 dash U A C G U A C G U G, and then you have the 3 dash. This is going to be the new RNA strand that you're gonna get. Got it? Shivanand, don't rush, do calm, ma'am. Okay. Okay, sure. Is it too fast? Is it difficult for you to understand? Did you understand this question? This is question number 13. Super important. If at all you are asked to make the transcription unit, you have to make like that. And always remember, we say that the promoter sequence is towards the 5 dash end of the coding strand. Since every work is given to the template strand, no, we need to give some reference to the coding strand. So we say the promoter is towards the 5 dash end of the coding strand. The terminator is towards the 3 dash end. Okay? Remember that. Got it? Got it? Can we go ahead? Yes, ma'am. Okay. Now, list the salient features of human genome. Now, this is a question where we all do rectification, right? Rectification means basically we just mug it up. But obviously, mugging up things in biology can lead to trouble. But yes, with numbers, we have no other option, right? So, human genome contains 3164.7 million base pairs. Remember, in this page, everything is in million, nothing is in billions. Okay, so 3164.7 million base pairs are there in our human genome. First of all, that. Second, the average gene size. The average gene has 3000 bases. But you know what? We have one very big thing. Okay? One very big thing. That is what? Dystrophin. Dystrophin is a protein where you have what? 2.4 million bases. 2.4 million bases. Usually we have 3000, but over here we have 2.4 billion bases. Vaishnavi, today I'll do math, tomorrow bio, ma'am. Bio is easiest. Okay, if it is easiest for you, then yes, definitely you can follow that. Shivanand, tomorrow at what time will you be releasing the recorded class? Tomorrow almost by 10 or 9 I'll be releasing it. Okay. Sri Hari, model question paper is being covered over here itself. All the model question papers questions are being covered over here for these chapters. The rest of the chapters I have recorded already, so you can follow that. Okay, none of the model question paper questions are left. Okay, next. Now the total number of genes is estimated at what? At 30,000, much lower than the previous estimates of 80,000. So total number of genes are what? 30,000 out there. Human genome actually contains what? 3164 million base pairs. Average gene size is what? 3000 bases. But dystrophin has 2.4 million bases. Now the functions are unknown for about 50% of the discovered genes. We do not know any functions of the 50% of genes out there. Less than 2% of the genome actually codes for proteins. Less than 2% actually codes for proteins as well. There are so many repetitive sequences that makes up the DNA. And my dear children, what is important is chromosome number 1 has the most number of genes. It is 2968. This is easy to remember number. Why? There is no 7. Rest everything is there. 2968. So 2968. Chromosome number 1 has the highest number of genes, and chromosome number Y has the fewest number of genes, that is 231. Chromosome number Y, which is found in males, my dear children, only few number of genes are there, and that is 231. Okay, now scientists have identified about 1.4 million locations where single base DNA differences occurs. You call it as single nucleotide polymorphism. What do you call it? Single nucleotide polymorphism. So please remember these numbers. This. These are very, very important numbers out there. Please go through them. Okay, done. Now explain the process of translation in protein synthesis. How does translation occur over here? Any idea? So first of all, you need to know there is a tRNA. tRNA, it looks like this, okay? There's a 5' end, there's a 3' end. At the 3' end, you have the amino acids being joined. At the 3' end of the tRNA, you have the amino acids being joined, and this process is known as what? Aminoacylation. Aminoacylation. Okay, now over here always understand you need a lot of ATP in this particular process. You need a lot of ATP in this particular process. That is also important. And over here you have the anticodon loop. Anticodon loop. So aminoacylation happens in the first phase. That is done. Sri Hari, ma'am, explain the post-transcription modification. Yeah, I will explain. I had explained it before, but yes, I can explain it once more. Okay, but listen to me now, over here. So this is aminoacylation that happens already in translation, okay, in the first phase. Now we need to actually do the translation. How does that happen? So first we have the mRNA. The mRNA is in 5' to 3' direction. Over here, the first thing that comes and attaches is the small subunit of RNA, so of ribosome. Ribosome has two subunits, one small and one big. So small subunit comes and attaches over here, okay, and then my dear children, your first tRNA will be coming and attaching on the initiator codon. This will come and attach over here. Then the large subunit will also come and form the complex. And now everything is super ready. Everything is super ready. Slowly what will happen? The ribosome is going to move throughout the mRNA. And while it moves, many tRNA will come and join. When tRNA comes and joins, they bring the amino acid. So the one that goes will pass the amino acid to the other one. So that is why you get a chain of amino acids like this. So while this chain of amino acids is being formed, you need to understand the amino acids between each of them, they have peptide bonds being formed over here because of the enzymes which are present in the ribosomes. The enzymes which are present in the ribosomes will be helping in the peptide bond formation between the amino acids. It will go on until the stop codon is reached. Once the stop codon is reached, my dear children, no tRNA will come and bind on the stop codon. Stop codon will be having a release factor being bounded on it. When a release factor is being bounded on it, the whole sequence goes off, and that's how you get the thing. And how do you frame this answer by yourself? First, you explain what is translation. It is a process of synthesizing a polypeptide or a protein from the mRNA template. Now, process of translation takes place in the cytoplasm of a cell where ribosomes are there. The protein factories are there. Now listen to me. First is activation or charging of amino acid. That is also known as amino acylation of tRNA. It is the process of attaching of amino acid to the corresponding tRNA. This is also known as charging of tRNA. This has taken place. Now we have what? Initiation. mRNA binds to the small ribosomal subunit. The start codon is recognized by the initiator tRNA, and then the largest subunit assembles, and now you have a big initiation complex. Okay? Going ahead, you have elongation. tRNA brings amino acid to the ribosomes, matching codons on the mRNA through its anticodon. It goes on. Peptide bonds form between the adjacent amino acid catalyzed by peptidyl transferase. This is the name of the enzyme. What is it? Peptidyl transferase. It makes the peptide bond. So peptidyl transferase. mDNA. What is mDNA? Vijay Lakshmi, why not from mDNA? And I'm not getting it. So now, the ribosome also act like a catalyst. You should also know one very important thing. Apart from peptidyl transferase, there is ribosome. Inside the ribosome also, there are some catalysts. Because, my dear children, what do you call this catalyst as? Suppose you have an example. In the case of bacteria, there is 23S rRNA. rRNA is something. Ribosomal RNA is something that forms up the ribosome along with some proteins. Over here, the rRNA also act like catalyst. Catalyst as an enzyme, someone that increases the pace. Okay, so 23S rRNA in bacteria is the enzyme, that is ribozyme, which is helping in the formation of peptide bond in the case of bacteria. Remember it, 23S rRNA. 23S rRNA. This is acting like a ribozyme. Fine, ribozyme moves from the direction of 5' to 3' end. Remember that also. Termination. When a stop codon UAA, UAG, UGA is reached, translation stop as release factors are going to bind. And now one more very important thing, since we're talking about translation, you should know in the mRNA, I already explained you before the 5' end and after the 3' end, you have some untranslated regions, you call them as UTR. They are very, very important for process of translation to happen smoothly. They are very, very important for what? Process of translation to happen smoothly. Apart from that, very, very important for stability of this thing, for process stability of mRNA and for initiating the process of translation also. Super important. These guys are there only in the mRNA. Got it? Shivanand, so cute, ma'am. I am from Bangalore district. Thank you so much for calling me cute. And hi, we are neighbors. I am from Bangalore as of now. So little louder, ma'am. Sorry to say. Okay. Okay, little louder. Okay, I'll try to bring up the energy. Seeing you energetic, I might get energetic. Okay, so you guys have to be super, super energetic. I will also be energetic. Now, this is the next question that we have. And this is going to be the last question from this chapter. And that brings a lot of energy, right? Ma'am, I mean, what is the role of DNA in this particular process? Arey Baba, what is the role of DNA in this particular process? Listen, who gives the information to form the mRNA? mRNA is going to code for amino acid and that codes for protein. But who gives information to form the mRNA? It is the DNA. Listen, the DNA which is there, it is inside our nucleus. It cannot move outside. It cannot move outside because if it moves outside, everything in our body is going to be disrupted because it's the main genetic material that we have. So what does it do? It transfers all this information to the mRNA. Basically, whatever information is there over here, it gives over here in the mRNA, that is known as transcription, right? It's not directly mRNA. Initially, you have hnRNA being formed. This hnRNA undergoes processing and that's how you get mRNA. And now this mRNA can easily move outside and help in the process of translation, that is this. Okay? Sri Hari, I will come to that. Okay, okay, should I come to that now itself? I can come to that now itself. Just give me a second. I think I discussed it somewhere over here. Where is it? Where is it? Yeah. I discussed it over here. Listen to me. This is the post-transcription modification. So from when we make DNA to RNA, you do not get a proper RNA. You get heterogeneous nuclear RNA, hnRNA. This RNA contains the coding sequences which are known as exons and non-coding sequences which are known as introns. Okay, got it? Basically, we need to remove these introns. We need to remove these introns and we need to just have the exons up there because that is something that we need. This is known as an mRNA. So first processing that we have is splicing. This is post-transcriptional processing. We remove the what do you call things out there from here. Okay. Okay, I'm coming up there. So next thing that will happen would be what? Capping. Capping means I am adding some group over here. 5-methylguanosine I am adding in the 5-end tail out there. And over here I am going to add some adenylene. This is known as capping and this is known as tailing. Both of them are very, 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 very important. Just give me a second. Guys, can you close the door? Okay, got it? So this is capping, this is tailing. Why is it done? Because this mRNA has to move out. And when it moves out, my dear children, it has to be protected. That is the reason. Okay, got it? So these are the post-transcriptional modification that we have. Now, Srihari, I hope you got it, dear. And NXT Sohail says, ma'am, happy birthday to your father. How do you know it's my father's birthday? That's quite surprising. Guys, and what is UTR? Where is it present? UTR is untranslated region, Sanya. I just explained it right now. So over here, it is present before and at the end of the mRNA. Before the start codon, after the end of the mRNA out there. Okay? Take a very short break. After this question, I am going to definitely take the short break. And UTR are very, very important for, as I told, for maintaining the stability, for helping in the process of translation to happen. And yes. Okay. Now, DNA replication is said to be semi-conservative. Why? This was experimented by Michelson and Stahl. This is a very important experiment. Listen to me carefully. So over here, my dear children, in the case of DNA, you have two strands, right? These strands are going to separate. These two strands are going to separate like this. Okay. And now, you will be having new strand being formed. So one strand is parental, another strand is being new. So this is why you call it as semi-conservative mode of replication. Okay, semi-conservative mode of replication. Okay, now one very, very important thing that I need you to remember is that you need to prove this thing. You cannot just directly believe it. So this was proved with the help of Michelson and Stahl experiment, where you know what they did? They grew some E. coli. They grew some E. coli in a medium which was having N15 isotope, nitrogen 15 isotope. And nitrogen is a part of what? Nitrogen is a part of your DNA. So if at all you're growing it in a nitrogen medium, obviously it will take up that nitrogen for making the DNA up, right? Now both the strands having N15 only. So which is why if at all I centrifuge the E. coli's genetic material out there, it will settle down somewhere over here because it is a heavy band. It is a heavy band. What is this medium that I am using for centrifugation? It is cesium chloride medium, CSCl. Okay, it is CSCl. Now my dear children, if I go ahead for 20 minutes, basically if I make the E. coli to grow in another medium that is having N14. Instead of N15, I am having N14 medium. Now the new DNA which will be made, it will be having N14 isotope, right? So one is green over here and one will be red. Red from the parental one and one is the green one. Now when you do the centrifugation, it is going to come somewhere over here in the middle. That means there's a hybrid band being formed. There's a hybrid band being formed. Now, my dear children, again for the next 20 minutes, a total of 40 minutes, every 20 minutes the E. coli is going to divide. For the next 20 minutes, I repeat the same thing, growing in the N14 medium itself. What will happen? From here, I get one strand. The new strand is being formed. That is also green in color because it is N14. Here, I get the red strand and the green strand is being formed again. So the parental strand is still being conserved, and that is exactly why I'll get hybrid ones and the newer ones, that is the lighter ones. If at all the parental strand were not being conserved, I would not get the hybrid strands over here. If I am getting it, that means it is being conserved. Okay, so this is the experiment that Michelson and Stahl did. He got the light and the hybrid band later. Remember, after every 20 minutes they are dividing, that is why it is written 40. So 20 minutes before, 20 minutes after, 2 rounds were done. Okay, this is Michelson and Stahl experiment. And also remember one very, very, very important thing. Okay, and that is one more experiment was done by Taylor and his friends. By Taylor and his friends, my dear children. This was done, not friends, colleagues. It was done in Vicia faba plants, that is faba beans. And this was done using radioactive thymidine. This was done using radioactive thymidine. This is one very important thing that I wanted to explain. And with that, we complete the important questions. One more thing that you need to remember in the case of transcription. Okay, in the case of transcription, always remember in the eukaryotes you have 3 RNA polymerases. In the case of prokaryotes, you just have 1. And in prokaryotes, transcription, translation can happen together in one particular time only, because why? Everything is in the same area. But in the case of prokaryotes, transcription and translation are not coupled reaction. They happen separately. So RNA polymerase 1 is there for transcription of rRNA. RNA polymerase 2 is for transcription of hnRNA. And RNA polymerase 3 is for transcription of tRNA, that is transfer RNA. So T3, 3 you can remember like that. And also, apart from tRNA, this is 5S rRNA, a different type of rRNA is there, that is 5S rRNA, and also small nuclear RNAs, that is also there. Please remember, this is super, super important. This is also super, super important. So RNA polymerase 1 is for rRNA. First preference is for rRNA only, then hnRNA, then tRNA, and also 5S rRNA and snRNAs. Hershey and Chase experiment, please go through it once. And different types of RNA polymerase, we discussed it. Genetic content in different organisms. Please know the genetic content in different organisms, such as what? You have 6.6 into 10 to the power 9 base pairs in the case of mammals and things like that. So I am giving you a short break as of now, because even I am tired. And then we will be quickly going ahead with the next chapter. Shall we go with human reproduction next? Or do you want human health and diseases? Can we get numerical problems is to be given? There are some questions which comes on the numerical basis from this chapter. I have discussed a few of them, and in principles and inheritance also you get some. Okay, and oh my God, that's so sweet of you. I didn't see this. Okay, happy birthday. Okay, Shivan, happy birthday to your father. Shalom, happy birthday to your father. Oh, that's so sweet. Mansi also. Thank you so much, guys. I'll share it with your, I'll share with my papa. Okay, I'll definitely share it with my papa. He will be really, really happy. The screenshot of this. Okay, human reproduction, human health and diseases. I see more people for human health and diseases. So we will start off that. But as of now, we'll take a short break because ma'am is super tired. And over here, how much time break do you want? 10 minutes. We'll take a 10 minutes break. So that means it is now 6:30. We will meet at 6:40. Be back at 6:40. Should I have made like this? No, I should be writing like this. 10 minutes break. 10 minutes Kit Kat break. Okay? And we will be meeting at 6:40. Be there. This is the hand. See you soon. The Kit Kat break is officially over. So, my dear children, let's be back. I hope you guys have energy, because I have come with lots of energy right now, and we are going to cover what chapter you wanted. You wanted human health and diseases. So let's cover that first. Yes, Angel Chintu. Hi, Angel Chintu. A very big hello. And Shivanand is asking, will you do live for mathematics? If at all I do live for mathematics, it is going to be a big trouble for you guys. So yes, Ankit sir is definitely going to do some live for mathematics as well. Fine. Chalo, chalo, chalo. No. Come on, let's boost up. We do not have much time. Let's start with the next chapter, that is human health and diseases. And here goes the 1 marker questions out there. The first question, human immunodeficiency virus replicates in what? B cells, NK cells, helper T cells, PMNL. PMNL stands for what? Polymorphonuclear leukocytes. This is model question paper 1, question number 7. Solve this. HIV virus replicates in where does it divide, increase in number? B cells, NK cells, helper T cells, or PMNL, polymorphonuclear leukocytes. Where? Hi ma'am, hi dear, new song by Ramzan. Uma Maheshwari is also there, says the answer for this question is C. Is this C? Answer is C. Always remember HIV. HIV, right? So HIV starts with H, and also helper T cells starts with H. In our body, helper T cells are super important to provide immunity. They are like the mediators which are there. If at all the mediators are not there, our immune system goes weak. And HIV virus just wants that, our immune system to be weak, so that they can flourish. Always remember, the first place that they go to and divide and increase the number is known as macrophage. And then later, this is known as HIV factory. This is where they will replicate, they will increase the number. Later, they will go and affect the helper T cells and start destroying them, such that our immune system goes less. Okay, so this is your answer for this question. Who all gave me the correct answer? Uma Maheshwari and also Singamanand, the beers. Singham. Yes, Singham also gave me the correct answer. Shweta dear, can we just check once? Start biotechnology. Sri Hari, biotechnology will be given in the recorded format tomorrow. I told you, from biotechnology principles and processes till biodiversity and conservation, everything will be recorded, given to you tomorrow morning, so you can watch it. Okay, watch it peacefully. Okay, so your answer for this question is C, and only 2 people gave me the correct answer. What about others? Yes, Karthik also gave me the correct answer. Sri Hari also gave me the correct answer now. Yeah, that's the Josh. Children, if you guys have energy, I also have energy. If you don't have energy, I also do not have energy. Okay? Second question, the drug which is depressant and slows down the body function is, it is a drug, but it is a depressant. It makes us sad, and also it slows down the body function. What is it? Smack, crack, hashish, or charas? Smack, crack, hashish, or charas? Crack is the other name for coke, and coke is the other name for what? Cocaine. And cocaine, my dear children, do not cause sadness. It gives sense of intense happiness. You call it as euphoria. So obviously this is not the answer. Hashish and charas, they are cannabinoids. They are also not ones that will be affecting our central nervous system. They do not affect mostly our central nervous system. So what is the answer for this particular question? Dharma is saying the answer for this question is C. C, yeah. Okay. Praju is saying the answer is A. Dharma said A first, then C. Oh my God. Prachi says the answer is B. Okay. B, Baba, crack is not the answer. Pratima says A. What is the answer for this question? It is only A. Smack is the other name for heroin. Smack is the other name for heroin. Have you seen the movie Heroin? Heroin, in that Kareena Kapoor is very much depressed. So you can remember like that. So heroin is something basically which acts like a depressant and slows down the body function. And you get it from where? You get it from Papaver somniferum. Papaver somniferum. This particular drug plant is also known as opium poppy, and this drug is under opioids. Heroin, morphine, both of them you get from Papaver somniferum. Heroin is a depressant and slows down body function. Morphine, which is there, it is a sedative, it makes us sleep, and also it's a painkiller. It is given to patients who have underwent surgery. So this is your answer for this question. Going ahead, this is opium poppy, and from the latex of the opium poppy, my dear children, what do you get? You get morphine. The morphine, if it undergoes acetylation, If it undergoes a process that is known as acetylation, you get diacetylmorphin, which is also known as heroin. It is also known as smack. I hope you will never forget it. And both of it is going to affect your central nervous system and also your gastrointestinal tract. Two things are affected by opioids. Okay. Now, symptoms of constipation, abdominal pain and cramps associated with disease, what? So, diseases are given over here: amoebiasis, richness, nostoc, ovulation, colostrum, diversity. This is your MQP1 question. Ma'am, is there a CS revision class? No, Baba, it's not there. Computer science revision class will not be there, but you have one-shot videos of computer science. You can definitely refer to that. Okay? Okay. So, I'm sorry I would not read your, your names out there. Children, if at all, you can write the answers like 3, A, B, C, D. It will be really, really good. Okay, symptoms of constipation, abdominal pain and cramps are associated with what? Amoebiasis. Over here, you have to choose from these options. So, your answer is amoebiasis. Amoebiasis is caused by Ant-amoeba histolytica. Ant-amoeba histolytica. Hi, Guru. And Shweta, you gave me the correct answer. Uma also, Prachi also, everyone. Okay. Next question. Antivenom given against snake poison contains what? So, snake, when it comes and bites you, okay, doctor gives you antivenom. What does it contain? Antivenom which is there, what does it contain? Any idea? Easy. Easy. Antivenom, what does it contain? Vijay Lakshmi also gave me the answer. My all the dear children who gave me the answer, sorry I could not read out your names, but you guys are doing awesome. Bye Dharma. Okay. Uma says the answer for this question is B. Can you guys give me the answer like 4, A, B, C, D? But anyway, I'll consider that. Antigen antibody complexes. Is that what we are given? Okay. Vijay says the answer is A. Okay. Okay. Antibodies. Okay. Okay, you guys are giving me different forms of answer. Well, the answer is antibodies. Listen to me. When you go with a snake bite means you are taking inside, antigen is being put inside your body by the snake, right? Now, you need to find the opposite of it, that is the antibodies that can fight. And you not wait for your body to produce antibodies. Rather, you should be given ready-made antibodies. And that is why doctor gives you antivenom. It contains ready-made antibodies against that venom so that you can survive. If at all you wait for your body, then what will happen? If at all you wait for your body to produce antibodies, you are going to go. Okay, so that should not be the case. And this is an example of what type of immunization? It is passive immunization. It is passive form of immunization. Always remember that. Here ready-made antibodies are given. Okay? Nikhil, ma'am, I'm CM student but watching this biology class. Oh, thank you so much. But please study CS, okay? Okay, chalo. So I hope you got the answer for this question. Similarly, if at all you're asked in tetanus also you get antitoxin given against tetanus. What does it contain? Again, it contains the antibodies itself. This is also passive immunization. Next question. The substance produced by a cell in viral infection that can protect other cells from further infection is? The substance produced by a cell in viral infection that can protect the other cells is what? Any guesses? Fifth answer. Okay, Shivanand says C. Bhai, the destroyer, says C. Important questions, ma'am. All these questions are important, dear. And please make videos for Hindi. Okay. I request you. Okay. I definitely cannot, but I will definitely ask the team to do so, okay? And Manjunath B, please, okay. So the answer for this question is C. What is the question? The substance produced by a cell in viral infection. My dear children, suppose this is a cell and this cell is infected by some virus. This cell is infected by some viruses out there. So what will this cell do? This cell is going to release some factors. What do you call these factors as? Cytokine factors. The names of these factors are interferons. You know what they tell? Interferons go and tells the other cell that, hey cell, you have to be safe. There is a virus attack happening over here. You have to be safe. It goes and tells the other cells to produce the immune cells so that other cells can be protected as well. So interferons will be your answer for this particular question. Okay, done. So your answer is C. For the next question, sixth question, you have to tell me 6A, B, C, D. 6A, 6B like that. Okay. Next question is this. Dash. Okay, there is no... Okay, dash is a very effective sedative and a painkiller. Morphine, endometrium, reserpine, fossils, vector, myometrium. What do you think is the answer for this particular question? Dash is a very effective sedative and a painkiller. What is an effective sedative and a painkiller? Any idea? Any idea out there? What is an effective sedative and a painkiller? Sedative means you feel sleepy, and it's also a painkiller. It is usually given to patients who have underwent surgery. The answer for this question is morphine. Yes. Morphine. Sana, Yasmin, Prachi, Uma, Kumuda, and Kavana. Okay. And Shivanand, Shalam, everyone. Okay. So the answer is morphine. Let's go ahead. Which of the following pairs correctly matches a disease and the pathogen causing it? So pathogen and the disease should be same. Fossilla. Okay. Typhoid, salmonella typhi. Is it correct? Typhoid is caused by salmonella typhi. Yes, ma'am, it's a bacteria. It's correct. What about pneumonia? Pneumonia is caused by Haemophilus pneumoniae. No, Baba. It is Haemophilus influenzae and Streptococcus pneumoniae. The names they have interchanged. Don't get confused. So this is wrong. Malaria is caused by Ascaris lumbricoides. No. Ascaris lumbricoides causes Ascariasis. Malaria is caused by what? Plasmodium. So this is again wrong. Now ringworm is caused by Entamoeba histolytica. Is ringworm caused by Entamoeba histolytica? It is a fungal disease. Entamoeba histolytica is a protozoan. It causes amoebiasis. Ringworm is caused by Trichophyton, Epidermophyton, and what? Microsporum. Trichophyton, Epidermophyton, and Microsporum. So only answer for this question is A. Let me see who all gave me the correct answer. Lot of you gave me the correct answer. Okay. Let me go ahead and give you the next question. Until then, I'll read out the name of the people. Uma, Vishnu, Kumuda, Soumya, Shalam, Vikas, Vishnu, and Mansi, Praju, Prachi, and Bhai the Destroyer, okay, and Ajay, all, Yasmin, Imran, all of you gave me the correct answer. Kavana also gave me the correct answer. Fine, solve this question. Hemozoin is a precursor of, hemozoin is what? Precursor of hemoglobin, toxin released from Streptococcus infected cell, toxin released from Plasmodium infected cell, toxin released from Haemophilus infected cells. What is the answer for this particular question? Until you tell me the answer in 8ABC format, I am not going to read out your names. Okay, 8C. Prachi says 8C is the answer. Okay. Mansi also says C. Uma says B. Okay. So my dear children, hemozoin is produced when? Listen to me carefully. I am going to talk about the story of Aishwarya Kamath is also there. See, it's great to see all of you guys now. Okay, listen to me carefully. I am going to say the story of Plasmodium. Okay, you have to be very, very attentive over here. So, listen, Plasmodium is a protozoan, agreed? And Plasmodium causes malaria. There are 3 species of Plasmodium: Plasmodium vivax, Plasmodium falciparum, and Plasmodium malariae. In that, Plasmodium falciparum causes the malignant or the deadly form of malaria. Now listen to me carefully. Plasmodium is there in the Anopheles mosquito. In the salivary gland of the Anopheles mosquito, you have a stage of Plasmodium. You call this as sporozoite. When the mosquito comes and bites us, it basically puts its saliva inside our body, because in its saliva you have some anti-clotting enzymes so that it can drink the blood very easily. But while it passes down the saliva, it also passes the sporozoite stage of Plasmodium. First question, in what stage is Plasmodium transferred to our body? In sporozoite stage. Now it is there inside the human body. Inside the human body, it has 2 camps. It is a terrorist. It has 2 camps. One camp is the liver. In the liver, it goes into the cells of the liver, that is hepatocytes, and undergoes asexual reproduction. When it undergoes asexual reproduction, it will form so many different type of Plasmodium out there. And now the cell which is there, it won't be able to take it up. So it will blast. And when it blasts open and releases all this Plasmodium to the next camp, which is your blood. In your blood, the second and the very important camp that we have is RBC. In the RBC, my dear children, now things are going to be very much different. In the RBC, they are going to again undergo asexual reproduction. And listen to me very carefully. Here 2 things are happening inside the RBC, and that is important. Suppose this is the RBC, you have the hemoglobin right over here, right? So this particular Plasmodium, it is going to break down the hemoglobin into heme and globin. You know what? Heme is very toxic for the Plasmodium. For the Plasmodium, it is very toxic, which is why it converts the heme to something else, which is known as hemozoin. It converts it for its own purpose into what? Hemozoin. And the same place, it is undergoing asexual reproduction to form a different stage. What is this stage? This stage is known as gametocyte stage. Okay, this stage is known as gametocyte stage. Listen to me very, very carefully. These gametocytes can later form the gametes, but that does not take place in human body. That does not take place in human body. Now, once the gametocyte has spread too much, it will blast open. And when it does, hemozoin is now released in our blood. And when it is released in our blood, hemozoin is toxic for us, which is why we get fevers and chills. And gametocytes are also mixed with blood. Now, a mosquito, when it comes and brings up some blood out there, it takes in these gametocytes. It will be taking in some gametocytes out there. And this gametocyte gets inside the gut of the mosquito, where they will form gametes and they will undergo sexual reproduction. Always remember, sexual reproduction does not take place in human body, only asexual. And here you get only hemozoin. Sexual reproduction takes place in the case of mosquito's body, and there it will be producing progenies. And again, there will be changes in the stages. There will be sporozoite stage, which will go in the salivary gland. That is how it goes on. In which cell is gametocytes being formed? Always remember, in the RBC, gametocytes are being formed. In which stage does it enter our body? In sporozoite stage. So your answer for this question becomes C. Why did I explain you this? Because a malarial parasite, the whole thing might become, life cycle might also be coming in. Okay, now Vijay Lakshmi, you have been asking me about the 5 marker. Dear, we will be coming to 5 marker. Hemozoin is responsible for fever. Yes, it is. That is why we get chills and fevers. Okay. Hemozoin is toxic for us, I told you, right? And that is exactly why we get fever and chills. Fine, let's go ahead. Which of the following is an autoimmune disease? What do you mean by autoimmune disease? Where your own cell kills your own cells. This is known as autoimmune disease. And you have options: rheumatoid arthritis, tuberculosis, malaria, AIDS. What is your answer for this particular option? Okay. Rheumatoid arthritis, tuberculosis, malaria and AIDS. Which of them is an autoimmune disease? Hi Jagannath. 9A is the answer. Okay, Kavana says A is the answer, rheumatoid arthritis. And Siddharaj is also saying A, Kumada also A, Speedax also A, Prachi. Yasmin A, Ajay, okay. Autoimmune disease, your own cells are killing your own cells. It is rheumatoid arthritis. It affects the cells of your joints. That is why you have joint pain. Okay. In AIDS, that is not happening. In AIDS, your cells are not knowingly killing your own cells. You get it? That is, another cells are taking up the machinery. But here your cells are, not able to recognize what is your cell and what is not your cell, which is why they're killing each of them. So it is rheumatoid arthritis. The answer for this question is A. Okay. 10th question, the site where lymphocytes interact with antigens and proliferate to become effector cells. Always remember, you have 2 type of, it's okay Vijay Lakshmi, if at all you do not know, you will learn like this, okay? 9A is correct. Yes, Soumya. Exactly. Mansi, Shalam, guys, do not be worried if at all I'm not reading out your name. You guys are doing awesome, okay? Okay. Chalo. Guys, do not spam. Okay, where are the lymphocytes going to interact with antigens and proliferate to become effector cells? That means they are going to show their effect. They are going to fight. Where is it? 10B. Siddharaj says the answer for this is 10B. Is it? Is it 10B? Always remember, you have 2 things. One is where they become antigen sensitive. The other is where they become effector cells. Okay. Antigen sensitive means basically it knows that, okay, this antigen is my dushman, it's my enemy. It just knows that. One is where it is going to basically attack on this antigen and kill it up. That is known as effector cells. So where are they born? Then that is the place where they become antigen sensitive. And that is what? Your primary lymphoid organs. Your primary lymphoid organs such as what? Your bone marrow as well as the thymus. This is where they mature, they proliferate and become antigen sensitive. Effector cells are the secondary lymphoid organs. For example, what? For example, you have your lymph nodes, you have your Peyer's patch, you have your spleen, you have your MALT, which occupies almost 50% of your lymphoid tissue. All of it, my dear children, all of it, lymph node and then spleen. This is where it becomes effector cells. So here, what will be your answer for this particular question in that case? In that case, Kumuda says 10B. Everyone says 10B. That is the answer for this question. Prachi says, okay. No one gave me the correct answer. Okay, someone has given me the correct answer. Praju gave me the correct answer. The answer is Peyer's patch and tonsils. So tonsils is also something. So apart from spleen, lymph node, malt, and also Peyer's patch and your tonsils, you have all this thing. Okay. Ma'am, we have tomorrow state level exam. What state level exam, dear? So your answer for this question is C. Did you get it? Everyone, I'm asking you guys. Did you get it? Hello, SpeedX. If at all you got the answer for this particular question, can you give me a thumbs up? Here you are asked about effector cells. You are not being asked about antigen sensitive cells, which is why your answer for this question is going to be C. See, Peyer's patch and tonsils. Why not the other option? Because here you have thymus, wrong. Here you have bone marrow and thymus, wrong. Here again you have bone marrow, wrong. Bone marrow and thymus are responsible for making it antigen sensitive. If at all you got this question, can you give me a quick thumbs up? Shalom, thank you so much. Thank you, Shalom, thank you, Shalom. Okay, going ahead with the next question. Injection of antitoxin in tetanus confers which type of immunization? Praju, thank you, dear. Injection of antitoxin in tetanus confers which type of immunization? If at all a person goes with tetanus and says that please give me the injection, what type of immunization is being given? Basically ready-made antibodies are given, and whenever ready-made antibodies are given, what is this immunization? Passive or active? Rupa, Prachi, thank you. Praju, everyone, thank you for giving me that thumbs up. Okay. What type of immunization? Ready-made antibodies. Is your body participating actively in producing antibodies? No. It is passive over here. So this is passive form of immunization. Your answer for this question would be B. Yes, Mansi. Exactly. Ajay Baba, you can check the answer once more. Okay. Kavana also is correct. Shalam is also correct. Praju is also correct. Siddharaj is also correct. Okay, so please go through. It is passive immunization. My dear children, suppose you are getting some infection. You're getting some infection. At that particular point of time, your body reacts, and when it produces certain things, that is active immunization or active immunity. Okay, or probably when you're getting vaccination, vaccination is what? Weak pathogens are being given to your body. And when weak pathogens are given, your body reacts to it by producing antibodies. So vaccination also comes under active immunization. But over here, you are not giving antigens, you are given antibodies, ready-made antibodies. So this is passive form of immunization, just like snake venom. Okay? Okay. Next question, question number 12. Please do this question. Tobacco consumption is known to stimulate the secretion of adrenaline and noradrenaline. The component causing this could be, what is there in the tobacco? Tell me. Chinmay also gave me the correct answer. Hi Chinmay. Raman Gowda gave me the correct answer. Goi Design Art gave me the Geo Design Art gave me the correct answer. Soumya gave me the correct answer. Navina Kumari gave me the correct answer. Varalakshmi, can you give the fixed question with marks? Dear, I already gave the video out there. It's there in the channel. It was released at 1 p.m. Please watch it. Okay, please watch it if at all you want the fixed questions video also. I will be sharing that link with you right over here. You can watch it. Okay? You can watch it later on. Well, I'm giving the answers for all those questions right over here. And I've included MCQ questions also. So this is the link for finding fixed questions. Always remember, fixed questions basically means you need to practice more, but at least gives you an idea of the most repeated concepts out there. Okay? Fine. A is correct. Everyone says A is correct. Yes, A is correct. Why? Tobacco contains A, that is nicotine, and nicotine is responsible for increasing your blood pressure. When you say adrenaline and noradrenaline is being released, that means your blood pressure is going to get increased. So if at all a question comes, which of the drug is responsible for increasing the blood pressure? It would be this. It would be this. Okay? Works? So you guys are correct while you gave me the answer. Try this question. Charas and ganja are the drugs which affect. While then I'll read out the names of the children who gave me the answer. So it was the 12th question, And I think starting from Kavana, Prachi, Chinmay, Mansi, Sunita, Shashank, Praju, Ajay, Raman Gowda, Shalam, and Pratima, Siddharaj, Yasmeen, Soumya, Varalakshmi, and Jayamma, all of you gave me the correct answer. Thank you so much. Okay, done. So the reason I didn't put the mark-wise distribution, Varalakshmi, is that there are chances that it could be here and there, and I didn't want you to get confused with that. That's why I told you, just understand the concepts of those questions which are being given, and you will be able to answer any questions that comes from there. Okay, Jagu, I'll call you Jagu. Okay, Jagu. So answer for this question is B, is what you are saying, cardiovascular system. Charas and ganja, basically, my dear children, are cannabinoids. And cannabinoids, where do you get it from? Cannabis sativa. Charas, ganja, marijuana, everything, they are going to affect your cardiovascular system. And this is one drug, listen to me carefully. Cannabinoids is a drug abused by sports people. Which drug is abused by sports people? It is this. Okay, it is cannabinoids which is abused by sports people. Super important. And it affects your cardiovascular system. Going ahead with the next question. A chemical structure is given below. Identify its plant source. Can you tell me from which plant do you get this particular thing? Okay, a chemical structure is given below. Identify its plant source. So over here, this particular thing is given. You have to tell me the thing. Any idea? Okay, by then I should be reading out the name of the people who gave me the answers. And I think it was Siddharaj, and then Manjunath is there, Shashank, and Jayamma, Praju, Prachi, Soumya, Chinmay. Chinmay dear, can you check the answer? And Jagu, and Jagu, I read out your name. Grishma, Pratima, Kavana, Jayamma, everyone did. Awesome. Shivanand, Kumuda, Chinmay. Okay, so over here the answer for this question would be what? C. Papaver somniferum. Look, this looks like an O. From Papaver somniferum, which you also call as opium poppy, you get opioids. You get what? Opioids. So which looks like O. So this is actually morphine. This is actually the structure of morphine. In your NCERT, two structures are given. One looks like an O, other has a ladder like this. And that is your cannabinoid. And that would be your cannabinoid. Please go throughout the pictures of each of them. They are super important. Okay? Now, the disease chikungunya is transmitted by what? Houseflies, Aedes mosquito, cockroach, and female Anopheles mosquito. What do you think is the answer for this question? By then, let me read out the names of the children who gave me the answers. It was C, the answer. No, Chinmay gave me the correct answer. Siddharaj, Shashank gave me the correct answer. Kumida, and Grishma, and Praju, Shashank, Kavana, and Chinmay, Sunita, Ajay, Shalam, and yes, that's all. Hi Shashank, hi Manoj. What is the answer for this particular question? It is B or D. So many options are there. B or D. Chikungunya is spread by B, Aedes mosquito. Remember, you have few mosquitoes to remember, okay? One is Aedes mosquito, it spreads chikungunya, and also dengue. Dengue is also spread by this particular guy. You have female Anopheles mosquito, it spreads what? Malaria. It spreads malaria. What about Culex mosquito? There is one more mosquito, Culex mosquito, it spreads the disease that is filariasis, or you also call this as filariasis or elephantiasis is spread by Culex mosquito. It is spread by Culex mosquito. It does not cause this disease. Okay, the causative agent is Wuchereria bancrofti. Remember, Wuchereria bancrofti. Here it spreads the virus. Here it spreads the protozoan. Here it spreads Wuchereria bancrofti. Okay, so the answer for this question is what? Just B, Aedes mosquito. Cool. Yes. Now, question number 16. Identify the enzyme which deviates from the central dogma of genetics during HIV infection. Identify the enzyme which deviates from the central dogma of genetics during HIV infection. Maths class will be on which time? Shashank, Baba, I do not know as of now. I should be talking to the team. I'll definitely let you know, okay? Okay, try this question, everyone. So many of you gave me the correct answer. It's so good. Okay. Identify the enzyme which deviates from the central dogma. What does the central dogma says? It says that from the DNA you get the RNA. From the DNA you get the RNA. From the RNA you get the protein. This is the central dogma. And DNA replicates itself to form much more DNA. So which enzyme deviates from the central dogma of genetics during HIV infection? That is the question. So over here in the case of HIV infection, HIV virus has RNA. And it gets inside our body. Our body cells have what? The nucleus has what? DNA. If it wants to incorporate into our nucleus, it also has to convert to DNA. So ulta has to happen, reverse of it has to happen, and that exactly happens. RNA converts to what? DNA, and then it gets incorporated over there. So which enzyme is responsible for that? Reverse transcriptase. Reverse transcriptase is an enzyme that is responsible for reversing the process of transcription. It is responsible for reversing the process of transcription. So your answer for this question is what? C. Soumya and Manoj and everyone else who gave me the correct answer, kudos to you. Chinmay, Shalam, I'm just reading out the names, okay? Because Rupa, Prajwal, Mansi, Jayamma, Pratima, Sangappa is there, Siddharaj also, everyone. Baba, you have Jagannath, everyone has state level, I know, that's why I'm here. Yes, I also have state level preparatory examination on Monday. Because when you have, I also have, is what I consider. Okay. Which lesson will be followed after this chapter? I think I should start with principles of inheritance and variation, Prachi. Okay, that's also an important chapter, right? We have 3 chapters which are left, principles of inheritance and variation, and then we have this thing. What do you call human reproduction, and then there is evolution. 3 chapters. Chalo, let's do. First question, name the causative organism of the following diseases. Filariasis is caused by Wuchereria bancrofti, Wuchereria malai. It is spread by Culex mosquito. Ringworm is a fungal disease and it is caused by trichophyton, epidermophyton, and one more thing. That is what? Trichophyton, epidermophyton, and one more thing is there. What is that? Microsporums, small spores. Malaria is caused by protozoan. That is what? This guy. Plasmodium vivax, Plasmodium falciparum, and Plasmodium malariae. In that, always remember Plasmodium falciparum. Okay, Plasmodium falciparum, which is there, it causes malignant form of malaria. What? Malignant, deadly form of malaria out there. Ma'am, take dew cough syrup for cough. Oh, that's so sweet of you, dear. But it's because I was talking so fast. Okay, okay, not Manoj. Okay, Mohammed Ajnas, this is Pakka question, right, ma'am? Listen, I am here to talk about all the repeated concepts that are being asked. We expect these type of concepts to come. The questions format will be changed here and there. In biology, you cannot tell that. And I'm telling you, whoever tells you that only this question is going to come, you should not be believing them as well. Okay? Please remember that. No one should be doing that. I can tell you with the repeated concepts, with lots of practice up there. I have taught PU2 for so long, my dear children, so I know that these are the repeated concepts. I can teach you that. That's the maximum thing that I can do, right? If at all I have set the paper, I could have told you that this is definitely gonna come. So yes. Okay? Chalo. So this is going to form the malignant cause of malaria. Going ahead. Mention the causative organism for the following diseases: Ascariasis, typhoid, and filariasis. Ascariasis is caused by Ascaris lumbricoides, a roundworm. Typhoid is caused by what? Salmonella typhi, it's a bacterium. Filariasis is caused by Wuchereria bancrofti, Wuchereria malai. Always remember, please know the names of all the diseases and what organism causes what. Okay? Ma'am, you will going be fast. Should I? Sunita says, ma'am, I see the physics class of yesterday, so the exam is easy. I'm telling you. Jayama, ma'am, model question paper baruta. The questions can come, may not come. But anyway, we are covering it. Okay? Next questions. So, next questions. Name the causative organism. So see, MQP1, MQP2, MQP3, all of them had this particular thing in common. They asked about diseases. So make sure you know about each of the diseases and the causative agents. It can show up. Ascariasis, again, Ascaris lumbricoides, ringworm, microsporum, trichophyton, epidermophyton, and typhoid, it is Salmonella typhi. We just discussed it. Now, important question starts from here. Okay, children, listen to me carefully. Name the group of genes that can have been identified in normal cells that could lead to cancer. There are some group of genes which are there in the normal cells, but it can lead to cancer. What are these genes? You call these genes as proto-oncogenes. What do you call? Proto-oncogenes. So basically, listen to me. A normal cell has a gene. This is proto-oncogene. Okay. Due to some problem, suppose some mutation has taken place over here, this will convert into what? A cancerous gene and cause cancer and start causing cancer. Same thing can be done with the help of a virus. Viruses are there that carry some oncogenes. Viruses are there that carry some oncogenes. If they pass on that genes to us, we will have cancer. But here you're not asked about the viruses. Here you are being asked about what? A normal cell, a normal gene which is there that can lead to cancer later on. So it is proto-oncogene. Now, which techniques are useful in detecting cancers of internal organs? What are the methods that you have to detect the cancer of internal organs? First is biopsy and histopathology. Suppose there's a tumor out there. You cut a part of it and you send for testing. You cutting a part of it is known as biopsy. Asking it for testing is known as histopathology. So, microscopic examination of tissues to detect the malignant cells is known as biopsy and histopathology. The next thing that you have is CT scan. Okay, or computed tomography. Here you are using X-rays. Here you are using X-rays to generate the three-dimensional image of the internal organs to detect the tumors. This is an important thing. Always remember, if at all you are asked specifically which is that particular diagnostic method where you need to find the internal organs, tumors and things like that, it will be CT scan. If at all it is asked in 3 marker, then you have to write all of it. Now you have MRI, magnetic resonance imaging. Over here you are not using X-rays. Here you are using non-ionizing radiations and also magnetic fields. Non-ionizing radiations are pretty much safer than ionizing radiation like X-ray. Over here, what are you going to detect? Specifically, you are going to detect any pathological defect in the living tissue. In the living tissue. Okay? Ma'am, next question, slow ma'am. Should I go very slow? Children, it is very fast for you. Should I go very slow? Okay? Fine. Now, these things you have to write. Apart from that, there is radiotherapy. Sorry, not radiotherapy, radiography, where basically, my dear children, you are using just normal X-rays, finding X-rays, that's all. Now, why are cancer patients often given alpha interferons as a part of treatment? Why is it given? Why is it given? Because, my dear children, alpha interferons are biological response modifiers. That means basically they can increase our immunity. That is why we give alpha interferons. The other interferons are different. That is being released by viruses, right? This is alpha interferons, and basically they are given to cancer patients for increasing the immunity. Okay? Studying timing, dear, this video will be saved in the channel. Yes, definitely. Shivanand, no ma'am, we cannot understand. Okay. So, I'll go slow. Okay. So, these are the questions which are important. Cancer as a whole is important. So, make sure you go through each and everything. Now, next question that we have, name the drugs, its source and nature in the given table. So, dash is the name of the drug. This is the source plant and this is the nature. So, over here, if at all I zoom this in, okay, source plant is Papaver somniferum. And the nature of this drug is what? It is a depressant. If it is a depressant and you get from Papaver somniferum, it has to be heroin. I told you heroin movie, Kareena Kapoor, sad, right? So, heroin is the drug that we get from Papaver somniferum and it is going to be a depressant. Okay? And next, cannabinoids is given. What is the source plant for cannabinoids? It is Cannabis sativa. What is the effect on it? Effect is on cardiovascular system. The only thing that affects your cardiovascular system is this. Okay. Now, next thing that we have is cocaine. Cocaine you get from Erythroxylum coca. What is the nature? What does it affect? It affects your central nervous system. Okay, it affects your central nervous system. This is super important. Especially in a central nervous system, it is going to affect the transmission of dopamine. If dopamine transmission goes on continuously, you get a sense of euphoria, and later after that, you will have hallucination. You will start seeing cow flying and everything happening. You will start seeing the cows will be flying and everything is there. Okay? Got it? So heroin, cannabis sativa, and effects on the central nervous system. Cool? Now, this question, I hope it's clear for you. Mallika, Jasmine Joy. Yes, dear, tell me. How do you know my name so nicely? Okay. Let's go ahead. We have described any 3 properties of cancer cells. 3 properties of cancer cell. This is one of the most repeated question out there. Okay. And Kannada alli madi, ma'am. Kannada alli mali. I told you I am learning Kannada. Nanu Kannada kalita idini. I have not learned it yet, but I definitely understand a few of things. Okay. Okay. Going ahead, here, 3 properties of cancer cell. One is they show uncontrolled growth. They keep on dividing. And the second property is related to this. Listen, a normal cell, when it is dividing, if it comes in contact, they stop their division. And this is known as contact inhibition. But over here, they lose the contact inhibition. They lose the property of contact inhibition. So this is the next point out there. Now the third point, they will spread throughout our body and keep on transferring the disease elsewhere in our body, and that is known as metastasis. Okay, children, guys, and hi Tarak, children, can you be restricted to this particular video? Listen, I already told you, there is one very, very important thing. If at all your energy goes down, my energy goes down. If you get deviated, I get deviated. So please don't do that. Okay? Fine. Now, what are lymphoid organs? Write the functions of the following in response to immune system in our body. I told you lymphoid organs are where the lymphocytes are going to develop. Okay, they are going to develop, they are going to show off their action. Okay, they are going to show off their action. Now here, you had 2 different type of lymphoid organs. One is primary lymphoid organs. Remember, I told you, one is where they become antigen sensitive. That means they know that this guy which is there is my enemy. But it does not act. And over here, it forms, it matures also. So these things are known as the primary lymphoid organs. These are bone marrow and the thymus. You know, bone marrow is a place where all your cells are being formed. Okay, bone marrow is a place where all the cells are being formed. And then later on, these cells, some of the cells, for example, B lymphocytes, they mature here itself. But some of the cells, they move to the thymus for maturing. And those are known as T lymphocytes. So this is where you got them antigen sensitive. Now, there is one area where they are going to show their effect. There is one area where they are going to show this effect. So this is known as being effector cells. And what is that? That is your secondary lymphoid organs. And what all the things are there? You have spleen, you have lymph nodes, you have Peyer's patch. So Peyer's patch is in your small intestine, and then you have tonsils. Tonsils also. Why do you call secondary lymphoid organs? Because here, my dear children, they are going to be there in so much amount, and they are going to fight with the other antigens out there. Okay, so please remember that. Ma'am, please, computer science, important questions. Dear, I will talk to the team about it. Please do not spam. If you all you spam, I cannot see others' thing. Okay, I'll definitely talk to that. Talk about it. Hari Prasad, yes dear, I am a Christian. Okay. I am a Malayali, I am a Christian. Okay, now coming up over here, lymphoid organs, primary organs. So organs where organs where the lymphocytes origin, maturation, proliferation of the lymphocytes occur. Primary organs are bone marrow and thymus. They produce antigen-sensitive lymphocytes, which migrate to what? Secondary organs such as spleen, lymph nodes to become effector cells after antigen exposure. Okay. Now, bone marrow which is there, it is the main site where all the blood cells, including lymphocytes, are being formed. And it provides microenvironment for the B lymphocytes to mature also. Thymus, which is there, it is again a primary lymphoid organs for T lymphocyte maturation. It is where they become antigen sensitive T lymphocytes and later differentiate to become effector cells. Not here, they go to the secondary lymphoid organs to become effector cells. Okay, got it? And thymus, my dear children, it goes slow. It grows. While we age, it will be growing smaller in size. Going ahead, you have spleen and the lymph nodes. Spleen is a secondary lymphoid organ that filters blood and traps the blood. You know what? Spleen which is there. Okay, spleen which is there, it is going to trap any antigen. trap any antigen, and the spleen is having so many lymphocytes. So if any antigen comes in, the lymphocytes basically kills it. So that's why it is known as secondary lymphoid organs. Apart from that, you have lymph nodes. So lymph nodes is spread throughout our body. You know, lymph nodes is spread throughout our body like this. If at all you have seen, you have your blood vessels, right? You have your blood vessels, okay, such as your arteries opening into capillaries and forming the veins. So in between that, some leakage can happen. Some leakage can definitely happen. And who is the one that will be collecting this leaked fluids and bringing it back to the heart? It is basically the lymphatic system. And the lymphatic system, which is there found in green over here, will be having certain areas where there are so many lymphocytes. And those areas are known as lymph nodes. Over here, if at all you touch over here, doctors go, whenever you have fever, they touch over here. Why? Because they are seeing if at all your lymph nodes are swollen. If at all it's swollen, it means that basically they are having dishum happening up there. Okay? Okay? Cool. Can we go ahead? Now the next question. This is what? Give the schematic representation of replication of retrovirus. Now let me read out what is there. My dream is to score 70 on 70. I know, I get it. You will get 70 on 70. Manoj is asking, what is my name? Okay, what is my name? My name is Jasmine. Okay. Nanni ma'am. Shashank. Thanks so much. Okay. I understand, Mank. Okay. Studying time, please do not spam. If at all you spam, I would have to restrict you from this chat session. So do not spam. I get it. You know, I am your teacher. I know that you're facing trouble in computer science. I'll ask my team to help you with that. Okay, I will definitely ask my team to help you with that. But as of now, it's not in my capacity. Okay? Works? Okay. Let's go ahead. Now, give the schematic representation of the replication of retroviruses. How do you explain that? Listen to me carefully. So over here, you have the retrovirus. This is a super important picture. This is a super important picture, and you will get so many questions related to it. So the virus has RNA. The virus has RNA. Suppose this is a macrophage that is going to come and affect. The RNA which is there, it is of no use here. Why? Because in our body, our cells have the DNA. So if at all it wants to use our machinery, it has to convert its RNA into DNA. This is reverse transcription. Who does that? It is reverse transcriptase that does that. And now you have your DNA, the viral DNA, it incorporates into our DNA with another enzyme out there. And now our machinery is used up. Our machinery is used up to produce what? To produce viral RNA, to produce viral protein, everything. And now viral protein, viral RNA, everything assembles, and now you have small baby viruses which goes and affects the other cells. This is how retroviruses work. And you should know this picture. Make sure you practice it very, very well. Okay? Fine. So this is the HIV life cycle, Ravi Shankar. I hope it's clear. I hope it's clear. And now, there is a question that I will show you related to this only, which came this year, and that is this. The flow chart shows replication of retrovirus in the host. Answer the questions given below. So what is the answer? Let's see. Retrovirus. Virus infects the normal cell. It infects the normal cell. While it infects, viral RNA is introduced into the host cell, that is our cell. Reverse transcription happens. After reverse transcription happens, what will happen over here? If reverse transcription happens, what is going to happen over here? Viral RNA is going to convert into viral DNA. Viral DNA, you got the viral DNA over here. Now the viral genetic material in the nucleus of the host, it goes, incorporates into the nucleus of the host. Now what will happen? Basically, you will produce viral proteins, viral RNA, and that will come out of the viruses will be coming out of the host. That's how it goes. Okay? You have got a question. Guys, please wait a second. I will come and I will read out everything. I'm just explaining something. Okay? So fill in the missing data in the boxes labeled as 1 and 2. We know 1 is what viral DNA is being formed. Box 2 is what viral components such as proteins and RNA are being formed. Now give an example for retrovirus. What is the example for retrovirus? Arey Baba, it is HIV. Okay, it is what? HIV, human immunodeficiency virus. Now name the disease caused by the retrovirus. It is AIDS, acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. And now name the diagnostic test for this particular disease. What is the diagnostic test? It is ELISA, enzyme linked immunosorbent assay. What is ELISA based on? It is based on antigen antibody binding. It is based on antigen antibody binding. Okay. So Prashant, ma'am, you're awesome. Thank you so much. And Kumuda, one viral DNA. That's what you were saying. Yes, I hope I have not missed anything. Ravi Shankar, I have explained the life cycle of Plasmodium. Please, can you see it? Go back and check it out. It's there. Okay. And okay. Anita, ma'am, from this question, can I get 40 to 50 marks? From, if at all you solve these questions with me, if at all you are someone who is practicing with me, you get to understand the concepts. These repeated concepts are usually being coming, so you may get really good marks. Okay, really good marks. Okay, done? Okay. Fine. I definitely understand studying time, but I'll definitely talk to the management if at all there is any possibility for you to get it. Okay? But I'm not sure. I'm really not sure. It depends on the availability of the CS teacher out there. Okay? Tarak, how many hours class is there? There are 3 more chapters and then we are done. Okay. Cool. Let's go ahead. So over here, I had some questions. Yes. Differentiate innate immunity and acquired immunity. What do you mean by innate? Innate is something that we have from our birth. For example, I have my skin, I have tears, I have mucus. These are all part of my innate immunity. It is from birth. It is non-specific. Does the skin say that virus you enter, bacteria you do not? Can it say that? No. It is non-specific. Whereas when you talk about acquired immunity, we acquire it throughout our lifetime. Okay, we are going to acquire it throughout our lifetime. And over here, it is very specific. If this antigen comes, I have to produce these antibodies. If this comes, I have to produce these cells. It is very much, what do you call, specific out there. So difference between innate and acquired immunity goes like this. Listen to me carefully. Innate is present from birth. Acquired develops after an infection, or basically when you get vaccination. Exactly. Kavanna. Okay, now going ahead. It is non-specific over here. It is specific to particular antigens in the case of acquired immunity. Here, no memory is there. Does your skin remember what has come, what has not? No. So there is no memory. But over here, since so many cells are being produced, especially acquired immunity means 2 different types of cells are being there, B lymphocytes and T lymphocytes. B lymphocytes produce antibodies, and T lymphocytes are super important because they directly go and kill the cells, or they help the cells to provide immunity. So acquired immunity, my dear children, they have immunological memory. Since these cells are being produced, there is some memory. Because when antibodies are released, there are one more thing which is being released, that is memory cells. And these memory cells, my dear children, always have an idea of what thing has come before and what are the things that I need to produce now. Okay? Yes, Kavana. Alpha Sonu, please once again explain. Okay. Okay, so over here, explain once again, innate immunity, it is present from birth. Your skin, your mucus, everything is a part of your innate immunity. Acquired is something that you acquire after an infection. I have got that immunity after an infection. For example, coronavirus came. Not everyone was able to sustain it. Why? Because your body reacted to it, but it was not winning. So in some people they were living. Why? In their body, the antibodies fought. Okay, so it is developed after your infection. Once more, if coronavirus comes, your body knows that it has come before and what antibodies do I need to produce this time. Okay, over here it is non-specific, does not know, does not have a specific action, whereas here it is very much specific. It knows what antibodies to produce against what antigens. There is no memory over here, but it has immunological memory. Please go through different barriers of innate immunity. There is cellular, cytokine barriers, and there is physical and physiological barriers as well. Okay? Fine. Kavana, innate immunity is to produce army of protein called antibodies. No, Baba. In innate immunity, there is no antibodies. In innate immunity, there is no B lymphocytes. Listen to my question. Listen to my thing very carefully. Acquired immunity is where you have 2 types of immunities. Listen, this is simple. There is 2 types of immunity in acquired. One is humoral immunity and one is cell-mediated immunity. One is humoral and one is cell-mediated. In humoral immunity, you have B lymphocytes. What lymphocytes? B lymphocytes. They produce antibodies and they also produce some memory cells. Cell-mediated immunity is produced by T lymphocytes. T lymphocytes are of different types. There are T cells, T cytotoxic cells, T helper cells. T helper cells are mediated for immunity. They tell the other cells to release, things like that. Whereas over here, T cytotoxic cells are going to directly kill any other cell which is going to be affected. Okay, that is how it works. Here also memory cells are being produced. So once the body has a memory that some attack has taken place, the next time it will be more active. So the primary response is going to be slower. The second response or anamnestic response is going to be very much nice. Okay, always remember, if at all, if a person is undergoing some graft, okay, that is basically some organ transplantation. Suppose I am taking kidney from someone else, my body will react to it. My body will react to it. Who is responsible for this reaction? It is the T cells. It is the T cells, because our body feels that it is a foreign thing out there. Okay, so acquired immunity, humoral and cell-mediated forms the acquired immunity. Innate means you have different type of barriers. I'm telling you, please, if at all you have an NCERT, I'm telling you, when I started off this particular session, I told you, have your notebook, write the topics that I'm telling you to revise. And that is, please go and revise the different barriers of innate immunity with all the examples. What does PMNL stand for? Everything. Okay? Okay. So now the next question. Mention the cells involved in accomplishing the acquired immunity. What are those cells? B cells and T cells. B lymphocytes and T lymphocytes. Children, I will be reading out your comments, okay, later on. I am teaching something right now. Now over here, name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind to the A and B sites. Over here you have a picture. And here the question is, name the molecules that are trapped in the lymph nodes. And they are responsible for the activation of lymphocytes. These trapped molecules are responsible for activation of the lymphocytes. So what are these molecules, my dear children? What are these molecules? These are basically antigens. Remember, this picture of antibody is super important. This picture is super important. You might be asked to draw it as well. You might be asked to draw it as well. This is the heavy chain, this is the heavy chain, these two are the light chain. Over here you have the antigen binding site. What do you have? You have the antigen binding site, right? And over here antigens comes and binds, and that's when it will be active. It is going to be active. So what is something that is trapped in the lymph nodes that activates this? It is antigens. Now what does H2L2 in an antibody stand for? H2 means two heavy chains, L2 means two light chains, and this antigen binding site which is there, this is where antigens comes and bind. Okay? Okay. Ma'am, it is the ability of immune system to fight pathogens is called immunity. It is correct? Yes, it is correct. Okay? Pudu Varsha Ashamsagal. Thank you, Tarak. Same to you. Happy New Year. Okay. Now, name the cells involved in humoral and cell-mediated immune response. Humoral immune response, what is the one responsible for it? B lymphocytes. It produces antibodies that circulates in the blood and the body fluids. Now, cell-mediated immune response is also there. It is carried by T lymphocytes, or what you call as T cells. And here no antibodies are produced. T cells directly attack. And always remember, if at all there is graft rejection that is happening because of this particular guy. Okay. Yes. And since we are talking about, antibodies, I want to tell you one more thing. There are how many type of antibodies? Remember, G-A-M-E-D, gamed, okay, gamed. 5 type of antibodies are there. The first antibody to transfer from the mother to the baby via the placenta is IgG. When you're giving ready-made antibodies, it is the example of what? Passive immunization. Again, if we talk about A is the second antibody which is transferred from the mother's milk to the baby. So A for Amma, you can remember like that. Okay, this is the second antibody. Again, this is an example of passive immunity. Ready-made antibodies are given. When you have allergy, what is the antibody being released? It is E. Antibody E. Allergy, antibody E. IgE. Antibody, you also call it as, what do you call it as? Immunoglobulin. They are proteins. Okay, they are proteins. Okay, now listen to me, one very important thing. Mast cells which are there are mast cells, you can call them mast cells also. Basically, they have the antibodies, that is IgE. When this IgE comes in contact with any other allergen, the mast cell is going to release some things. One is histamine, and the second one is serotonin. Histamine and serotonin. And you know what? This histamine is going to cause your blood vessels to dilate, which is why there will be a lot of immune cells coming up, and you will be having, okay, immune cells will be coming up, and you will be having a swelling and things like that. That is why we say that in allergy you have got swelling. So what is the way in which you can treat it? What is the way in which you can treat it? You give antihistamines. Remember, this is also an important question. You give antihistamines. Second thing, you give adrenaline. Third thing, you give steroids. These are the three ways where you can treat allergies. Okay, Mohammed dear, what you couldn't understand? What is the thing that you couldn't understand? Can you please tell me now? Nagaraj is here. Hi, Nagaraj. Okay, so G and A passed from mother to the fetus, one through placenta, one through milk. The milk is known as colostrum. And E is produced during allergies. Remember that. And now the name of the drugs, I hope you know the name of the drugs. Please go through all the drugs. If at all you are forgetting something somewhere, do not do that. Okay, so I will add a page up over here, and I will just finish off this particular topic, and then we are done with this chapter. Okay, listen to me very carefully. You have opioids. You have cannabinoids. You have coca alkaloids. Both of them affects your central nervous system, CNS. Here, apart from CNS, it also affects your gastrointestinal tract. Okay, here in the CNS, it affects your dopamine transfer, which is a neurotransmitter. Okay, here in cannabinoids, it affects your cardiovascular system. Okay, here you have two drugs, one is heroin and one is morphine. When morphine undergoes acetylation, that's when you get heroin. Heroin, which is there, it is a depressant, and it is a depressant, and it slows down your body. Morphine, which is there, it is a sedative, it makes you sleep, and it's a painkiller. It is given by doctors to the patients who have underwent surgery. Okay, now this is also known as smack. You get this from the latex of opium poppy. Over here you have multiple things, charas, ganja, marijuana, everything comes under cannabinoids, and you get it from cannabis sativa. The leaf of this particular plant is given in your NCERT. Go to the picture of this. Cocoa alkaloids, since it is affecting the dopamine transport, you know what happens? It is going to create euphoria. You get a sense of happiness because dopamine is responsible for happiness. Later, it causes hallucination. If in more doses, it causes hallucination. And this is where comes the next drugs that you need to remember, which mostly children do not. There are three plants which causes hallucination. One is your erythroxylum coca, that is giving coca alkaloids. Erythroxylum coca. Please remember, it is native to South America or North America? South America is what I believe. Can you check and let me know? In your NCERT, can you check and let me know? Erythroxylum coca, it is native to America, North or South? I'm not able to remember it exactly. Please check it out and let me know right now. It's there in the paragraph. Okay, it is super important. You have got questions like that. Now, that is one plant. Apart from that, there is another plant that can give you hallucination, and that is one is Atropa belladonna, and the other one is Datura and Atropa belladonna. Datura's picture is also there. Please go through it. Okay. Yeah, South America, exactly. So it is South America. Okay. These three of them are hallucinating plants, or gives hallucinogens, my dear children. Apart from it, you have three other drugs. Barbiturates, amphetamines and benzodiazepines, BAB, they are there for treating insomnia. That means if at all you are not able to sleep, then this is given. And also it is able to treat depression. It is there for treating insomnia as well as depression. That is benzodiazepines, amphetamines and barbiturates. Always remember that. Apart from it, you have tobacco. Tobacco has what? Nicotine. Nicotine, and nicotine increases your blood pressure. Nicotine increases your blood pressure. The drug which is mostly abused by sports people is cannabinoids. Sports people. Apart from it, please remember the effect of, okay, the effect of this particular thing, anabolic steroids on male and females. Anabolic steroids on males and females, please go through that. This is also important. Towards the end of your chapter you have it. In the case of females, it causes masculinization, you get acne, you get mood swings and all of it. In the case of females, in the case of males, again you have depression, mood swings. Apart from that, you would be also having lower testicular size, less rate of spermatogenesis, that also happening. Please go through that. Okay, I think IHG got a different. So this is not a physics lecture by any chance. If at all you felt it is, I'm so sorry. So by this we end up the chapter. That is what, if at all you want to take a screenshot, you can. That is human health and diseases. And now we are getting off with the next chapter. Okay. My dear children, I hope this video is helping you. What is the next chapter that we should be going ahead with? I think it's principles of inheritance and variation. So where is principles of inheritance and variation? It's right up there. Yes. Important diagrams of preparatory examination. Dear, it's there in the fixed question video. The important diagrams. Okay, the video has been released in the channel. You can watch it in the video section. Ma'am, great biology session. Could you please request the team to do a session on second PUC? Yes, definitely, Prithvi. We'll definitely try. Okay. Okay. Principles of inheritance and variation. We are soon going to begin that. Starting off with the first question. Here it goes. Progenies with A, B and O blood groups are not possible from which of the following mating parents? Okay, progenies with A, B and O blood groups are not possible from which of the following mating parents? You do not want A, B and O to come. You do not want A, B and O blood group to be coming in. Okay, Kavana says the answer for this question is A. What do you think is the answer? This is an MQP1 question, model question, paper question. A, B and O blood groups are not possible from. Listen, for anyone to have AB blood group, one should be coming, A should be coming from one parent and B should be coming from another parent. Right? That is one thing. So we should avoid any blood groups that have A here and B there on the opposite sides. Okay, let's see. Here you have A, here you have B, I cannot have this. Here you have A, here you have B, I cannot have this. Here you have A, here you have A, here you have B, I cannot have this as well. What is the only option that I have? And that is this, IO. And that is this. This is the way where I cannot have parents, progenies with AB or O blood group. Let's try it out. How can we try it out? Let's see. So if someone has O blood group, it will be, the genotype will be IO. If someone has AB blood group, the genotype will be IA, IB. Now, if at all you are fusing them, whatever you do, it will be IAIO. A is dominant over O, so this will be A blood group. This and this, IBIO. Again, B is dominant over O, so B blood group. This and this, again, A and O. This will be A blood group. This and this, B and O, this will be B blood group. There is no AB, no B, no O blood group. So which is why your answer for this particular question would be this. Okay, Ravi Chandran, ma'am, can you give a simple definition for fetal ejection reflex? It is the process by which fetus is being, okay. It is a process by which the fetal is going to be moving out of the uterus, and over here there are combined effects of what? Both the fetus as well as the placenta. Okay, so your answer is A. Going ahead with the next question, try this question and by that time I can give you a proper definition here. So what is the possible blood group of an offspring when both parents have AB blood group? If both parents have AB blood group, guys, can you tell me the answers like this? 2, A, B, C, D, whatever is it? Can you give an answer like that? 2B, 2B, 2B is everyone saying. Okay, 2B is basically this, A, B and AB. So what is a possible blood group of offspring when both parents have AB blood group? If A and B is there, this and this you get AA, that means A blood group. This and this you get AB, that means AB blood group. This and this you get AB, that means AB blood group. And finally, B and B, that means it is B blood group. So obviously A blood group is possible, AB is possible and B is possible. You guys are absolutely correct. Okay, you guys are absolutely correct. Are you? Why I have marked this? No. This is wrong, okay? The answer is this. Okay. Third question. Gene located very close to one another on same chromosome tend to be transmitted and are called as what? Tell the answer for this and by that time I'll read out the name of the people who gave me the correct answer. Mansi, Kavana, Prachi, Shashank and Nagaraj and Tarak, Shivanand and Pratima. Everyone gave me the correct answer. Ravi Shankar also. Okay, try this question. Rupa also. Pallavi also. Genes located very close to one another on the same chromosome tend to be transmitted and are called what? Okay, 3C. C is what? Linked genes. Are they known as linked genes, my dear children? Huh? It is linked genes. Let's see. So over here, if I am saying, this is very, very important, okay? Listen to me very close, carefully. So over here, I have two genes, okay? Here I have A, here I have B, here I have small a, here I have small b. Now suppose there is interchange happening up over here. When it happens, my dear children, what is going to happen? The capital B and small b remains to be the same, but here there is an exchange. Small a comes here and capital A goes there. So you find what? New combination. This is known as recombination. When does recombination happens? When the genes are far away from each other. You call this as loosely linked. You call this as loosely linked genes. Okay, on the other hand, suppose the scenario is something like this. Small a, small b is over here, capital A, capital B is over there. Exchange is happening. Whatever happens, my dear children, capital A, capital B is going to remain at the same position and small a, small b will also be together. That means they are tightly linked. The tightly linked genes which are there, they show lesser recombination. They show lesser recombination. So over here, the genes located very close to one another on the same chromosome tend to be transmitted together. Why? Because they are tightly linked genes and they are known as linked genes. C is your answer for this particular question. Okay. Now, going ahead, try it out. What is the answer for this question? Can you try it out? Which three scientists independently rediscovered Mendel's work? Which three scientists independently rediscovered Mendel's work? Who are the, and fetal ejection reflex is the natural reflex that causes strong uterine contractions to push the baby out during the labor or the childbirth. This is the easiest definition I got from ChatGPT. Okay. Now, hello ma'am, send the passing package of PIU. Dear, I have given you the fixed question. You can watch the video. That video is already out. Okay, so sorry I could not read out the name of the children who gave me the correct answer for the previous question. Let's try for this. Okay. Who were the three scientists who independently rediscovered Mendel's work? It was De Vries, Correns and Schemark, and this happened in the year 1900. Okay, this happened at this particular point of time. Okay. Going ahead, we have this question. A plant having the genotype capital A small a, capital B small b will produce dash kinds of gamete. Dash kinds of gamete. Solve this question for A, B, C, D. Try this out. Try this out. Okay, you started off. This is fourth question, okay? This is fourth question. Try this out. D is what you are saying. So, Naga Bhushan is saying D, Kumuda is saying B, okay. The type of gametes that will be produced over here, if at all the genotype is like this, what will it be? Someone is saying B. Most of you guys are saying B, so let's check it out. Listen, over here there is a formula, 2 raised to the power n. N stands for number of heterozygotes. Number of heterozygotes. So over here, capital A small a, this is one heterozygote because the alleles are different. Capital B small b, this is another heterozygote. Here also alleles are different. So 2 heterozygotes I have. So 2 raised to the power 2 is equals to what? 4. So the answer for this question would be B. The answer over here will be what? B. 4 gametes are going to be formed. Okay, so Tarak, Nagraj, Anita, Vinuta and Naga, Prachi, Prashant, every one of you are absolutely correct. Okay, Sikandar also, all of you. If at all I'm missing any names, please forgive me, okay? Fifth question, try this question out. Fifth question, fifth A, B, C, D, what is the answer? Which of the following is not a pair of opposing characteristics Mendel studied? Which is not a pair of opposing characters that Mendel studied? Axial and terminal, full and constricted, green and yellow and pink and white flowers. Which is not a pair of opposing character? Axial and terminal flowers. Axial means from here, from the place where a branch has to come, a flower is coming. That is axial position. Terminal means at the tip it is coming. Okay, which one is dominant? Axial is dominant. A comes before T, so you can remember axial is dominant. This is correct. Full and constricted. Now the pots where you have the, my dear children, you have the green piece. It can be full or it can be very dry and constricted like this. So which one is dominant? It is full one. Now green and yellow pots. Green is dominant because when you go to the market, obviously you take the green pots rather than the yellow ones, right? So green ones are the dominant ones. In the case of seed, it is ulta. Seed, my dear children, seed may yellow is dominant and the green is recessive. You might have seen in the experiment done by Mendel, right? So pink and white flowers is a wrong thing because it's not the pink flowers, it is violet flowers. Violet, V, okay, violet is the dominant one and the white flowers are recessive ones. So this is the pair of, not the pair of opposing character, rest everything is. Got it? Yes. Please go through the entire thing. It is super important. How many pairs were there? 7 pairs were there. Now next thing, chromosomal maps or genetic maps were first prepared by. Chromosomal maps or genetic maps were first prepared by Sutton and Bovary, Bateson and Punnett, Morgan or Sturtevant. Who gave the genetic maps? My dear children, I'll tell you one thing. If at all you are my student and you do something great, won't I remember it? Obviously. Similarly, Thomas Hunt Morgan, who was working on chromosomal theory of inheritance, came up with this one particular thing, that is linkage and recombination. Very far genes will have very much recombinations out there, right? And if the genes are very far from each other, they show more recombination, right? And if at all the percentage of recombination is more, I understand that they are far, and with the help of that I can locate the genes. So who was the person who was able to locate the genes and map them? It was the student of Thomas Hunt Morgan, and that is Alfred Sturtevant. Who all are giving me the answers correct? Let me see. Oh my God. So there are so many people who are giving me answers correct. I hope you're getting it. I hope you're understanding it. It's not Morgan. Morgan gave linkage and recombination. The student of Morgan gave genetic mapping, and that is Sturtevant. Okay, with the percentage of recombination, he got to understand how much far they are, and that's how he mapped the genes. Got it? Okay, children, getting it, understanding it? Is it going very fast? Is it difficult for you to understand? Please let me know. And if at all you have not subscribed to the channel, subscribe to it, and do not forget to hit the like button also. Let's make it 200 this time. Possible? Okay, answer is D. Question number 7. Which of the following is incorrect regarding ZW-ZZ type of sex determination? Which of them is incorrect regarding ZW-ZZ type of sex determination? It occurs in birds, females are homogametic and males are heterogametic. Where do you see it? What is happening? Listen, ZW and ZZ is there. Listen, over here it is the females that is going to produce two different type of gametes. Males are going to produce same type of gametes. This is known as, this is an example of female heterogamety. Female heterogamety, two different type of gametes being formed. Okay, one is Z and the other one is W. And my dear children, you can remember it by W for women. W for women. Okay, so they are the ones which are females over here. And this is seen in the case of birds. This is seen in the case of birds. Ma'am, going speed, I'll slow down. I will slow down, okay? So it occurs in birds. This is correct. You have to find what is incorrect, right? You have to find what is incorrect. And females are homogametic. Is that possible? Females are not homogametic. They are heterogametic. Females are heterogametic and males are homogametic, correct. Total number of chromosomes is same in both male and female. Yes, the number of chromosomes is going to be same. Abhishek, hello dear, namaste. So over here, what is wrong? The only statement that is wrong is B. Yes. So Prachi, Nagaraj and Kumuda and Vikas gave me the correct answer for this particular question. Please get your answer correct. Here you have been asked about incorrect one. Okay, you are asked about incorrect. Arun, Aruna and Nagaraj, okay, all of you. Good, good, good. Going great. Now question number 8. Okay, question number 8. Try to solve this. This is important. A woman with normal vision whose brother is colorblind marries a man with normal vision. A woman who is having normal vision, okay, whose brother is colorblind. Her brother is colorblind, marries a man with normal vision. Okay. They have 4 children, 2 sons and 2 daughters. One is colorblind while the other son and both daughters have normal vision. The pattern of inheritance for the disease is what? The pattern of inheritance for both the diseases. First of all, it's color blindness, and color blindness is what? It is sex-linked recessive disorder. You need not study all this thing and blow up your head. Color blindness is a sex-linked recessive disorder, so please go ahead with that. That's all. Okay? Got it? Abhishek, ma'am, you're so beautiful. Thank you so much, dear. You're also beautiful. Okay? So your answer for this question is nothing else apart from it. 8A is the answer. Those of you getting your answers wrong, dear, color blindness, hemophilia. Color blindness, hemophilia are sex-linked recessive disorders. Males are going to be affected more than females. Remember that. Okay, in your NCERT, no sex-linked dominant disorders are given. So don't worry about it. Next question. If both parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? If both the parents, okay, if both the parents, my dear children, are unaffected carriers, that means basically I am saying there is HBA and HBX. Suppose this is HBA and this is HBX. Listen, it is an autosomal recessive disorder. So that means if X is something that is causing the problem, then the people who are having something like this, both the alleles are going to be the same. That's when a person will be having the disease. Now here parents are the carriers. That means both of them are having this allele, but they are having a normal allele as well. They are having a normal allele as well. Okay. Now suppose you're asked what are the chances of pregnancy resulting in an affected child. So here male and female, anything can be affected. But let's see how much percentage will be affected. So this is HBA, HBA. That means it is a very normal child out there. Over here, HBA and HBX. This is going to be a carrier but still normal, just a carrier. Here, HBA, HBX again a carrier but normal. Last, HBX and HBX. Here the person will be having the disease. Here the person will be having the disease because both the alleles have come together. It is an autosomal recessive disorder. It will be only seen in recessive condition. Now listen to me very carefully. over here, what are the chances of pregnancy resulting in an affected child? Out of 4, only 1. So 1 by 4 is equals to what? 25%. So your answer would be 25%. Your answer is A. Who was saying D? Is A. It is A. Okay. Manasalayo. My dear children, do not panic. So when you get your questions wrong, write it somewhere that this is the question that I got wrong. I have to come and revisit it again. That's how we study. Okay, it's 25%. Okay? Let's question. Okay, now we do not have any MCQs. We directly have questions. You will be seeing 2 marker as well as 5 marker. So are we ready? Are we ready? I hope we are. Let's begin. First one, a 2 marker question which is there in the MQP1, model question paper 1 of this year, and that is recombinants obtained in both Mendelian as well as Morgan experiment, yet the processes involved are different. Comment. Recombinants obtained in both Mendelian and Morgan's experiment, but the processes involved are different. Comment on it. So basically in Mendelian's experiment, in Mendel's experiment, he got recombinants because of incomplete, because of this thing that is known as, what do you call, independent assortment. Okay, now you will not understand this thing until and unless I explain you something else. So for that, I would have to go down a bit. Not a bit, a bit more. So listen, we will study the interaction pattern in a dihybrid cross, and there we will be understanding how does recombinants come into picture according to Mendel. So initially, my dear children, 2 plants were taken. One was round and yellow, and one was wrinkled and green. Now listen to me very, very carefully. Okay, round and yellow was a dominant trait, so it was like this. Wrinkled and green is a recessive trait, so it is like this. If at all you want to make the chromosome, it appears to be like this. Capital R, capital R, capital Y, capital Y. So from this, from each pair of chromosome, only one is going to come in the gametes. Why? Because only half the number of chromosomes are going to carry in the gametes, right? So from here, either way, whatever you do, you get capital R. From here, whatever you do, you get capital Y. Similarly, from here also, whatever you get, you are going to get small r and small y combination. Right? Now when these gametes are going to fuse in the F1 generation, what is the new thing that you will get? You will get capital R, small r, capital Y, small y. Capital R, small r, and capital Y, small y. Now how does it appear to you? One is capital R, another one is small r. One is capital Y, another one is small y. This is how it appears in the F1 generation. Now we need to cross the F1 generation again because Mendel wanted to see what happens in the F2 generation. So when we cross it, let's see what happens. So I am going to erase this up. If at all you want to take a screenshot, you can. Okay. So when I do that, listen to me carefully. When capital R, small r, capital Y, small y is taken, similarly capital R, small r, capital Y, small y is taken, what exactly happens up over here? Capital R, small r, capital Y, small y. Okay. From here, one can come. So it can be capital R. From here, one can come. It can be capital Y. From here, one can come. That is capital R. From here, small y can come. Next combination that we have. From here, small r can come. From here, capital Y can come. From here, small r can come. From here, small y can also come. These are the 4 combinations that one can get of the gametes. 4 type of gametes can be formed from here. 4 type of gametes will be formed from there. Now, if I put them both in this direction, if I put here 4 of them and here 4 of them, and if I fuse them, capital R, capital R, capital Y, capital Y, I will get 16 combinations. How many? I'll get 16 combinations, my dear children. And how is this 16 combination divided? What is the phenotypic ratio? It is 9:3:3:1. 9 of them are going to show the dominant parental character, that is what? Round and yellow. 1 of them is going to show the recessive parental character, that is what? Wrinkled and green. And over here, you will be finding new character, non-parental character. That is, if round was with yellow, round will be with green now. If wrinkled was with green until now, wrinkled will be with now yellow. You get it? So 9:3:3:1. This is the non-parental combination, and this is what I'm exactly trying to explain you guys. Why did you get non-parental character? Why there is new combination? Why there is new recombination according to Mendel? Because he said independent assortment is happening. Alleles of a pair of a gene independently separates from the alleles of the other pair of genes. So capital R does not say I will just go with capital Y. It is also going with small y. Why? Because independent assortment is happening. That is why recombination is there. So according to Mendel, because of independent assortment, recombination is happening out there. And always remember, in this type of thing, you may get question that almost these many offsprings were there. Out of them, how many will be showing non-parental character? So you should know how to do the calculation. Out of 16, 6 of them are doing. Out of 16, 6 of them are non-parental. Now, I've given the number to you. Suppose you are being asked, out of 60, how many of them are going to show the non-parental character? Then multiply it and you will get your answer of non-parental offsprings out there. You get it? You get it? Genotypic ratio, Pallavi, oh my God. Okay, so usually the phenotypic ratio and everything is asked. So Mendel, my dear children, worked according to this particular principle. That is, independent assortment leads to new combination. Now we will go to, I hope this question is clear. Now we will be going to, this came in model question paper this time, okay. We will be going to the question that we started off with, which was this. So, Mendel crosses recombination occurs due to independent assortment of genes on different chromosome. Now, according to Morgan, things are different. According to Morgan, recombination occurs due to crossing over between linked genes. That means new combination occurs when the crossing over happens. From here to here, the crossing over happens. That's when you get new combinations out there between the linked genes. Got it? So, this is it. You need your answers, write this, and you will get your 2 marks for this particular question. Now, the next question, differentiate between linkage and recombination. I already told you, linkage means what? Tendency of genes located close to each other on the same chromosome to be inherited together. Recombination means opposite of it. It is a process by which you find new combinations of gene, and it is happening because of crossing over. Linkage reduces the chances of new genes combination. Recombination increases the chance of recombinations out there. Got it? Simple question. Next. Identify the symbols used in pedigree analysis. This symbol is given. When a male and a female is joined with 2 lines, that means their association is very much strong. It is very much strong. It shows mating between relatives. It shows what? Mating between relatives. This is known as consanguineous mating. If at all male and female is affected and it is giving a male, this is what? A male being affected. That is another symbol that has been used over here. So consanguineous mating, please remember it. And one very important thing, please go through the pictures or pedigree chart given in your NCERT. One picture is of myotonic dystrophy, that is an autosomal dominant disorder. Another picture is of this particular thing. Another picture is of, what do you call it? Apart from myotonic dystrophy, you have one more thing. Sickle cell anemia, that is an autosomal recessive disorder. So one is dominant, another one is recessive. Please remember it. Always remember, how do you understand dominant and recessive disorders? If at all none of the parents are affected, as in basic, not none of the parents. If at all, suppose you see that over here, it's like this. Okay. If in every generation, if at all someone is being affected, it's a dominant disorder. Or if you see that in one generation, no one is affected, in the next generation, it is affected, it is a recessive disorder. It is a recessive disorder. So if at all it is showing in every generation, dominant disorder, if at all it is skipping generation, then it is recessive disorder. Okay. Now, parents with male child affected with the disease. You are talking about? Okay, I didn't know. So what is haplodiploid sex determination mechanism, and mention an animal which exhibits it. Haplodiploid sex determination mechanism is being shown by honeybees. Okay. Here, in the case of honeybees, the females will be always diploid in nature. That means they have 32 number of chromosomes. Whereas males which are there, they will be having half the number of chromosomes. That is haploid nature. So, now the females, since they are 32 number of chromosomes, they will undergo meiosis while forming gametes. And now, my dear children, in the gametes, you have only 16 chromosomes. Whereas in the males, they cannot undergo meiosis. They will just undergo mitosis. Okay, and now you have 16. Suppose this 16 and this 16 came together, you have got 32. and that's how a female is being formed. But any other egg which is there, which is not being fertilized, suppose fertilization is not being happening, and still you are getting something that is going to be a male. And this process is known as parthenogenesis. Not partheno, it is partheno. R is also there. Parthenogenesis. This is haplodiploid form of sex determination. Over here, if at all, if you see, it does not, male does not have a father. It does not have a father, but it has a grandfather. Why? Because the mother is formed when two things comes and fuse. So grandfather is there, father is not there. Okay. Hi Priyanka. Varalakshmi, ma'am, again, when will you come live? So I'm not going to come live within these 2 days, but yes, you will be having recorded videos. Okay. So I'm trying to complete everything important up here itself, whatever important chapters are there. The rest of the chapters you will get tomorrow morning itself. So you need not worry about anything. Okay. Now define point mutation. Give an example for point mutation. What do you mean by point mutation? If at all there is a change in the single base pair, you call this as point mutation. For example, in the case of sickle cell anemia, you had GAG, but instead of A, it is replaced by U, and now it is known as GUG. Instead of A, it is replaced by U, and now it is GUG. Here it was coding for glutamic acid, and now it is coding for what? It is coding for valine. And because of this substitution happening on the 6th codon, your hemoglobin structure is being done. Your hemoglobin structure will not be able to be proper. The quality issues will be coming up. If hemoglobin is not proper, obviously the cell carrying it will also be not that great. That is, your RBC will be sickle-shelled in shape. So this is the classic example of point mutation. Okay. Next question. What are the conclusions drawn by the T.H. Morgan from crossing experiments in Drosophila with respect to linkage? He said, the higher the linkage, if the linked genes, if they are very close to each other, there will be lesser recombination. Okay, and if lesser is the linkage, that means they are very far from each other, there will be higher recombination out there. Okay, that is what he said. And he gave the things also. For example, he spoke about two character, yellow body and white eye. Yellow body and white eye genes are very close to each other. They will be very close to each other, which is why the rate of recombination is very less, only 1.3%. The maximum recombination percentage is 50. Out of that, only 1.3% has been shown by yellow body and white eye gene. Why? Because they are very close to each other. Okay? If at all you see the percentage of recombination more, that means they are far away from each other. Now, why did Thomas Hunt Morgan take fruit flies for his experiment? What was the reason why he took fruit flies for the experiment? Give any 3 reasons. So he took fruit flies for his experiment because it can be grown on simple media. They complete their life cycle very, very quick, so he can see anything that comes up out there. A single mating can produce large number of offsprings, and there is a clear differentiation of what? Sexes. That is the reason that he took the fruit flies out there. Now, what are the chromosomal disorders? Give an example. What are the chromosomal disorders which are there? The disorders in human beings are caused due to absence. Okay, so chromosomal disorders basically means over here, suppose there is loss of a chromosome, it leads to chromosomal disorder. There is grey mass of chromosome, it leads to chromosomal disorder. Or basically the structure of the chromosome is being disoriented, that is also leading to chromosomal disorders. Example, Klinefelter syndrome, Turner syndrome, Down syndrome, all of them. Okay? Fine. Now, going ahead. Question number 8. Give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individuals. A simple cut is going to result in non-stop bleeding. This is what disease? Chalo, you guys guess. Come on, come on, come on. So until now I was talking. Obviously I will also get tired. Now you talk for some time. A simple cut leads to non-stop bleeding. Come on, come on, come on. Hemophilia, exactly. It would be what? Hemophilia. Hemophilia, in hemophilia, my dear children, a protein which is responsible for clotting is being affected. So this is hemophilia. Ayo. give reasons. Okay, so why is this happening? Because, my dear children, a protein which is responsible for floating, that is being affected. That is why you get this thing. Now, Turner syndrome affected females are usually sterile. Why is that so? Because in Turner syndrome, instead of having XS chromosome, the person has XO chromosome. There is one loss of one particular chromosome, my dear children, which is why the female will be having rudimentary ovary. Rudimentary ovary means obviously they will not be able to process well, and they will not be able to process to produce what? The gametes out there. Okay, so that is why they are usually sterile in nature. Now, in Morgan's dihybrid cross experiment on Drosophila showed that flies having genes for yellow body and white eyes exhibited lesser recombination. If less recombination is there, then this is because of what? Because the genes are close to each other. They will move together only. That is why less recombination. They are tightly linked genes. Now, inheritance of skin color in humans show different phenotypes. Why inheritance of skin color shows different phenotypes, my dear children? Because it is coded by multiple genes. So, usually we have studied that one gene codes for one protein, right? Or basically one gene is responsible for one character. That is what we have studied until now. But please understand, it does not work like that. You have some exceptions. What are the exceptions? So, in the case of polygyny, multiple genes are responsible for one particular character. Multiple genes for one particular character. Suppose over here I say there is capital A, capital A, capital B, capital B, capital C, capital C. Now over here the genotype is capital A, capital A, capital B, capital B, capital C, capital C. All these genes, when they are active, it makes the skin color very dark. It makes the skin color very, very dark. Okay, so we have multiple genes are being affected. Now, if at all you want to, if at all you're asked, which one gives the lightest skin color, how do you give the answer? My dear children, if at all the alleles are recessive in nature, for example, you have AA, BB, CC, this will be the lightest skin color. In between that, you have capital A, small a, capital B, small b, capital C, small c. This also have lighter skin color. But this has the lightest skin color. Over here, always remember, multiple genes are responsible for one character, which is why this is known as polygenic inheritance. This is polygenic inheritance. Okay. Now, the next thing. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think it is getting accumulated? Because, my dear children, one very important protein or one very important enzyme is not being produced, and that is phenylalanine hydroxylase. Phenylalanine hydroxylase is not being produced, which is why the conversion of phenylalanine to tyrosine does not take place. Which is why phenylalanine is going to be accumulated in our body in the form of phenyl pyruvic acid, and this will cause mental retardation when it gets accumulated. Also, it is not even reabsorbed by body, so which is why it is released through our urine and it gives a very foul smell. Okay, so this is an autosomal recessive disorder. Please remember that. This is also an example of pleiotropy. Pleiotropy means where one gene shows multiple effects. So over here, multiple genes coding for one character, that is polygyny. Over here, one gene affects multiple character. So over here, obviously this thing is not happening, which is why PPA is being accumulated, that is phenyl pyruvic acid. Apart from that, melatonin is also not being produced. Why? Because tyrosine is super important for formation of, not melatonin, melanin. Okay, for production of melanin. And if melanin is not there, you will have reduced pigmentation of your skin, your hair, everything out there. Okay, so two characters is being affected. That is why this is an example of pleiotropy as well. Pleiotropy as well. Okay, so please write the answers like this. It is there in the PDF. You can get it if at all you take up. You join the WhatsApp channel, you will get the PDF up there. Cool. Works. Now, going ahead, few autosome linked recessive gene blood diseases occur in human population. Among them, some are related to quantitative and some are related to qualitative things. So explain that. How do you explain that? It is very, very, very simple. Remember, so over here I have two things. This is chromosome number 11. This is chromosome number 16. Chromosome number 11 has the HBB gene. Okay. Because of some mutations in the HBB gene over here, my dear children, it is not able to produce the actual beta hemoglobin. So beta, there are two chains to form a hemoglobin, alpha chain and beta chain. So beta hemoglobin chain is not being produced really nicely over here, which is why obviously the hemoglobin which is going to be formed will be having bad quality. And now if this is there in a cell, the red blood cell becomes what? Sickle-shelled in shape. Okay, this is the case of sickle cell anemia. How does this mutation happen? Why does the beta globin chains quality get bad? Because, my dear children, on the 6th codon position, instead of having GAG, it has GUG. A is replaced by U, which is why the amino acid which was there, it was glutamic acid. It has now been replaced by valine, and we do not want this valine. And that is a problem of the whole thing. Okay, remember A comes before U, so it was A1, then U. It was G first and then V. Okay, G comes before V. Remember like that. Okay, so this is an example of qualitative disorder, qualitative blood disorder, because the quality of beta globin chain is being bad. Now over here, if at all you talk about thalassemia, which is a quantitative disorder, you need to understand that HBB has two genes, right? Suppose this is not working well. So that means the amount of HBB is going to get reduced. The amount which is there, it is going to get reduced, which is why we will be having the disease. Here the quality is not being reduced, the amount is being reduced, which is why this will be a quantitative disorder. Okay, now on the other side, if at all you talk about alpha, okay, in the case of alpha chain, you have 4 genes coding for it, 4 genes. Either this can be affected, in that case little amount of alpha globin chain will be reduced. If both are affected, little more will be reduced. If all of them are affected, all of them will be reduced. So it's a qualitative, it's what? A quantitative disorder, whereas over here in the case of sickle cell anemia, it's a qualitative disorder. Okay. In sickle cell anemia, only beta globin chain's quality is affected. In the case of thalassemia, alpha and beta both can be affected. Alpha is there on 16th chromosome and beta on the 11th chromosome. Both of them are autosomal recessive disorder. Both of them are blood disorders. Remember that. One more blood disorder is there in your NCERT, and that is your hemophilia. But that is not what autosomal linked. It is sex linked blood disorder. Got it? I hope you got it. Now, so this is the answer. Please go through it. It is going to definitely help you out. Going ahead, define aneuploidy. Give 2 examples of aneuploidy. Aneuploidy means you're losing a chromosome or gaining a chromosome. That is aneuploidy. Give 2 examples of aneuploidy. You have Turner syndrome. You have Down syndrome. Everything is an example of aneuploidy out there. So I'll talk about each of them. Listen to me very, very carefully. One is Down, one is Turner, and one is Klinefelter's. Here you have also been asked about the karyotype of each of them. So in Down syndrome, the 21st chromosome, listen, in our body we have how many chromosomes? We have almost 46 chromosomes. If we talk in pairs, there are 23 pairs. There are 23 pairs. The 23rd pair which is there, it is sex chromosome. So apart from that, 22 of them, or basically 44 of the chromosomes are autosomes. They are what? Autosomes. They are the normal chromosomes out there. Only 2 are sex chromosomes. In the case of Down syndrome, the 21st pair, okay, in the 21st pair, you should be having something like this, but you have an extra chromosome out there, which is why you say it is trisomy. Trisomy of 21st chromosome. Trisomy of 21st chromosome. How do you express it in the term of karyotype? So you say, instead of usually you have 44 autosomes plus XY, or 44 autosomes plus XX depending on the sex. Over here you have 1 extra autosome, so I'll write 45 autosomes plus XY. So that is a Down syndrome patient who is a male. This is 45A plus XX, that is a Down syndrome patient who is what? A female. Got it? In Turner syndrome, things are going to be kind of different. In Turner syndrome, you have monosomy X chromosome. 1 X chromosome is missing out there. So this is XO condition. 1 is missing. This is monosomy. And over here, what will be the, karyotype? You will be having 44 autosomes, but XO condition is there. Or you can express and write it as, 45 XO. That means in total, instead of having 46, you just have 45. Okay, that is also possible. Yes, symptoms. Please go through all the symptoms. They are super, super important. Pallavi, kudos. Pallavi is someone I'm seeing from the starting of the video. Awesome. Next, Klinefelter syndrome is where you have an additional X chromosome. So XXY condition is happening up over here. In this condition, one extra X chromosome is there, which is why we will write 45 autosomes plus XXY, or we can write 47 XXY. That means one extra chromosome is there. Okay. Please go through the symptoms of each of them. They are all super important. In Klinefelter's, you have masculinization, right? It's a masculine, overall masculine development is there, but you have gynecomastia, that is development of breast in the case of Klinefelter syndrome, and spermatogenesis is not going to happen. In Turner syndrome, the females are going to be sterile. Okay, they are also going to have certain masculine features are there, but overall masculine development is here itself. Don't get confused. Okay, and Down syndrome is basically your broad palm increase, mental retardation is there, a lot of other functions are there. Please go through them. Cool? Yes, ma'am. Okay, now represent schematically the results of one gene inheritance. This is something that you probably know, so I'm not going ahead. Just throw capital T here, small t, you will get all things as capital T, small t. You do the selfing, you get two things, two ratios. One is phenotypic and one is genotypic ratio. According to the phenotypic ratio, my dear children, it will be 3:1. 3 of them are going to be tall, right, like that, and 1 of them is going to be dwarf. That is the phenotypic ratio. Genotypic ratio is going to be 1:2:1. 1 basically stands for capital T, capital T. 2 of them are going to be capital T, small t, and 1 of them is going to be small t, small t. Got it? Okay, so please remember the ratio and make the entire thing. This came in model question paper 3. Solve it up. Okay. Next thing. Okay, categorize the following as either autosomal or sex-linked disorders. Hemophilia. Hemophilia is autosomal disorder or sex-linked disorder? What do you think so? Hemophilia, which is out there, it is autosomal disorder or is it sex-linked disorder? Tell me, tell me, tell me. Hemophilia is sex-linked recessive disorder. Thalassemia and phenylketonuria are autosomal recessive disorder. Myotonic dystrophy, the pedigree chart is given. Please go through it. And it is, my dear children, okay, it is, my dear children, what disorder? It is autosomal dominant disorder. The only dominant disorder that is given in your NCERT is this. And that is in the form of pedigree chart. Okay, so please go through it. Allosomal. Rashi, hi. Yes, it is allosomal. Hemophilia is an allosomal disease. That is sex-linked. Sex chromosomes are also known as allosomes. Okay. Now, what conclusion will you arrive after careful observation of the given diagram? That is given diagram. Valine is there. So here it is GUG. Here instead of GAG, you have GUG, and here you have valine. That means substitution has taken place. Point mutation has taken place over here, and that is why the person has sickle cell anemia. Okay. Cool. So that's a very cute comment that I got. I do not know how to pronounce it. Rennie Eager. We the students want to extend our heartfelt gratitude to you, ma'am, for taking out time to come live on YouTube and revise everything with us from the bottom of our hearts. Thank you. That is the sweetest message that I saw today. Thank you so much. It's all for you guys. Do well in your exams. That's all. Okay? Now, identify the disorder by the following symptoms. Individuals with overall muscular development but with gynecomastia. I told you overall muscular development, that means you're talking about Klinefelter syndrome. And with gynecomastia, that means development of breast. So obviously, this is going to be what? This is going to be what? It is Klinefelter syndrome. Klinefelter syndrome. Okay? Got it? Yes. So here I have written everything. You can go through. Explain thalassemia as an example of Mendelian disorder. We already studied about thalassemia. just talk about that it is autosomal linked disorder, what exactly happens in alpha thalassemia, the alpha genes are being affected, in beta thalassemia, beta genes are being affected. It's a quantitative blood disorder. Mention about that. Okay? Cool? Yes. Now schematically represent the incomplete dominance by taking inheritance of flower color in Snapdragon as an example. So how do you show incomplete dominance, my dear children? In Snapdragon, it's super simple. It is what? Super simple. Listen to me carefully, okay? Okay. So you have capitlar. This is considered to be red flower, and you have white flowered plant, that is RR, smaller. When you fuse them, what do you expect? All of them are going to be capillar, so you expect that capillar will be dominant. But you know what? It is not dominant. It is incompletely dominant, which is why it gives pink color. So this is actually something that moves away from law of dominance, right? And to prove that, one more time, selfing was being done. While selfing is done, capillar smaller is taken, and now you self it. You get capillar, you get capillar smaller, you get capillar smaller, and you get smaller. What do you see? One is capillar, two of them are capillar smaller, and one is smaller. You expect this also to be red in color, but you know what? These are pink. This is the only one which is red. Here you have a small allele, that is why recessive allele, that is why it is pink in color. It is incompletely dominant. And here it will be white. So what is the phenotypic ratio? 1 red, 2 pink, 1 white. That is 1:2:1. What is the genotypic ratio? Capillar 1, capillar smaller 2, and 1 smaller. So that is also going to be 1:2:1. So in this particular case, okay, always remember the phenotypic and genotypic ratio is going to be the same, and the dominant one is not going to be completely dominant over the recessive one. Mansi says incomplete dominance, the dominant gene is not completely dominant over the recessive, hence intermediate product is obtained. Exactly. Pallavi, thank you, dear. Thank you to you as well. So that's so cute of you when you guys give me energy. Okay, so we are here, we will do it. Okay, hi ma'am, I am present. Hi Sagar. Okay, so you have question number 15. Sex-linked recessive blood disorder occurring in about 8% of males and only about 0.4% of females, leading to defect in the cone of the eye. Over here, the first thing that you have to keep a notice on is that males are affected more than females, right? And it is going to cause defect in the cone of the eye. What is this particular disorder? This particular disorder is going to be what? It is going to be color blindness. Always remember, in color blindness, since it is a recessive disorder, suppose in the case of females, one is affected, the other chromosome is still there to show up this, which is why she will be just a carrier, she will not have the disease. But in the case of males, if at all one is affected, it will still show the character. That is why males are being affected more. Okay? Males are being affected more. So over here, your answer for this question is what? Color blindness. Next thing, sex-linked recessive disorder affecting a single protein involved in the clotting of blood. In clotting of blood, my dear children, if clotting of blood is affected, what is this particular disease? Can you tell me? It is going to be hemophilia. It is going to be hemophilia. So over here, the answer for this question is going to be hemophilia. Great. Got it? Now next. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. It's an inborn error of metabolism. It's there from the birth, and it is an autosomal recessive trait, and it leads to accumulation of phenylalanine. What is this? What is this? It is going to be phenylketonuria. Okay, it is going to be phenylketonuria. Accumulation of phenylalanine means phenylketonuria, right? Next thing, autosomal ringed recessive blood disorder, substitution of amino acids in the beta globin chain of hemoglobin. If beta globin chain of hemoglobin is having a problem and substitution is also happening, that has to be sickle cell anemia. So what Prachi gave me sickle cell anemia for another option, I think so, or are you ahead? Not know. Anyway, the answer for this is what? Sickle cell anemia. Now, autosome linked recessive blood disease, reduced rate of synthesis of globin chains of hemoglobin. What do you think is the answer for this question? Reduced rate is there. Okay, reduced rate of synthesis of hemoglobin is there. Quantitative problem, and that would be what? It is going to be thalassemia. Fine. Slim Shade, hi ma'am, you are awesome. Thank you so much, Slim Shade. Okay. Got it, children? Okay. I think that's the last question for principles of inheritance and variation. Let's keep it up. Okay. Okay. Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous, heterozygous. Pedigree symbols are used to analyze these disorders. Now, this is an MQP question, and this is a very important and a very tricky question. Okay, so you need to understand it. So just listen to me very carefully. Okay, are we ready? And before that, can we make it 100 this time? 100 likes, and all of you who are watching this video, I hope you have subscribed to the channel as well. So I'm going ahead. Autosomal dominant disorder. It's an autosomal dominant disorder, and over here you have a square that is being colored. That means this person is affected. This is a male who is being affected. Well, autosomal dominant disorder, and it to be affected, obviously it should be having something like this. Capital A allele has to be there. So either it can be in this condition or it can be in this condition, this or this. One capital A or one dominant allele has to be there, which is why your answer for this question would be this or this, as mentioned over here. Now, it's an autosomal recessive disorder and the female is not being affected. If it is an autosomal recessive disorder, it will only show up if the condition is like this. Recessive ones will be there, right? Now over here, this particular person does not have this disease. Even though we are talking about autosomal recessive disorder, this person does not have this. And if this person should not have it, we should be adding it with one capital alleles. One capital allele out there. So it would be what? Capital A, small a again. Or it can be capital A as well. Got it? Okay, the next thing, sex-linked dominant disorder. Now, sex-linked dominant and this person is not affected. If this person has to be affected in this particular case, then my dear children, it is male first of all, and he should be having the capital allele, that is X capital A, to be affected. But here he is not affected. That means you have to put the recessive allele, that is small a. That will be what? Small a. Okay, now next one. Sex-linked recessive. Sex-linked recessive, and both of them are affected. So both of them are affected and sex-linked recessive. So in this particular case, it should be like this in the case of males because there is a recessive allele, and in the case of females, both of them should be small a, small a. Got it? And here we complete your principles of inheritance and variation. Please go to the pedigree chart of myotonic dystrophy. That is very, very important. Chetan says that, hi ma'am, you are a gem. Thank you so much. You are also a gem. Okay, so shall we go ahead? So I will give you two options. One is human reproduction and one is evolution. Which is a chapter that you want me to complete as of now? So I have got less energy. Okay, my energy level is going toop. But I can complete one chapter. Which is the chapter that you want me to complete? Human reproduction, or is it the other one? Choose. Human reproduction, or will it be evolution? So do not worry if I am not able to cover the other chapter. You have the Brahmastra series. I will put down the link in the description section for the chapter I am not able to complete. Okay, there you can just go through the series. It has the same questions. Okay, I have explained it really well out there. So do not be worried. You can cover it. It's not like you cannot. But I am just telling you, giving an option. Human reproduction is what I found in more amount. Okay, human reproduction. So let's go with human reproduction then. So evolutions, I will definitely give you the link for Brahmastra series. You can please go ahead and start solving question. I think evolution and reproductive health, both chapters are left. Please go through that. So my energy level is drooping down, so I will definitely not be able to cover it up. So you have reproduction. Where's the chapter reproduction? Let me check it out. Okay. It starts from here. Okay, let's solve. 41 people are watching. Okay. Now, secondary oocyte formation from primary oocyte occurs in which of the follicle? My dear children, I will be a bit fast, okay? Secondary oocyte formation from primary oocyte occurs in which follicle? Secondary oocyte formation from primary oocyte. So that means primary oocyte has underwent division and now it has formed the secondary oocyte right over there. And it is also going to form the first polar body. Where does this division take place? Where does this division take place? 1C. Mansi says the answer for this question is C. Tertiary follicle. Is it tertiary follicle? So you have primary follicle, you have secondary follicle. Both of this place will be having primary oocyte within it. When tertiary follicle is formed, my dear children, that's when the primary oocyte within the tertiary follicle is going to divide and going to give secondary oocyte out there. So your answer for this particular question would be C. Your answer for this particular question would be C. Okay. It is not B, it is not D, it is C, tertiary follicle. Remember, over here you have the primary follicle where just few granular cells are surrounding the primary oocyte. Okay. Now you have got few more granular cells and you also get a theca layer. This is your secondary follicle, but inside that still you have the primary oocyte. Now, this theca layer differentiates into inner theca interna and outer theca externa, and you also have an antral cavity out over here. And finally, the primary oocyte which is there, it is now going to divide into what? A secondary oocyte and the first polar body. So this is happening within the tertiary follicle. Always remember that. Now, the tertiary follicle which is there, okay, tertiary follicle which is there, it will be going ahead. Tertiary follicle which is there, it is going to develop into Graafian follicle as well. Okay. And always remember, in the Graafian follicle, you have the secondary oocyte. You do not form the ovum. The next division also is on a pause. It has not been completed. It's not there. Okay. So your answer for this question is C. Now, second question. The cells that secrete testicular hormones, androgens, are the cells that secrete testicular hormones. But your sir told A is correct. No. A cannot be correct here. Okay. The cells that secrete testicular hormones or androgens are what? So always remember, inside the seminiferous tubules that you have the Sertoli cells and the spermatogonia. These Sertoli cells are going to act on the spermatogonia and help in the production of sperms. Right? Now, in between two seminiferous tubules, you have some Leydig cells as well. Leydig cells as well. What do? They release testosterone. And they are very, very important. Okay, so here you are asked, the cells that secretes the testicular hormones or the male sex hormones will be what? It will be Leydig cells. B is your answer for this particular question. Okay. Next question. Which of the following statement is true? LH surge induces menstruation. Does LH surge lead to menstruation? What do you think? Now, you might not be knowing this particular question. If at all you do not know menstruation, which is why we will come over here, solve menstruation, and then we will go back there. So listen to me very, very carefully. Okay. At this particular point of time, you have a cycle of 1 to 29 days or 1 to 28 days. The first 5 days, which is known as menses, that is when the endometrium is going to shut down. Why is endometrium shutting down? Because two very important ovarian hormones are very less in amount, that is estrogen and progesterone. Their level goes down, and that's when endometrium is going to shut down, and that's when you have the menstrual phase. At this particular point of time, under the influence of pituitary hormones, that is FSH and LH, the follicles which are there, they are going to start developing because we need ovulation to happen, the next oocyte to be released. That is why this particular thing is happening. So we have officially entered the next phase, that is known as follicular phase, where the follicles are developing. And while the follicles are developing, you should also know the follicles are going to release some estrogen out there. The estrogen level goes up, that means the endometrium is soon going to be regenerated. It is going to be regenerated, which is why it is known as proliferative phase also. It is proliferating, follicular phase is also happening, both of them together. Now, when the level of estrogen goes really high, it leads to LH surge. It will be leading to what? LH surge. Now when LH level goes high, that's when the follicles are going to rupture and you will be having ovulation. When does this happen? In the middle of the cycle, that is almost on the 14th day. Okay, almost on the 14th day, this is particularly going to happen. Okay, and my dear children, listen to me carefully. Once ovulation has taken place, whatever is left behind is known as corpus luteum, and this corpus luteum is actually maintained by the LH and the FSH. This corpus luteum is now going to release two hormones, one is progesterone and one is estrogen, but majorly progesterone. And when the level of progesterone goes up high, my dear children, the endometrium becomes very much thickened. Becomes what? Very much thickened. And over here, the estrogen also goes up little much. Estrogen also goes up little much. But there is one problem. When the level of progesterone increases, it does not want FSH and LH to increase. It tells that, hey, you do not increase. And when FSH and LH is not there, do you think corpus luteum will be maintained? No. And if corpus luteum is not there, do you think progesterone will be there? No. So if at all, fertilization has not taken place. Because if fertilization has taken place, there will be someone else to take care of the corpus luteum. If at all fertilization has not taken place, my dear children, what will happen? This whole thing will happen. The corpus luteum is going to disintegrate. If corpus luteum goes off, my dear children, nothing of this persists. It will also go down. And that is exactly why you have the next menstrual cycle taking up there. This phase, the last phase that you have, where the endometrium is very thick, it is going to secrete things. It is known as secretory phase. It is also known as luteal phase. Okay, it is also known as luteal phase. Fine. Now, over here, if at all I go back to the question that we were solving, that is, which of the following statement is true? LH surge induces menstruation, does it? No, it induces ovulation, not menstruation. The second thing, regression of corpus luteum increases progesterone. Regression of corpus luteum, do you think it leads to increase in progesterone? No. Regression means it is getting broken down. That means progesterone will also get broken down. Wrong. Diploid egg is formed after second meiotic division. Array, egg is also always going to be haploid. Can it be diploid any time? No, my dear children. So this is also wrong. Now, embryo differentiate from the inner cell mass of the blastocyst. This is correct. You have different type of embryos out there. The embryo which gets implanted, is known as blastocyst, which has two layers. One is trophoblast layer and the other layer is inner cell mass. The other layer that you are having is inner cell mass. The inner cell mass is going to form the future embryo. The trophoblast is going to attach to the endometrium and going to help in the formation of placenta. Okay, so in that particular case, what is your answer for this particular question? The best boy, Sagar and Rashi, the answer for this question is what? D. Okay? Yes. Now, scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. Scrotum temperature will be how much degree lower? It is 2 to 2.5 degrees Celsius lower. So I have got two options, either this or this. Let's see. Now, each of the testis has about B compartments. How many compartments are there? 250 compartments are there. And, okay, so again 250, 250. Now, it measures C centimeter in length. How many centimeter in length, my dear children? How many centimeter in length? 4 to 5. 4 to 5. So this is your answer for the question. Always remember, testis which is there, the width is 2 to 3 centimeter and the length is 4 to 5. The length will be 4 to 5. So your answer for this particular question is going to be B. Who said D? Baba, it's not D. It's going to be B. Why B? Listen, you're asked about the length, not the width. The length is 4 to 5. The length is 4 to 5. Don't get it wrong. Small information, if at all we get it wrong, it will be a big trouble later on. Okay? Now, in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? If FSH is not produced, what stage is going to be affected? This is a question number 5. Try it out. Try it out. Mansi, you gave me the correct answer here. Rashmi also. And Prashi also. Try this particular question out. If at all FSH is not secreted by the anterior pituitary, which stage is going to be affected? Listen, you have two hormones, LH and FSH. LH is going to act on what? The Leydig cells. And the Leydig cells are going to produce what? The androgens, that is the male sex hormones. It is going to get inside the seminiferous tubules, where FSH are going to act on the Sertoli cells. And when they act on Sertoli cells are going to release some factors, okay, some very important factors. These factors are very important such that in the testis, the level of androgens are maintained. If the level of androgens is not maintained, spermatogenesis is not going to happen properly. And in spermatogenesis, one major process, that is conversion of immature spermatids, the kids, to mature form of sperm, that is spermatozoa. That is not going to take place. So this is the thing. Formation of spermatozoa is going to be affected. Your answer for this particular question is going to be B. Always remember. And what do you call this as? You call this as spermiogenesis. You call this as spermiogenesis. So your answer for fifth question is B. Not C, it is B. Why B? Spermatids to spermatozoa conversion is being affected. So formation of spermatozoa is being affected. Cool? Yes. Next, to produce 2000 sperm and 400 ova. To produce 2000 sperm and 400 ova. How many spermatogonia and oogonia? How many spermatogonia and oogonia are required? What do you think is the answer for this particular question? If 2000 sperm, listen, one spermatogonia which is there, it gives rise to 4 sperms. Here, how many of them are there? 2000. So 2000 divided by 4, how many? 500 will be there. So each 500 of them, if they give 4 sperms, that's how we get 2000 sperms. So 500 spermatogonia. But what about ova? What about ova, my dear children? One ova, which is oogonia, which is there. Oogonia, when it undergoes oogenesis, it gives only one ovum. So if I am saying 400 ovum is there, how many oogonia will be there? That will also be 400. So your answer for this question becomes what? B. It becomes B. Okay, yes. This is a question that all of you got correct. Awesome. Lakshmi, Bhuvan, everyone. Shivakumar, Vikas. Awesome. Okay, next question. Which among the following has 23 chromosomes? Which among the following will be having 23 chromosomes? Spermatogonia, zygote, secondary oocyte or oogonia. Half of the number of chromosomes. Spermatogonia will be having diploid. Only when it undergoes meiosis and forms secondary spermatocyte, that's when it will be having haploid. Zygote is always diploid. Secondary oocyte, yes. Everything that has secondary term will be having haploid number of things. So secondary oocyte, secondary spermatocyte, both of them will be having haploid, that is 23 number of chromosomes. Seventh answer is what? Seventh answer is C. Children, you're getting your answers correct, right? Mansi, Harshita, Shivakumar, everyone, Shweta, all of you are doing awesome, okay? So since I do not have much energy out there, I'm not calling out your names. But yeah, you guys are doing good. Now, identify the wrong pair with respect to menstrual cycle. What is the wrong pair? Formation of corpus luteum 15 to 28 day. Formation of corpus luteum is from 15 to 28 day. Do you think so? Guess. Yeah, it is there, right? It is happening up there. After ovulation, it happens. So this is correct. Secretion of progesterone 15 to 28 day. When corpus luteum is there, that's when secretion of progesterone will be there. That is also correct. Regeneration of endometrium from 5th to 13th day. That is also correct. Regeneration of after menses for 5 days, regeneration is going to happen. This is also correct. What is not correct? Disintegration of endometrium 5th to 13th day. Disintegration happens from 1st to 5th day. It happens from 1st to 5th day. So your answer for this question is going to be D. Who said B? Adigitum. Who said B? Adigitum. Adigitum means, in Malayalam, adigitum means adigitum. How do you say? Okay, so it is D. Okay, Soumya. Yes, Vikas. Yes, Ananda. Hi, Varsha. Pallavi. Okay, fine. Next question. The secondary oocyte after ovulation is covered by a non-cellular layer. Not after ovulation. Basically, when it is in the Graafian follicle, the secondary oocyte forms its own layer. Remember, secondary oocyte will form its own layer. And it is non-cellular. It is not made up of any cell. It is known as zona pellucida. It is known as zona pellucida. This non-cellular thing, this is an important thing. This is zona pellucida. Now you are saying D. Arey Baba, we are solving question number 9. What is the answer for question number 9? It will be C. Okay, done? Clear? I hope it's clear. Next, during pregnancy, the fetus develops limbs and digits. Limbs and digits. Now, this is one of my favorite topics out there. Question number 10. Okay, question number 10. Everyone solve. Let me see who all gives the answer correctly. Ayan says C. Ayan says C. After 6 weeks. After 6 weeks. After 6 weeks. That means... Listen to me carefully, okay? The first month, baby will be having heart formation. So when we die, we check the heartbeat. Similarly, when we are alive, the first thing that we check is what heart is beating or not. So first month, you have heartbeat. The second month, that is there. Second month, you will be having limbs, that is your limbs, and you will be having digits. Limbs and digits. Okay, that is second month. Second month means in 1 month, how many weeks are there? 4 weeks. 2 months means 2 into 4, 8 weeks. So your answer for this question will be what? After 8 weeks. After 8 weeks. But let's go ahead, let's talk more. So basically, after 3 months, what is going to happen? Most of your organ system and organs are going to form. Your external genital organ, that is going to define whether you are a male or female, that is also going to be formed after 3 months. 4th month is break. 5th month, the baby says, God, I want to move around. I need to have some hair. So you have the first movement, FF, remember, 5th month, first movement, and also first hair on the head of the baby in the 5th month. Okay, and now 6th month, God says you wanted hair, no, have hair on your body also. So the babies will be having small baby hairs on also eyelashes, and the eyelids will be separated. So all these things happen in the 6 months. Okay, now over here you are asked about 8 weeks. So yes, this is the data. Please remember week by week data also. You know month by month, month into 4 is equals to week. Okay, okay. Now, statement 1. The process of release of sperms from the seminiferous tubule is known as spermatogenesis. Arey Baba. Arey Baba. The process of release of sperms from the seminiferous tubules is known as slim shade. I just have some 5 marker questions and then I will be stopping it. Okay? So, please bear with me. Okay, the process of release of sperm from the seminiferous tubules is not known as spermatogenesis. It is known as spermiation. Aviation, spermiation, not spermatogenesis. Wrong statement. The next one. The spermatids are transformed into spermatozoa by the process called spermiogenesis. Spermatids to spermatozoa by spermiogenesis. Yes. Kids to zoa, right? Spermiogenesis, this is correct. So, statement 1 is false, statement 2 is correct. Statement 1 is false and statement 2 is correct. D is your answer for this particular question. Okay, so 11D, who gave me the answer? Mansi gave me the answer. Bhuvan also gave me the answer. Great, 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 great. Next, choose the correct option from the table given below for the formation and dissolution of the labeled part. What is this part? This is pellucida, zona pellucida. When is it? Who forms it? Secondary oocyte forms it. Who forms it? Secondary oocyte forms it. Who dissolves it? Sorry, when does it get dissolved? After fertilization. Okay, after fertilization and everything happens, my dear children, it is going to be dissolved. So your answer for this question becomes D. Okay, it also becomes D. 12th answer is D. Now we are going ahead with the 3 marker and 5 marker questions of human reproduction, and with that we will be completing our live. I'll be giving you the links for 2 Brahmastra videos. Please watch it. One is for evolution and second is for reproductive health, which I am not able to complete today. I've completed everything else, so please bear with me. I hope it's okay, right? Okay, what is menstrual cycle? We already discussed it. Menstrual cycle is the monthly cyclic changes that happens in the ovary and the uterus that occurs in the human female for about 28 days. What are the functions of the pituitary hormones? 2 hormones are there, FSH and LH. FSH stimulate the growth and maturation of ovarian follicles, and these follicles produces estrogen. That is important. Estrogen, what does it do? It causes repair and proliferation of endometrium and also causes LH surge that leads to ovulation. Cool? Yes, ma'am. Now, what is placenta leads its function? So, I told you the trophoblast layer of the blastocyst, the embryo, okay, one type of embryo that is known as, one stage of embryo that is blastocyst. It has trophoblast layer and inner cell mass. You know, this trophoblast layer which is there, it comes and implants into the endometrium, and you have the inner cell mass. Inner cell mass. The trophoblast basically forms structures like this. This is known as chorionic villi. Together the maternal tissue as well as the coronal villi forms the placenta, and the inner cell mass will be forming the actual embryo. This placenta is going to be a temporary vascular connection between the mother as well as the fetus. It will help in nutrition because the transfer of nutrients happen, respiration because oxygen is being supplied and removal of carbon dioxide takes place, excretion because removal of metabolic waste also happens, and endocrine function because it is very much important. There are four very important hormones which are being produced, and that is HCG, human chorionic gonadotropin, HPL, human placental lactogen, progesterone and estrogen. HCG, HPL, estrogen and progesterone, these are the very important hormones which are being released, so please go through it. Ananda, part 2 will be released tomorrow morning, please stay tuned for that. Okay, and it is going to be a recorded format, so please watch that video, it is going to be definitely helping you out. Now name the pituitary and the testicular hormones involved in spermatogenesis. Same, FSH is basically, it acts on Sertoli cells as I told you, and it stimulates spermatogenesis, and LH is there, basically it is secreted by anterior pituitary, it acts on Leydig cells to produce androgens out there. We studied both of them, right? So I'm not going up much. And parturition is induced by complex neuroendocrine mechanism. So over here comes fetal ejection reflex. Someone was asking about that. So listen to me carefully. In fetal ejection reflex, basically what happens is that, suppose this is the uterus that is there, fetus is growing up over here. Fetus, when it tries to move out, there are some receptors that becomes active. This receptors gives nervous signal to the mother's maternal pituitary gland. Okay, posterior pituitary. And now this signals, nervous system signals transforms into what? Hormonal signals, and that is oxytocin is being released. So basically it's the baby along with the placenta, because placenta is also going to release some hormones. Both of them together are going to trigger the expulsion of the baby, and that is known as the fetal ejection reflex. Placenta and the fully grown fetus, both of them. And now the receptors are active, it gives oxytocin is being produced. The oxytocin which is there, it comes back and acts on the myometrium. On the myometrium there will be certain receptors for oxytocin. When it comes and binds, it will cause contraction, relaxation, and the baby will be moving out. Okay, and the baby is definitely going to move out. This is how things are going to work. Now, label diagram of female reproductive system. Make sure you go to the entire diagram really well. This is super important. Then you have mammary glands also. Please go through it. You have a formula that you can remember. MAT. Dal fell on mat. M stands for mammary lobes. A stands for alveoli. Alveoli opens into tubules. Tubules opens into duct. Duct opens into ampulla. And ampulla opens into lactiferous ducts. Lactiferous ducts. This is how things goes on. Thank you, ma'am. This 2 hours class is very useful for my preparatory. You're most welcome. Okay. Now going ahead, represent the spermatogenesis. Again, over here is spermatogonia. It undergoes differentiation, just mitosis, to form primary spermatocyte. From here, the meiotic division starts off. From here, you have secondary spermatocyte. They will be haploid. Secondary spermatocyte again divides to form spermatids, and then spermatids can, they transform into spermatozoa. Cool. This is known as spermiogenesis, and the process of removal of sperms is known as spermiation. Here you have the sectional view of the male reproductive system. Please go through it. This picture is also important. Now, there are a few important other topics, that is this. And apart from that, the role of accessory glands out there in the case of males. So one is seminal vesicle, one is prostrate, and one is bulbourethral. Listen to me. Seminal vesicle is paired, bulbourethral is also paired. Okay, seminal vesicles secrete fructose, which is very much important for giving energy to the sperm. Bulbourethral provides lubrication to the penis. Now, the unpaired gland is prostrate, and it releases two very important things, calcium and the enzymes. Together, their secretion is known as seminal plasma. All of their secretion together, you call it as seminal plasma. That is super important. Okay, so seminal plasma is rich in fructose, calcium, as well as the enzymes. Fine. So with that, I complete today's live. make sure you go ahead with the what do you call, hormones found in gestation. So during pregnancy, placental hormones, 4 of them, and 3 hormones increase in manifold, and that is prolactin, cortisol and thyroxine. These are the 3 hormones that increases in amount. And then you have 1 hormone releasing from the ovary, and that is known as relaxin. And that is known as relaxin. It relaxes the ligaments. Please remember, that is released by ovary, not anything else. Okay, ma'am, you are a hard worker. Thank you so much. You guys are also hard workers. And with that, I end the live. As I've told you, I'll give you 2 video links for Brahmastra of evolution as well as reproductive health. Please watch it. Tomorrow you will be having part 2 video of this particular thing, but it is a recorded format, so you can directly watch it. It will be long, but definitely helpful for you. Okay, so that's all. I hope you guys have subscribed to the channel. Wishing you all the best. We have done a long, oh my god, it's not even completing. Okay, long video out there, and I hope this was helpful for you. Wishing you all the best. Lots of love. Shloka, ma'am, salute to you. Although I'm not a bio student, but I get all your efforts. Thank you so much. That is very sweet of you. Mansi, thank you so much, ma'am. You were energetic till now. You stood for us. I also appreciate your hard work. Thank you so much for being there. Thank you, Surbhi. That's so sweet. Okay. So, my dear children, I'll end up the live right now. I'll be sending you the link. Please be there for tomorrow's session. Tomorrow is not live. It's recorded. Watch it. Okay? Wishing you all the best. Lots and lots of love and biggest hugs to all of you. Bye-bye. Take care. You're most welcome, Zainab. Thank you, Shweta. Thank you, Vikas. I'm all hearing you all. Okay. Bye-bye, Mansi. Thank you so much. You too. Thank you, Lakshmi. Everyone, thank you so much.